Hello, everyone. My name is Audrey Luby. I'm a PhD student under Charlie Martin, Mike Allen, and Laura Reynolds. And today I'll be talking to you about fish sounds. Click. Fish sounds have unique benefits as a sensory cue underwater. Acoustic cues can often travel further than other types of cues, and their propagation does not rely on water clarity or light levels like visual cues, nor is largely dependent on water movement like chemical cues. It therefore makes sense that many species of fish would evolve the ability to use sounds to communicate. Click. There are two loosely defined types of fish sound production that I'll talk about today. Active fish sounds, like the calls of the local Gulf toadfish, are produced deliberately in association with a particular behavior or situation, frequently with specialized sonic organs or structures, and are generally used for communication. You can think birdsong as an analogy. Passive sounds, like the splashing of mullet or chewing food, may not be associated with specialized sound producing physiological structures, nor with specific behaviors or situations, though they may still serve some signal function. Unlike with active sounds, it's assumed that all fish can produce passive sounds in certain contexts. Click. Fish are one of the largest groups of sound-producing vertebrates and have the greatest diversity of sonic organs. Fish have been shown to produce active sounds in every behavioral context, including feeding, reproduction, aggression, defense, and social interactions. Other organisms, including dolphins, birds, and even invertebrate larvae, may use fish sounds as a sensory cue to hunt prey, avoid predators, or find suitable habitat. But because of the lack of awareness of fish sound production, research into them has been deprioritized in comparison to other sound producing groups like birds and marine mammals. Click. The production and detection of fish sounds and their associated functions can be impeded by anthropogenic impacts such as habitat degradation, changing climatic conditions, and noise pollution. But it has been difficult to assess the population and community level effects of human activities on fish sound production due to a need to understand more completely how prolific fish sound production really is and the when, where, and why it happens. Click. Fish sounds also have remote sensing applications through the use of passive acoustics. Fish sounds in particular can be and have already been used to detect and monitor spawning for fisheries management, to map the spread of invasive species, and to assess habitat complexity and degradation, among other applications in marine, brackish, and freshwater environments. Despite these evident advantages, and you may be sensing a theme here, the use of passive acoustics to monitor fish populations has been historically constrained by the lack of a global survey of known soniferous species and an accessible library of sound recordings to identify fish sounds as already exists for other taxonomic groups. Click. So I set out to address these critical gaps in information related to sound production in fishes. To find all of the references that included any examination of a fish species for sound production and basic information on the species that was studied, I conducted a systematized review. I looked through almost 3,000 potentially relevant references spanning over 60 countries and written in 11 different languages. They covered the years 1874 through 2020 and included journal articles, dissertations, abstracts, and even a music record from the year 1952, my personal favorite. I assessed these references for relevance and excluded about 2,100 references. That means 833 references were relevant to my research objectives and qualified for data collection. Click. So in reading all of those 833 references, I found 1,185 unique fish species that have been examined for sound production. Of those, only 124 were not shown to make sound when studied, though they may still be shown to produce sounds upon further examination. Regardless, that still means there are records of 1,061 species that were shown to make some type of detectable sound. Of those, 989 species made active sounds, 141 made passive feeding sounds like chewing, and 141 were shown to make other passive sounds like splashing or digging. And not only do I have these total numbers, which could be interesting in their own right, but I also know exactly what species were reported by which studies. This opens up a world of possibilities for studying global trends of fish sound production. Click. I chose to look at the global distribution of actively soniferous fish species using native species data for FAO fisheries designated areas from FishBase to create this bubble map. The larger and brighter the circle, the greater the number of actively soniferous fish species. At least one actively soniferous fish species was found in every region except for around Antarctica, likely more due to a lack of effort. So those 4 regions only had 2 species that were even examined for sound production. The majority of the other regions had at least 100 actively soniferous fish species. So actively soniferous fishes have been found around the world except for Antarctica, at least so far. Click. On the more local level, the Western Central Atlantic and Gulf of Mexico was actually the area with the most known actively soniferous species. I used my data set to find out that our local Suwannee River Estuary is home to at least 75 fish species that produce active sounds, which is roughly 25% of the total number of fish species in the area caught by the Fisheries Independent Monitoring Program. Click. I also wanted to look at how well these species were distributed taxonomically. This phenogram shows the species that were examined for sound production in my review. I was able to map whether the species were shown to make active sounds in blue or not in black, and I labeled the fish families with the most actively soniferous or non-actively soniferous species. Actively soniferous fishes were spread across 132 families and 33 orders. While there are clusters of non-actively soniferous species, they are found in several different taxonomic regions, and actively soniferous species are found throughout. This demonstrates that active sound production is not limited to a select few fish families, as some might expect, but is in fact a taxonomically prevalent behavioral trait in fishes. That being said, only 3.5% of fish species have actually been examined for active sound production, either auditorily or physiologically, and only 2.8% of fish species have been shown to produce active sounds. This dearth of information is important to recognize, as independent evolution and subsequent loss of active sound production have been widely documented across the diverse array of fishes, making assumptions of sonifery based on related species difficult compared to other groups like birds or marine mammals. So, though the taxonomic distribution of active fish sound production is extensive, far more studies are needed before we have a clear idea of just how many fish species make active sounds. Click. While I was able to conduct my initial data collection on such a large scale, I did have to limit the complexity of the data I could collect to very simple categories. So thanks to the work of my amazing collaborator, Santiago Bravo, we were able to conduct a follow-up data collection. Santiago collected data on a subset of the 353 marine subtropical fish species that produced active sounds reported in peer-reviewed articles. He recorded information on a variety of topics, including the testing environments used to study the fish, what types of behaviors were associated with sound production, and the different terms used to describe the sound they produced, for which we found 68 different terms. My favorite word was the so-called boop sound produced by some toadfishes. All of this descriptive information can be used to identify potential species producing an unknown sound in a particular passive acoustic recording, as well as to study fish sound production behaviors more generally. Click. While we're still exploring the data, we did find that more marine subtropical fish species produced active sounds associated with distress compared to other behavioral contexts like territorial interactions, reproduction, or competitive feeding. This was true for the total number of species regardless of testing environment, shown in the graph on the left, and also when species were tested in captivity or in semi-wild environments like net enclosures, shown in the graph on the right. In the wild, however, more species produced active sound during territorial interactions, with distress and reproduction close behind. Part of this trend may be due to the ease with which distress can be simulated through common techniques like waving a net at a fish, but this still demonstrates the importance of active sound production in fishes related to distress, in addition to these other behaviors. Click. So I've now shown a few of the results we found by looking at our compiled data sets, but these data sets have the ability to provide many other insights into fish sound production beyond what I've talked to you about already. There's a few other examples. We are already using the data to determine the habitat types actively soniferous fishes are found in, to highlight how understudied passive fish sounds have been compared to active sounds, to point out species requiring further testing due to uncertainties reported in their examination, and to learn about the influences of testing methodology in studies of fish sound production. And while it has been fun using the data to learn more about fish sound production ourselves, the primary goal of this work has always been to make the data publicly available as an information source that can be used for future research efforts and to engage the public. Click. So I and my amazing collaborators are now in the process of building a website to make all of the data we have collected easily accessible and searchable, as well as to begin compiling fish sound recordings and acoustic measurements that can be used to identify fish sounds from recordings. Users will be able to search by taxa, region, water type, acoustic measurement, sound type, publication, and other information. The creation of this fish sounds library will facilitate new research on fish sounds, assist in remote monitoring applications, and expand the ability for more complex analyses of trends in fish sound production. The Fish Sounds website should be available by the end of the year, if not sooner. So hopefully by the next FAS GSS, you type fish sounds into your favorite search engine, our website will pop up and you'll be able to explore the data yourselves. Click. Click, Trent, please. Oh, I had 2 more slides, but it was just the acknowledgements. I want to acknowledge all of the people and institutions who have been involved in this project so far. It has truly been a collaborative effort across 3 countries and several institutions. We have also received a lot of help and advice along the way, and I'm personally just so grateful to all of the amazing people I've gotten to work with and learn from so far. Thank you also to Trent for clicking through my presentation for me. With that, one last click. I'd be happy to take any questions now, or you can feel free to email me. Thank you.
So do we have any questions for Audrey? We have a few minutes left.
Audrey?
Hi.
Thank you for there. In your research, have you done any work with the U.S. Navy? Because they have some of the best years in the ocean you can imagine for those submarines.
Yes.
And necessity makes uses to get you money. And, you know, maybe some of this is be important to them eventually too.
Yeah, that was one of the really interesting things for me to find out in this study was that the Navy was actually the primary ones funding fish sound production research when it started around the 1950s in earnest. They actually funded a lot of projects because they would hear sounds while they were using the submarines, and then they wouldn't be sure if it was animal noises or actually made by enemy ships. So a lot of the work that was done in the 1950s up to the 1970s, identifying fish sounds was actually done with funding by the Navy. And they've continued to do a lot of soundscape research even to today. I haven't personally worked with them yet, but it definitely presents exciting funding opportunities for me in the future.
Thanks, Audrey. If there's time, I'll ask a really quick question. That's awesome. I still remember my first snorkeling trip in Australia and listening to sooty grunters and just like the immense sound underwater. Simon Linke has done some really interesting work at Griffith, right, on this work. Question, how many of your studies, or do you recall how many of your studies actually showed how anthropogenic factors are influencing the soundscape or basically the ability of fish to make these sounds?
Yeah, that's been a really increasing trend lately, and some of my co-authors have actually published some really cool reviews and meta-analyses on that recently. So mostly it's shown like anthropogenic noise pollution can have a lot of impacts through masking, or by causing direct physiological stress, or by causing the fishes to have to change their pitch to produce sounds, which might cause additional strain. There was actually a really exciting article published in Science that one of my co-authors was on. I believe it was Sounds in the Anthropocene. It was in the previous issue of Science, but I highly recommend reading up on that. It covers a lot about noise pollution just in the ocean in general.
Cool, thanks.
Yeah.
Great, thank you, Audrey. We're gonna get moving on to the next presenter, who is Adam Searles. and he is advised by Dr. Martin. And here we go.
Hi, everyone. My name is Adam Searles. I'm a first-year PhD student with Charlie Martin and Laura Reynolds, and my research currently focuses on the dietary overlap between Western Atlantic sea bream and sheepshead in the northern Indian River Lagoon. Click. So, as we all know, climate change is facilitating the poleward spread of many tropical marine organisms, and these tropical organisms are having profound impacts on recipient ecosystems. However, herbivores have been seen as having the most profound effects, as they can, one, compete with local herbivores for food and space, and two, actually cause ecosystem-wide cascades by grazing directly on local foundation species such as seagrasses and macroalgae. And this is relevant to us because, as you can see by these data collected by Florida Fish and Wildlife's Fisheries Independent Monitoring Program, tropicalization is likely happening in the northern Indian River Lagoon. The hotspot analysis on the left indicates that Western Atlantic sea bream have expanded significantly northward over the past 20 years, and that this northward spread was associated with a 49-fold increase in the abundance of this tropical herbivore. Simultaneously, we've observed significant declines in their sister species, the local sheepshead, with 2019 accounting for the lowest recruitment as well as the lowest catch per unit effort of adults ever recorded in the Indian River Lagoon. And while it's obviously hard to infer anything from two trend lines, the rise of sea bream is a concern for a few reasons. One, the sheepshead of the northern Indian River Lagoon, unlike everywhere else in their range, are actually herbivorous. They feed predominantly on seagrasses and macroalgae in this area. Secondly, the northern Indian River Lagoon historically has been a seagrass-dominated system. However, in the past few decades, they've lost up to 97% of historical cover. So the rise of a tropical herbivore in this area may spell disaster for whatever seagrass is left, as well as restoration efforts currently going on in the area. Click. So based on this, we want to know, click, does the tropical sea bream display dietary overlap with local sheepshead populations? And click, does this facultative herbivore consume seagrasses in the northern Indian River Lagoon? Click. So I saw gross gut contents, now you do too. To answer these questions, I conducted gut content analysis by excising stomachs, identifying all contents down to lowest possible taxonomic level, and then I measured the mass of each content and used percent mass of each content to analyze the data. So here on the left, we have red drift algae, click. Then we have a chloropa rhizome, click. Then haliduli, down at the bottom, click. Then we have a brittle starfish, click. And then we have some green hair algae, click. And despite visual similarities between all these items and diets, there was a statistical difference, click. Permanova revealed significant differences between the diets, and to investigate these differences further, I used a canonical analysis of principal coordinates, which is an ordination method similar to an NMDS. So the farther apart 2 points are, the more different they are. And as you can see by this plot, there really isn't much to see. There was a lot of overlap in these data, no clear patterns or trends or clustering, and even our item vectors really didn't reveal any clear patterns. So look at the actual items, click. I used a similarity percentage analysis. And what this does is it actually identifies the items that contributed most to the significant difference between the diets. So with average proportion of mass on the y-axis, the item that contributed to the most was red drift algae, followed by green hair algae, unidentified mass, which is common in gut content analysis, and then haliduli. So looking at red drift algae, we can see that sea bream ate more than twice of as much red drift algae as sheepshead did. However, I think it's important to note that sheepshead also ate more red drift algae than anything else, meaning that there's likely a lot of overlap going on and a pretty high potential for competition. Furthermore, if we look at haliduli, which is the fourth item down there, haliduli is the most common seagrass in the Indian River Lagoon. Yet neither species really ate that much. Sea bream ate the most, but only 10% of their diet was made up of seagrasses. However, this might be due to the fact that there really isn't much seagrass left in the Indian River Lagoon, and their distribution is pretty patchy. So we need to be careful when interpreting these data. Thank you. Feel free to email me any questions.
Great. That was right on time. Well, good job. We're gonna move on to the next one, which is Hannah Gottesman, advised by Dr. Will Patterson and Dr. Laura Barbieri.
Okay, awesome. So hi everyone, I'm a second semester PhD student, and I'll be outlining kind of the current status of biophysical modeling of larval dispersal for gag grouper in the Gulf of Mexico. But before I get into it, I'd like to acknowledge my collaborators listed here, and then also the members of FWRI's Movement Ecology and Reproductive Resilience Lab for all of their tireless field work to help me get the information I've needed. Click. So in 1992, a shipping container of rubber ducks dropped tens of thousands of bath toys into the ocean. Here's some clips from coverage of that event. And I'd like you all to imagine if you were tasked with recovering these ducks, how would you find them? Click. So to do this, you might want some information about the ducks, how many of them there are, their size, how long they can float, where were they dropped. You might also want some information about the ocean conditions they'll experience, so the currents. And ultimately, you'd like to be able to map where these ducks end up. Now, I know you're probably asking, what does this have to do with gag grouper? But actually, an approach very similar to this has massive utility for their sustainable management. Click. So for some quick background on gag, they're found in the eastern Gulf of Mexico, and they're an economically important species for both commercial and recreational fisheries. They also serve some pretty important ecological roles. However, their stock status is pretty uncertain, and it appears that the gag population in the Gulf may actually be experiencing some trouble. So to better understand their resilience under fishing and also potential environmental disturbance, it's vital to account for their interesting and complex life cycle. Click. So gag exhibit a pretty complex life history. They're protogynous hermaphrodites, which means they actually begin life as females and then ultimately transition to male later in life. They also exhibit a complex set of migrations over the course of their life. So larvae settle in estuarine habitat in West Florida before transitioning to nearshore habitat as juveniles. In the winter, mature females migrate to offshore spawning sites where males remain all year. Click. So here we have a visual depiction of that cycle for your reference. And to really get at the processes driving kind of reproductive success and population connectivity for gag, one area that needs more analysis is the connectivity of spawning sites to nursery habitats. So from the offshore sites to nurseries in estuaries, and trying to quantify the relative contributions of these sites and how much fishing they can withstand while remaining productive. Click. So now that brings us back to the ducks. To better understand gag larval dispersal, we actually use a framework, a Lagrangian framework, in which you drop particles into a 3D velocity field and can then track their movement based on certain characteristics of the particles. So like the ducks, we drop the particles into a simulated ocean. However, unlike rubber ducks, these particles can actually be released at depth and exhibit unique behaviors and traits. So to do this, we use the connectivity modeling system developed by Paris et al. And we populate that with oceanographic information from the HICOM model, which assimilates daily satellite altimetry, CTD casts, and Argo float data. Click. So to outline how biophysical modeling actually works, this is a schematic. Of course, you can ignore most of the details, but what I want you to get from this is that you introduce behavioral and biological traits for the particles. You also input environmental conditions from the HICOM model. Running these together, you then generate a multi-year model that serves as sort of a predictive index of reproductive success and resilience. So it's a way to try and get at future recruitment of gag. Click. So for behavioral and biological traits, I've included a table here with what was used in the earliest model for gag in the Gulf in 2013, and then the updates that we've made as part of the working paper that I'm working on. So as you can see, a lot of this, these data are actually derived from other species of grouper, but we've had to make do with what we can find. For the update, though, fortunately, we were able to deal with kind of the lack of good reproductive data for gag. So I'll outline a few of those things in the coming slides. Click. First, interestingly, we actually found novel evidence that upon introduction to seawater, gag eggs are positively buoyant. So if you'd like to click the video, see if you can get it to play. Thank you. So what you'll see here is eggs freshly sampled from a gag being introduced to seawater just sampled from the surface. And you can see them trailing in sort of like a spiral towards the surface. So it was actually thought previously that gag eggs are negatively buoyant initially, but this is part of an ongoing investigation to better parameterize that. Click. So here we see the sites where we released particles. And this is kind of a complex process to develop this. So first, it was confirmed where gag were caught that were actively spawning. Batch fecundity was determined for these fish. And then using the number of fish and the average fecundity at each site, a relative fecundity could be determined. We then scaled this by the curve of spawning intensity over the peak spawning period, which is February 1st to April 18th. And then release particles at these sites every 4 days for the duration of the peak spawning season in our simulation. Click. Okay, so having done that, we generated these preliminary outputs. You can see that the black release points are shown as well as red settlement locations, and the rainbow trails indicate where gag particles moved over the course of the simulation. I like to call everyone's attention to the bottom left corner of each of these figures. It shows the number of gag eggs or larvae released in the simulation that actually made it to suitable settlement habitat. So as you can see, for the range of our study, 2020 had the highest recruitment for survivorship as a proxy for recruitment. And then 2016, 17, and 18 also showed relatively high numbers. We saw the bulk of that success in the Northern Gulf and off Florida Bay in the south. Click. So this all sounds very nice, but as you can probably guess, this type of modeling is a developing field, and there are a lot of challenges remaining before it can be a really useful tool. I'd like to call your attention particularly to model validation, which in the past decade or so in which this field has taken off has sort of been a neglected component. Click. So I attempted to ground truth our model findings using a 2019 paper by Walter Ingram, in which age 0 gag caught in West Florida estuaries. Their abundance was measured, and I was able to use that for a direct comparison to our results. So looking at just the last few strong year classes, 2016, 17, and 19 from that paper, in comparison to our model, this matched up fairly well. But for further refinement, we'll need to investigate on a finer spatial scale which estuaries are actually seeing success, because Charlotte Harbor tended to drive trends of abundance in this 2019 paper, whereas our model suggests that we should see significant survivorship more towards the north, so around St. Andrew, St. Joe, and Big Bend. Right, click. So of course, there's lots remaining to do, but this is just an overview of some of the ongoing work and also directions that I believe the field needs to take kind of in the upcoming years, with an emphasis on effective ground truthing, and also increasing the amount of data available specific to the species of interest. Okay, click. But I believe through this process of iterative improvement, ultimately, this model can be a very useful fisheries management tool, both for stock assessment by providing a predictive index of recruitment, and also as a tool for planning marine protected areas and assessing their effectiveness by determining which spawning sites are highly connective to estuaries and their relative contribution of surviving larvae. Additionally, to assess whether this model will actually be a valuable tool, I posit that its ability to predict variability in reproductive success is critical. It also must illustrate population structure and connectivity in a realistic manner. And altogether, this will help us better understand the reproductive resilience of gag grouper in the future to help better preserve the population in the Gulf. And with that, I'm happy to take any questions. Thank you.
Great. So we've got about 3 minutes for questions. So if you have any, feel free.
Hey, Hannah, this is Bill Lindbergh. Is there very much literature on ichthyoplankton behavior? The reason I ask is that the blue crab larval behavior was well studied, decades ago, and contributes substantially to their transport back into the East Coast estuaries. I'm wondering if there's something similar that could be factored into your modeling based on the ichthyoplankton.
Yes, definitely. So this is one area where the model definitely struggles. And I think this is the case for a lot of species, but with gag in particular, we just now updated the kind of their vertical distributions as a way to illustrate kind of their swimming behavior. But this had to, again, be based on data that are generated from all epinephrine larvae that were found in a mock nest hoe. So for gag specifically, there's not a lot of information, and I think that's the case for a lot of other fish species, actually. But yeah, to more accurately and realistically depict their movement, I think larval behavior needs to be incorporated much more heavily, because it's actually, in many cases been seen that they can add kind of an element of diffusion that goes beyond just what we think of as the advection of particles. So yeah, thank you for that question.
Thank you.
So we've got 1 more minute. So I think Don Berenger raised his hand, it says in the chat. So.
Hey, Hannah, great talk. I appreciate it. I'll sort of second Bill too. I mean, finding lobsters, that's my thing. And but I didn't do the work, and a lot of folks have done some of that behavioral work too. And it's amazing how it changes the patterns of connectivity when that kind of behaviors are worked in. But I won't belabor that. But I will ask you, when you did your buoyancy experiments with the eggs, did you consider, is there any likelihood that when those eggs are under pressure, that changes their buoyancy?
Yeah,
Like if you put them in some kind of a pressure chamber to test the same thing at depth where they would have been released?
Yeah, that's a great question. So this is an area that I'm very excited about, and it's also very, very new. So like I said, up until a couple days ago, we didn't know what gag eggs would do when introduced to seawater. So that's going to take much more extensive laboratory investigation looking at, I think pressure is an excellent point, as well as the differences between fertilized and unfertilized eggs. Because those were unfertilized, but fertilized eggs, their aquaporins close, so then they may have changes in their density, also varying salinity and temperature to simulate, the differences you'd see throughout the water column.
Gotcha.
We discussed that in my lab group that, in addition to the potential that gag eggs could be maybe stuck under a thermocline, to the north, you might even see like a halocline where there's river inputs. So yeah, it's a pretty challenging question to look into, but I think
a little bit more to do
that
there,
hits
sure.
a critical
But you
point.
considered it. Cool. All right. Well, thanks.
Unfortunately, we have to move to the next presenter, who is Stephanie Winn, who is advised by Dr. Allen, and she has a pre-recorded talk. So I'll go ahead and just click through that for us. Can we hear that, or is it not working?
No, we can't hear that, Trent.
Let me try one more time.
And Econfina estuaries. Hello everyone, my name is Stephanie Winn, and today I'm going to be talking to you about river discharge and the influence on seagrass-associated nekton communities within the Steinhatchee and Econfina estuaries. Many studies have shown that changes in freshwater discharge can impact the diversity of food and habitat resources, such as the abundance of zooplankton and phytoplankton, seagrass, and even oyster reefs. Thus, in coastal estuaries, the input of freshwater discharge from rivers and streams is one of the most influential factors affecting nekton community structure and production. Understanding these effects of the natural and anthropogenic impacts on these communities is vital to the conservation and fisheries management. The objective of this study was to investigate the relationship between freshwater discharge and the effects on seagrass-associated nekton communities within the Steinhatchee and Econfina estuaries. This study utilized Fisheries independent monitoring data collected by FWC from 2008 to 2018, as well as USGS river discharge data. It focused on two estuaries in the northern Big Bend region, Econfina and Steinhatchee. Here we have average annual discharge for Steinhatchee in blue and Econfina in red. As you can see, Steinhatchee has higher overall discharge rates, but they each follow similar trends across the years. Discharge data was broken into categories of high, medium, and low flow years for each estuary. Results from Permanova indicated that community composition varied significantly between estuaries and discharge levels. These differences can be visualized by the bootstrapped metric MDS plot. Discharge levels are indicated by color. High is blue, medium is green, and red is low. The open symbols are Steinhatchee, and the closed symbols are Econfina. There's clear separation between estuaries and between discharge levels for Econfina. For Steinhatchee, however, we can see that there's a lot of overlap between the high and medium flow years. Here we have some results from the two-way ANOVAs. The letters above the bars indicate significance. On the top left, you can see that overall total relative abundance were similar among discharge levels and estuaries. The top right figure shows species richness. For the Econfina estuary, we can see that species richness increases with medium to high discharge. The same pattern can be seen for Steinhatchee, however, these differences are not significant. On the bottom, you can see diversity increases with medium flow years in Econfina, but no significance was seen for Steinhatchee. I also performed ANOVAs for several abundant and economically important species to test how discharge affects individual species. With a quick glance at the overall trends, you'll notice how relative abundance varies significantly across years and discharge levels for each estuary. In conclusion, these results indicate that river discharge is a significant factor affecting nekton community structure within the Steinhatchee and Econfina estuaries. Overall, the trends seen were generally stronger in the Econfina estuary. We also saw that the effects of river discharge on relative abundance varies by species. I would like to take a second just to thank my committee, Mike Allen, Charlie Martin, and Caleb Pertobal, as well as the Cedar Key FIM crew for all their hard work. Thanks, everyone.
Great. Up next, we have Jordan Bajema, advised by Dr. Will Patterson.
Hi, I'm Jordan Bajema. Today I'm going to talk to you about estimating nursery sources for common snook along Florida's nature coast. For those of you that are unfamiliar, the common snook is a large popular game fish. They're a Eurohaline species, which means they are found in both salt and fresh water, and they're commonly found throughout the southern peninsula of Florida. Next, click. So snook are thermally limited in range. They can't survive temperatures lower than 12 degrees Celsius for an extended period of time. And historically, this was about the 28-degree north latitude, or roughly the Tarpon Springs area. But in recent decades, they have been found increasingly more common north of that line up to the Suwannee River Estuary. It's commonly thought that this is attributed to rising temperatures, or more specifically, fewer days below that lower lethal temperature limit. Click. So FWC's Fisheries Independent Monitoring Group has historically collected samples in the Suwannee River Estuary. And with time, they've started noticing an increasing level of snook caught in their samples. So if you look from the 97 to 2006 histogram, one snook was caught. But then you go to 2007 to 2011, and 47 fish were caught. And then 2012 to 2015, 127 snook were caught. Finally, from 2016 to 2018, 450 snook were caught, and this represents an exponential increase in numbers. It's important to note here the size structure of these catches. Initially, it was predominantly large fish that were caught, but as we move forward in time, more of those smaller size classes were represented in the catch. This is important because those smaller size classes are indicative of local recruitment to the stock. Click. So my research aims to find what proportion of these expanded range snook are locally produced versus what proportion are immigrants from other systems. And then importantly, what were those sources of those immigrants, and have the relative importance of their contribution changed over time? So traditionally, to track fish movement, you would stick a tag in it and hope to catch it later on. But as you can see from this picture of a juvenile snook, they're pretty small, so you can't use the traditional tagging method. So what we're going to do is use a natural tag. Click. So natural tags are going to be developed using otolith analysis. We've collected juvenile snook from Cedar Key all the way down to Charlotte Harbor. We're going to take otoliths out of those snook. We're going to clean them and we're going to pulverize them. And then initially, we're going to run them through an isotope ratio mass spectrometer and look at the delta carbon-13 and delta oxygen-18 levels of those fish. Now, hopefully what we'll see is something like this middle figure, where each nursery source is well defined and you could easily assign a fish to a nursery. However, we could see something like the top figure, where the nursery sources are not well defined and you have a hard time accurately assigning a nursery source. So if that happens, we're going to take that otolith material and we'll run it through a different kind of mass spectrometer called an inductively coupled plasma mass spectrometer. And we're going to look at element to calcium ratios of barium, lithium, magnesium, manganese, and strontium. So with these new chemical signatures, along with the isotopic ratios, we'll plug those values into what's called the discriminant function. And what comes out of that, hopefully we'll look at like this bottom figure, where each nursery source is well defined. You could easily assign an individual to a nursery source. That is what's known as the natural tag. It's these signatures from the chemicals and the isotopes. And then once you have that natural tag developed, you can take an adult snook otolith and you can take the core out of it, run it through the same machines, and then put those values into the discriminant function, and that will predict a nursery source. Click. So we expect the otolith chemical signatures are going to provide a robust natural tag, and we'll be able to easily identify the nursery source for individual snook along the west coast of Florida. It's more specifically in the Suwannee River Estuary. We expect to see more local recruitment of the adult snook over time. And finally, the management system must account for fisheries and ecological implications of these increasing snook populations in this system, whether that be competitive interactions with other game fish or population dynamics questions with a new self-recruiting population. I'd like to thank my funding sources, UF IFAS, Nature Coast Biological Station, Florida Fish and Wildlife, and Z-Man Fishing Products. Thank you.
Great, thank you, Jordan. Up next, we have Elizabeth Walsh, advised by Dr. Tuckett and Dr. Hill.
Hello everyone, my name is Elizabeth Walsh. I am a first-year master's student here at the University of Florida, and I'm currently preparing to have my proposal finalized. The focus of my research will be comparing behavioral and morphological characteristics of both the eastern and western mosquitofish, along with my advisors Quentin Tuckett and Jeffrey Hill. Eastern and western mosquitofish have been routinely introduced around the world for the controls of mosquitoes since the 1900s. These two species were long considered to be just one, and the identity of introduced populations, especially those noted before 1990, is often unknown. Thus, mosquitofish have one of the largest ranges and longest record of ecological impact of any introduced fish. Click. Eastern and western mosquitofish are very similar morphologically and can be difficult to tell apart in the field. The two species were considered one of the same until they were split in 1988, where they were now considered two separate species. Mosquitofish are piscids, or live bearers, and males possess a gonopodium. This gonopodium is a modified anal fin used for reproduction and is the simplest way to differentiate between these two species. Western mosquitofish have flat teeth on the bottom of their gonopodium, while eastern mosquitofish have sharp edges, as seen on the picture to the right here. Click. The introduction of mosquitofish, both eastern and western, has been viewed as an attractive alternative for insecticides globally. However, mosquitofish are highly aggressive, and they have the ability to destroy small-bodied fish, frogs, and other aquatic organisms in areas which they are introduced to. This aggression has actually led them to become part of the IUCN's top 100 invaders, and this was the only listing I could find in the IUCN where two species were lumped together into one listing. The map here shows both the native range and invasive range of the mosquitofish, and as you can see, populations have spread globally. Click. Invasion success isn't always a guarantee though. A recent study in Japan, pictured here to the left, showed that male guppies have the ability to remove western mosquitofish populations through reproductive interference. This interference has caused an overall decline of the western species for this particular area. However, these results are a contrast to the study performed by Dr. Tuckett, Dr. Hill, and their colleagues. Their study utilized the eastern species rather than the western, and it was concluded that guppies have limited survival and growth when in the presence of eastern mosquitofish. In their study, both low and high densities of the eastern species had detrimental effects on guppy populations, and the results can be seen here on the right. This variation in guppy survival and growth may be due to the fact that these two species differ in fundamental ways, including their behavior. The eastern mosquitofish could possibly be deemed more aggressive, which in turn would increase their invasiveness when introduced to new areas. Click. The goal of my research is to determine if it is acceptable to lump these two species together or to note any differences between the populations. Populations of each species will be sampled across the native range along the same latitude, as pictured here in the following map. I will determine if invasiveness varies among species and populations, as well as find the ultimate and proximate causes for this variation, if it is present. Click. Collected fish will be transported to the Tropical Aquaculture Laboratory in Ruskin, Florida. Habitat analyses from source populations will be recorded, and mesocosm trials will be performed. These mesocosms will be established with a community of small-bodied fish prior to the introduction of mosquitofish in order to properly assess invasiveness. Photographs will be taken of each fish collected, and geometric morphometric analyses will be utilized to identify any morphological differences among populations. Behavioral analysis will also be performed with both aggression and boldness recorded through aquarium trials. These methods will highlight how much variation exists, if any, among mosquitofish populations in terms of invasiveness, morphology, and behavior. Click. Thank you to the members of my committee, Quentin Tuckett, Jeffrey Hill, and Lindsay Reisinger, for assisting me with my studies. And thank you to the faculty, students, and staff at the Tropical Aquaculture Laboratory. All papers used for this presentation have been cited here, and I thank you all for your time.
So hi, my name is Kaitlyn O'Neil. Today, I'm going to talk to you all about developing a robust index to describe ovarian condition in captive adult red drum, Cyanops orphalatus. I work for the Florida Fish and Wildlife Conservation Commission at the Stock Enhancement Research Facility, which is within the Fish and Wildlife Research Institute. And I've been lucky enough to have been granted permission to use some of the work that I'm doing there for research on my master's thesis. Click. So red drum is a species of interest to us because it's an iconic inshore sport fish that supports a major fishery. The goal of stock enhancement in regard to my work at the Stock Enhancement Research Facility is to manage, conserve, and restore marine sport fish using the responsible approach that was updated by Lorenzen et al. 2010. We aim to do this in part by optimizing spawning of our broodstock through identifying spawning conditions and optimizing spawning quality. And one piece that we're currently missing is a clear description of oocyte maturation in our captive red drum, which would aid in optimizing the spawning process. Click. For a bit of background, oocyte biopsies were collected in 2015 and 16 during regression, regeneration, and spawning, and they were preserved for staining and histological analysis. The goals of this analysis are to classify the reproductive condition of the ovary, describe oogenesis during captive maturation and spawning, and then ideally to create a tractable tool with direct application in red drum hatchery management, as well as a broader relevance for finfish aquaculture, especially when evaluating novel species for stock enhancement. Click. We knew we wanted to be able to quantitatively assess our histological images, but we also knew that bias could be introduced due to the large size difference that exists among the stages. So we had to develop a method that could mitigate possible bias, and assumption-based stereology is prevalent in literature, especially for determining fecundity in fish. It provides a way to estimate the quantity of structural features of an object using a series of two-dimensional images. So this is the direction that we decided to take by applying this method and analyzing each oocyte developmental stage individually, so that we could then evaluate the ratios of the stages over the maturation and spawning phases of our fish. Next. So this is an example of a single field of view from one oocyte biopsy with several stages of oocyte development. And the way that assumption-based stereology works is that each image or field of view is analyzed in two ways. Click. In the first analysis, a Weibull grid is overlaid onto the image, which is represented by the fine yellow line segments. The endpoints to each line segment are considered the points on the grid. Click. So these points are then labeled with the stages of oocyte that fall directly underneath. We're using a staging schema created by Greer et al. In 2009. Now, according to the DeLisi principle, the volume fraction of an object, so in this case, it's each oocyte stage, which is examined individually, is proportional to its area fraction in a cross-section, and that's ascertained by these point counts. Click. The second analysis of the image ignores the yellow line segments and instead uses the red and green borders, which are considered the counting frame. Oocytes of each stage are counted so long as they lie within or touch the green borders of the counting frame. Any oocytes touching the red borders of the frame are excluded from the count. And the counting frame and its rules provide unbiased counts according to literature. Click. So after each image is analyzed in these two ways, we have a volume fraction and an unbiased count for each oocyte stage. This data is then applied to a stereological equation shown here, which results in the number of developing oocytes in a given stage I. The equation also includes two coefficients that take into account the geometric shape of the oocytes in order to represent the three-dimensional structure for a two-dimensional image. So once the equation is applied for each oocyte stage, the quantities can be compared throughout maturation and spawning phases using ratios. Click. In these photos, you can see some of the variation throughout maturation leading up to spawning, and eventually atresia is evident, like in the lower right-hand image. Again, we're hoping to then be able to use the ratios that we gather to classify the reproductive condition of the ovary and describe oogenesis during the captive maturation and spawning schedules. We're also thinking we might be able to use this histological analysis as a proxy for real-time wet mount biopsies in a practical hatchery setting, not just for red drum, but also for other finfish. Click. So here's the work that I referenced, and just a big thank you to those that have made this work possible, and then for all of you for listening.
Great. Thank you, Kaitlyn. Up next is Keel Condy, who's advised by Dr. Bill Patterson.
All right. Hello, everybody. So like Trent said, my name is Keel Condy. I'm a first-year master's student advised by Will Patterson. I'll be presenting my research on some snook range expansion effects. I'd like to acknowledge my other co-authors for their contributions to this project as well. Click. So with an upward shift in global temperatures, we're seeing a decrease in the number of cold days in the eastern portion of the Gulf of Mexico. And these cold days are an important range limiting factor for some species like common snook. Historically, in this portion of the Gulf, snook range up to just north of Tampa Bay around Tarpon Springs. But in recent years, there's been increasing numbers of snook caught up towards the Nature Coast region, including Cedar Key and the Suwannee River Estuary system. So we want to know how this new introduction is going to affect other species in this system, namely spotted sea trout and red drum. These species occupy a similar ecological role as snook, being large predators in this estuary, and both are key fishery species with economic importance to this area. Click. So the objectives of this study will be to delve into the dietary habits of these three sportfish species and really look at how the diets and ecological niches overlap between each other. So to investigate their diets, we'll look into gut contents of the species, and for trophic niche determination, we'll be using muscle tissue stable isotope analysis. Through looking at these factors, we can get a better understanding of the interactions taking place in these estuaries and see potential competition or predation effects between them. Click. So to accomplish this, I'll be sampling with Florida Fish and Wildlife's Fisheries Independent Monitoring Program, or FIM, who conducts monthly surveys in Cedar Key and other areas of the state. So fishes harvested through FIM are going to have muscle tissue collected and whole stomachs removed, which will then be used, as previously mentioned, for stable isotope and dietary analyses. Click. So for the diet analysis portion of the project, we'll be taking the whole removed stomachs and preserving them and their contents in ethanol, and then later be using a dissecting microscope to itemize and sort prey and then dry and weigh each item. These prey counts will then be used to test for differences in the diets among size classes and for each species by percent mass, percent frequency of occurrence, percent number, and percent index of importance. And this data is going to give us information on the sampled fish's most recent meals before collection, give us a better idea of what exactly they're feeding on in that environment and how that differs between the species in question. But it's not going to give us information on their trophic niche. Click. So for that, stable isotope analysis will be the other part of my research, and it's going to complement the diet data in that area. So sampled muscle tissue is going to be dried and analyzed using stable isotope mass spectrometry. And by using muscle, we'll get an integrated signal over about the past 6 months due to the slow turnover rate of this tissue. So we'll get a more long-term insight into the fish's feeding habits. And then from the spectrometry data, we'll be looking mainly at two constituents of the tissue, the first being nitrogen 15 to 14 ratios. Nitrogen 15 is enriched in organisms through their diet by around 3.4 per mil per trophic step versus their prey items, leading to higher ratios in higher level consumers and predators. So by determining this ratio, we can see at what trophic level an individual is feeding at and how that might compare to our other species of interest. And then the other component of analysis will be the ratio of carbon 13 to carbon 12. And this varies less across trophic steps and more through the source of the energy that the consumer is feeding on. So with variability of carbon isotope ratios in the basal resources, we can determine whether species are feeding within similar trophic pathways or if their diet comes mainly from different sources. This figure down here shows a kind of idealized food chain where you have these stepwise increases in isotopic values to track the movement of energy throughout the system. With greater relative values of nitrogen 15, you can see the progression from basal producers to upper level predators feeding along a single trophic pathway. And that pathway is shown by the kind of linear increase in carbon isotope ratios across these different steps. Click. So the results from these analyses will have use in further studies to parameterize ecosystem models for the Nature Coast area and example potential ecological effects in that region of further population increases in snook. So the introduction of snook to this area as temperatures continue to increase will present a growing target for anglers along the Nature Coast, but potentially at the expense of some of the other key historical fisheries in that area. Finally, I'd like to acknowledge these institutions for providing funding and for their support of this project. Thanks.
Great, thank you, Keel. Up next, we have Savannah Gandy, also giving a 5-minute talk, and she is advised by Dr. Barry and Dr. Josh Patterson.
Hello, my name is Savannah. I am a master's student. Today I'll be discussing one of 3 parts of my thesis, macroinvertebrate and fish response to major weather events in Clearwater Harbor, Florida. Click. The data for this project comes from a long-term sampling set from Clearwater Marine Aquarium. The amount and temporal scope of the data provided a way to look into multiple major weather events. Climate change is expected to enhance these events, making them more intense and frequent. So understanding how animals respond to them is important, especially in coastal marine environments that are susceptible to rapid environmental changes associated with major weather. So main questions for this project were, is there a sunless change in time periods before, during, or after these events? Is seagrass providing refuge from these events, and which species are responding? Click. So a little overview of the study area and sampling effort. Clearwater Harbor is tidally connected to the Gulf of Mexico. It's mostly composed of sand, mud, and seagrass habitat. And for anyone not familiar with the area, it is north of and interconnected to Tampa Bay and intracoastal to Clearwater Beach. Clearwater Marine Aquarium has been conducting otter trawl net pools on a public eco tour called Sea Life Safari for several years. And being a prominent tourist destination, tours go out nearly every day and sample sometimes multiple times per day, which means a lot of samples were collected. And this particular project uses over 10,000 samples from 2009 to 2019. So the sampling effort allowed for a somewhat unique situation in that samples were collected sometimes very close to extreme weather events and sometimes during them. Click. 6 events were looked at: a cold snap in 2010, tropical storms Debbie, Andrea, and Colin, and hurricanes Hermine and Irma. To determine response, impact dates were identified as dates closest to where the event was near Clearwater Harbor. And in most cases for the tropical cyclones, this was the landfall date. For the cold event, I considered this the entire duration of the event. From there, samples were divided into time periods before, during, and after the major weather event, and then assemblages were tested for variation among these time periods using Permanova. Seagrass was also tested as a factor to see if it was contributing to the time period variation. And out of the 6 events, Tropical Storm Andrea and Hurricanes Hermine and Irma had a response. And for Tropical Storm Andrea and Hurricane Hermine, this was only among the event time periods, but for Irma, this was marginal with an interaction for the event time periods and seagrass cover. Click. The variation in event time periods can be seen in the cat plots here for Tropical Storm Andrea and Hermine. Closer triangles indicating more similarity. Pairwise tests and similarity of percentages were then used to identify which time periods and which species were contributing to variation respectively. So highest contributing species are overlaid on the plots. For Tropical Storm Andrea and Hurricane Hermine, the most differences were found between time periods before and after the event. For Andrea, pinfish, pigfish, Jenny Mahara, and grass porgies were the species responsible for the most variation. And for Hurricane Hermine, pinfish, pigfish, and Jenny Mahara contributed to the most variation. Looking at average abundances for these species before to after the event, Tropical Storm Andrea had decreases in average abundance, but Hurricane Hermine had increases. However, for both, pinfish and pigfish showed increases during the storm. Click. For Hurricane Irma, variation in assemblages was found to be from sample locations with less than 30% seagrass coverage. Pinfish, pigfish, and Jenny Mahara and lane snapper were the species that contributed to the most variation. Unlike the other storms, though, the average abundances increased for some species but decreased for others. And then pinfish kind of spiked again like the other storms during the event, along with Jenny Mahara. And looking further into the variation in seagrass cover across event time periods, diversity, richness, and abundance increased in locations with 30% seagrass coverage during the time periods closest to the storm, suggesting that there may have been some movement towards seagrass habitat, potentially as refuge when there was peak storm intensity. Click. So putting this all together, response to major weather events is dynamic and varies among events. Assemblages responded to 3 of the 6 major weather events investigated in the study, notably the 3 strongest storms. However, response was not uniform. The lack of responses for other events suggests a level of resilience in the species study. The role of seagrass habitat also varied, being unimportant for most events except for Hurricane Irma, the strongest and closest storm to Clearwater Harbor, suggesting that shelter use may only be important during very close or very powerful events. And finally, the 3 most common and abundant species collected in the study appear to be good indicators for major weather. Overall, this variation is reflective of the naturally dynamic nature of coastal marine systems. Understanding response, especially levels of resilience and the importance of habitat, will become increasingly important should these events intensify as expected. And of course, a big thank you to my committee and Clearwater Marine Aquarium for this information.
Great, thank you, Savannah. Up next, we have Derek Chamberlain, who is advised by Dr. Will Patterson and will be giving a 15-minute talk.
Yeah, awesome, Trent. Thank you. As you mentioned, my name Eric Chamberlain. I'm in Will Patterson's lab. I'm going to be presenting derived estimates with islands radiocarbon. Click. So gray triggerfish are a very common reef fish in the northern Gulf of Mexico. They're targeted pretty heavily in both recreational and commercial fisheries. And they've experienced historical overfishing and were actually listed as an overfished stock in the early 2000s. And while some steps have been taken to reduce the exploitation of gray triggerfish and help to rebuild the stock hasn't been recovering at expected rates. So that sort of led us to the question of why we would have this mismatch between our expectation from the stock assessment and our experience with the actual measurements of the abundance. Click. So one potential mechanism for this is the misestimation of age in gray triggerfish. Age is a really important parameter in stock assessment. It's used to estimate really important rates such as mortality and growth. And currently, gray triggerfish age is estimated using section dorsal spines. Now, there's some associated issues with estimating age with these dorsal spines. We know that there's a really high average percent error, which measure of the precision or the reproducibility of age. So we haven't done any validation studies to see that the age estimates produced from dorsal spines reflect the true age of the fish. And it sort of raises the question of why we're not using otoliths, which is a more traditional hard part used to estimate age. Click. And the reason for that is that gray triggerfish have extremely small otoliths that are very difficult to extract, and they're very fragile. You can see in this micro CT scan of a juvenile gray triggerfish on the left that the otoliths, the sagittal otoliths, labeled in blue and green just behind the islands, are extremely small. Meter or two across, whereas the dorsal other otoliths are very fragile, making a preparation and use for age estimation more difficult. Click. But we started to look at some micro CT scans of individual otoliths, and we noted that there appear to be increments on the otolith. I'm cutting out my video. Hopefully the audio comes back. It seems like I might have some internet connection issues. Trent, if you can hear me, can you click?
Yeah, that seems to be the problem. So maybe just keep your video off.
Okay. So yeah, if you click this, it spins the otolith, and we can see some of the unique structure of the otolith and what appears to be some increments on the top right-hand portion and the bottom right-hand portion. So we wanted to look at these otoliths to see if they these increments represented annuli, or were the annual age of the fish. Click. So our objectives of this study were to estimate age for gray triggerfish using section dorsal spines, section vertebrae, and whole otoliths, and then to validate the age estimates to determine which hard part produces the most accurate estimates of age, and then to determine differences in length at age as a function of sex, and fit new growth curves based on validated age estimates. Click. So we collected just over 1,100 gray triggerfish in the field using a variety of different methods. And from each of these fish, we removed the sagittal otoliths, the vertebrae, and the dorsal spines, and we estimated age from each of these hard parts for each fish. We then had a second reader that was actually Keel Condy, who estimated age for each hard part as well, so we could compute a between reader average percent error to get a metric of the precision of the age estimates. Click. To then validate the age estimates to determine which hard part is the most accurate, we utilize the bomb radiocarbon chronometer. Now, this tool is based on the premise that in the 1950s and 1960s, there was a really rapid increase in the amount of radioactive carbon, or carbon-14, in the world's surface waters, in the ocean surface waters, due to the atmospheric testing of nuclear weapons. Now, following the cessation of testing of nuclear weapons in the 1970s, there's been a steady linear decline in the amount of radiocarbon in the surface waters as those enriched waters have mixed with deeper depleted waters. So we can actually gather a natal signature from a fish. Typically, we core out the otolith core of a fish and then measure the amount of radiocarbon in that sample and match it up with our radiocarbon chronometer based on our age estimate to try to validate the age estimates. Now, we require about 1 milligram of material to analyze for radiocarbon. And since gray triggerfish otoliths are so small, we wouldn't be able to get that much material out of the otolith core. So we had to find an alternate structure. And in this case, we utilized eye lenses. Eye lenses have a lot of the same characteristics that make them very useful in a similar fashion as otoliths. They're formed very early in life, and they grow throughout the fish's life. And once they grow, they are metabolically inert. So those tissues under the surface layer are not being reworked, and thus the core of the eye lens represents the natal signature of the fish. We then assess the bias in the age estimates by shifting our age estimates by plus or minus 1 to 4 years and computing the sum of squares of the residuals. And the model with the lowest sum of square residuals represents our most parsimonious fit. And if it's the null model, that means that we don't have a bias in the age estimate. Click. So if we dive into our results, when we were looking at the hard part precision, or the average percent error between our readers, the otolith APE was 4.3%. That means that we had really good agreement between age estimates. And you can see a plot of that on the right, the KC age estimate done by Keel Condy again, and the DC age estimate read by myself. And most of our points line up right along that one-to-one line of agreement, meaning that we're getting a really precise estimate of age with otoliths. Then when we computed the vertebra APE, that was at 10.9%, so a little bit higher, and our dorsal spine APE was at 16.2%. So it's quite a high average percent error, but that was in line with what we've seen in the literature where the APE is often above 15%. Click. So with those results, we were able to determine that otoliths produce the most precise estimates of age, but we now needed to look at the bomb radiocarbon chronometer to see if they were accurate. So this represents the null model of each of our hard parts, and the colored triangles represent our eye lens core radiocarbon signature and the age estimate derived from that hard part. And you can see on the left that our otolith-derived age estimates, all of those red triangles fall within our 95% prediction intervals, which are computed from the reference chronology of known age red snapper otoliths and Gulf of Mexico and Caribbean corals. So all of our data fell very close to that, to what we would have expected from the bomb radiocarbon chronometer and inside those prediction intervals. Now, if we look all the way over to the right at the vertebrae, those yellow triangles, you can see that most of our points fall within the prediction intervals, but there are a couple that fall outside. And it looks like we have a little bit more variance in the vertebra-derived age estimates. Now, if we actually look at the dorsal spines, a lot of our data points are falling well above that upper 95% prediction interval, and it appears that we have a pretty substantial bias in the dorsal spine-derived age estimates. Click. Now, this plot represents the best fit model for each of these, and you can see that for both the otolith-derived age estimates and the vertebra-derived age estimates, the null model of no bias was the most parsimonious fit, had the lowest sum of square residuals. Whereas for the dorsal spines, a bias of plus 2 years had the best fit, the lowest sum of square residuals to the radiocarbon data. Now, this indicates that we're underestimating age with dorsal spines by 2 years, which has really important implications for the stock assessment of gray triggerfish, considering that dorsal spines are currently used in estimating age for gray triggerfish. Click. So after we completed that validation portion of the study, the rest of our analysis we only conducted on otoliths because we validated that as an accurate and precise estimate of age. So I then looked at the lengthed age data for ages 3 to 7, and we did find significant differences in the lengthed age, with males being larger than females, and it was significant in ages 3 to 6. It wasn't quite significant at age 7, but we did observe the same trend where males were larger than females and it was very close to being significant. I believe the p-value was about 0.06. So we did find some evidence of sexually dimorphic growth in great triggerfish, which is important if we consider that the stock assessment growth curves are only computed based on combined sexes, and they do not split those out into male and female growth curves. Click. And again, in the von Bertalanffy feed growth curves, we again see that evidence of sexually dimorphic growth, with the males having a larger L infinite of 504 millimeters relative to the females at 484 millimeters. And one important thing to note is that our estimate of the Brody growth coefficient, the K factor, at 0.28 for males and 0.25 for females, is actually quite a bit lower than what we've seen in the literature for dorsal spine derived age estimates, indicating that when we estimate age with otoliths, we're actually observing slower growth than when we estimate it with dorsal spines. Click. So to wrap things up, otolith-derived age estimates were both accurate and precise, while our dorsal spine-derived age estimates underestimated the true age of great triggerfish, and they were also extremely imprecise. If you recall, that APE was well above 15%. The vertebrate-derived age estimates were accurate based on the Baum radiocarbon chronometer, but they had more variance around around those prediction intervals, and the average percent error was much higher than the otolith-derived age estimates. And again, we found further evidence that great triggerfish exhibit sexually dimorphic growth, which would be important to incorporate into the stock assessment models. Click. So some future work I'd like to conduct is some stock assessment simulations to test for the implications of aging error and incorporate otolith-derived age estimates into the gray triggerfish rebuilding plan to determine how the accurate estimation of age affects the timeline to recovery for the stock. Click. And finally, I'd like to thank my funding sources, the National Marine Fisheries Service and the University of Florida School of Forest Resources and Conservation, all of the Patterson Lab members, and the captains and crew of the Intimidator and the Dreadnought for helping us collect samples. With that, I'll take any questions you might have. Hopefully we didn't lose too much to the freezing.
So we've got about 2 minutes for questions, so maybe one or two.
Hi, Derek. Good talk. Curious, any initial ideas, hypotheses on how your findings might affect the stock assessment results with regards to the bias in the spine ages and also the dimorphic growth?
Yeah, that's a really great question. One of the most important inputs for the assessment is an estimate of longevity. So how old the fish live to be. Now, our oldest ages in this study are actually younger than what are currently used in the stock assessment. So we didn't increase the longevity, but I think that there are important implications in the fact that our otoliths are estimating older ages than dorsal spines. So my guess is that we just didn't capture that oldest fish to increase the longevity. But if you increase the longevity, your estimates of the natural mortality rate are going to go down, considering it's an age-derived natural mortality rate. So that would imply a less productive population that could sustain a lower fishing mortality. So that's what I would expect to see, and there could be some implications of that from the reduced Brody growth coefficient in the growth curves.
Great, and maybe one quick one, if anyone has something quick.
Hey, Derek, Zach, I would just say real quick, I would check out some of the advancements in Bayesian growth model fitting because they have the ability to incorporate both readers. It might help you with your vertebrate reads and incorporating some of that uncertainty that you have.
Yeah, that'd be great. I appreciate it, Zach. I'll shoot you an email.
Yeah,
and get some of those papers. Yeah.
Great. All right. Up next, we have Scott Alford, and he'll be giving a 5 minutes talk, and he is advised by Dr. Martin.
All right. Thanks, Trent. So hi, everybody. I'll be presenting my concepts and progress of an experiment to assess how salinity changes affect the interactions between multiple predators through different Q types and a common estuarine fish, the Gulf killifish. Click. So ecological community structure is determined by a species' ability to tolerate environmental conditions and then interact with other species at a location. Environmental conditions determine where a species can exist through tolerance to localized conditions, which is conceptualized here as environmental filtering. Subsequently, species distributions are then determined by their ability to interact with other species present, as illustrated by this simple 3-species food chain. Click. So top-down effects are a prominent biological interaction that shapes ecological communities and subsequent ecological function. Predators can influence prey in 2 ways. They can directly affect prey abundance through consumption, like in my animation, which generally is limited to removal of prey in immediate proximity to a feeding predator. Click. But predators can also affect prey indirectly through non-consumptive effects, or NCEs. These effects are derived from prey recognizing predator cues that they perceive as risk. And this broadens a predator's effects since it affects all individuals that can detect these effects. NCEs are resultant behavioral changes dependent on predator identity and cue types, like visual or olfactory cues, and can decrease function of prey populations, such as their top-down effects on local resources due to more time being devoted to predator avoidance. Click. So abiotic factors can also influence predator-prey interactions. Estuaries in particular are highly variable environments with mixing of freshwater and coastal marine waters, creating environmental gradients determined by freshwater inflow. For example, salinity can change rapidly, as illustrated here by local, and by here on the top right, by local salinity going from brackish conditions to near freshwater over a tidal cycle along Cabbage Reef. This variability subsequently could affect estuarine community structure and function through which species are present and interacting. For example, prey species that are ubiquitous in estuaries and have high site fidelity, like killifish, may be exposed to predation from more freshwater-associated predators such as largemouth bass when salinity is lower, estuarine predators such as redfish when there is more mixing, or a combination of both at transition zones. Furthermore, physiologically stressful environments, nailed it, environmental conditions, didn't nail it, associated with riverine input could alter these interactions through changes in predator efficiency or the ability of prey to recognize and avoid predators. Click. We designed a lab experiment to determine how changes in salinity and changes in predator diversity can affect anti-predator behavior of a widespread prey species, the Gulf killifish, by focusing on 2 specific questions. First, given the common killifish predators are highly mobile and their local abundances may change with tides, we wanted to examine if killifish response varied when exposed to visual and/or olfactory cues from a predator commonly associated with estuarine environments, represented by redfish, and from a freshwater predator whose abundance depends on lower salinity, largemouth bass. We also used a combination of both predators to examine emergent effects of increased predator diversity at transition zones. Second, we want to examine if changes in salinity may lead to a disruption of killifish predator avoidance behavior. To do this, we additionally exposed killifish to 3 salinity treatments shown in the graph of salinity measurements taken from experimental tanks on the right. We exposed killifish to either increasing salinity by starting it at 5 ppt and then increasing it to 10, maintain salinity by just keeping it 5 for the entire experiment, or decreasing salinity from 5 to near 0. Click. So I ran this experiment at the NCBS wet lab from March to May 2020, set up experimental units consisting of an experimental tank, an adjacent visual cue tank, and a sump tank that was used to manipulate salinity within the experimental tank while exposing killifish to olfactory cues from predators within the sump. The experiments were performed by placing 3 killifish in an experimental tank, letting them acclimate, and then simultaneously exposing them to visual cue, olfactory cue, and salinity treatments by removing visual blockers and pumping water through the experimental tank. We then recorded killifish behavior for 1 hour and are now using MSJ to measure several behavioral metrics related to killifish location, movement, and anti-predator responses. These include metrics such as the amount of time spent within refuge versus open bottom, size of school formed by the 3 fish, and the amount of time moving versus not, so we can compare behavioral responses among treatments. Click. So as of right now, we're about 1/3 of the way done processing our recordings. We will use these results to assess effects of changes in salinity on estuarine predator-prey interactions, both through environmental stress and changes in predator diversity. We'll be able to determine if stress from salinity changes causes variation in killifish response to certain predators or cue types through their anti-predator behavior and refuge use, which is seen in these videos here on the right. We'll also examine killifish response to cues from a predator commonly associated with their preferred estuarine habitat compared to a less common predator to determine if killifish may be less susceptible to changes in predator diversity due to generalized anti-predator responses, or if perhaps they are more susceptible to predation due to inability to recognize cues from less common predators. Click. With that, I would like to thank you all for listening and everyone that's been involved in the project. Email me if you got any questions.
Great, thank you, Scott. Up next, we have Banna Kavalin, who's giving a 5-minute talk and is advised by Dr. Reisinger.
Hi, everyone. So I'm going to be talking about my research on the impacts of interspecific variation in crayfish behavioral traits on stream ecosystem function. Click. So animal behavior can impact the environment through behavioral traits related to consumption and nutrient cycling. Ecosystem ecologists commonly do not account for the role of animal behavior in shaping ecological processes. Additionally, individuals within a species are commonly assumed to share the same behavioral traits and have the same ecological impacts. Research has been showing recently that interspecific trait variation can have impacts on ecosystem function and structure. However, these studies are commonly conducted in mesocosms and thus do not account for the role of environmental variables in these systems. Click. So my research focuses on if among population variation in behavioral traits is a stronger predictor of ecosystem function than environmental drivers in natural systems. Click. So I focus on the critical ecosystem function of stream metabolism because stream metabolism is the flow of organic carbon, which is the fuel that powers ecosystems. It's measured through gross primary productivity, or GPP, and ecosystem respiration, or ER. Benthic organisms in streams can impact metabolism, especially in the water column, by translocating nutrients from the benthic zone to the water column through nutrient excretion or bioturbation. Click. So I focus on the model organism of rusty crayfish found across the Midwest, and I hand collected 11 populations in temperate wadable streams ranging from northern Michigan to northern Kentucky. Rusty crayfish have been shown to have strong ecological impacts at the whole ecosystem scale, and especially in invaded ranges. 7 of my populations marked with triangles on the map are in the invasive range. So thus we hypothesized that rusty crayfish behavioral traits would differ among populations and that behavioral traits and density would strongly impact water column metabolism. Click. So using AICC, I compared 3 models, crayfish population characteristics, environmental variables, and a null model to see which is a stronger predictor of stream metabolism. Specifically, I looked at the behavioral traits of activity, boldness, and foraging voracity using common condition laboratory behavioral assays. For example, this image shows a picture of the activity assay I conducted where I timed and estimated the average level of the crayfish was walking over a series of a whole day. I also quantified an estimated crayfish density in the field. The environmental variables that I looked at were water temperature, canopy cover, and human land use. I measured stream metabolism in the field and used light-dark incubations, chamber incubations for water column and benthic zone metabolism. I also estimated whole stream GPP and ER using oxygen sensors. But today I'm only going to talk about my water column metabolism data. Click. So first I found that behavioral traits all differed among rusty crayfish populations. Specifically, you can see in population R4, R4 is consumed 4 times on average more chironomids than population R9. Additionally, activity substantially differed, where population R10 had activity levels at 15 times more than population R2. Click. So then I found that crayfish behavioral traits and density were stronger predictors of water column metabolism compared to environmental variables and the null model. Plot A and B shows the relationship between water column GPP and boldness and activity, where populations with more bold and more active crayfish had higher water column GPP. The same was for populations with more bold and dense crayfish had higher water column respiration. Click. So overall, in support of my hypothesis, I found that behavioral traits differed among rusty crayfish and that interspecific trait variation may be ecologically relevant in natural systems. Boldness and activity were what seemed to be the stronger predictor of water column metabolism instead of foraging voracity, which may be attributed to bioturbation and nutrient translocation from benthic zones to water column. Additionally, rusty crayfish has been widely introduced in the Midwestern region. And so understanding the role of behavioral trait variation in rusty crayfish populations can potentially predict the ecological impacts of this invasive species. Click. Thank you. I would like to thank my whole lab and the Trout Lake Station team. And if you have any questions, please feel free to email me. Thank you.
Great, thank you, Banna. Up next, we have Cheyenne Stratton, who's giving a 5-minute talk and is advised by Dr. Reisinger.
Thanks, Trent. So my research focuses on the role disease plays in crayfish invasions, and today I will be talking about how a disease outbreak may cause the collapse of an invasive crayfish population. Click. Aquatic invasive species such as lionfish and zebra mussels can cause severe economic and ecological impacts. One of the hypotheses of why invasive species are thought to be so successful is known as the enemy release hypothesis, meaning when the invasive species is introduced into a new area, they leave their native pathogens or enemies behind, leading to fitter individuals being introduced to the area. However, it is unclear what happens to invasive populations as natural enemies or pathogens accumulate in the invasive species. Click. Specifically, what would happen if a disease outbreak occurred? I will be sharing a case study with you all of this potentially occurring in an invasive rusty crayfish population. Click. Rusty crayfish are known to cause ecological impacts through competition and habitat alteration. This is an example of their impacts on a macrophyte community. The left photo shows a rusty crayfish exclosure where we have a thriving macrophyte community. On the right, we have a rusty crayfish enclosure where the macrophyte community has been completely decimated, and these impacts extend to macroinvertebrates and fish. Click. We have the unique opportunity to study the rusty crayfish invasion in Trout Lake, Wisconsin, indicated by the red star on the map. This lake has yearly trapping data of rusty crayfish dating back to the 1980s as it's part of the North Temperate Lakes LTER dataset. We can see by this graph here that after rusty crayfish were introduced to the lake in the 1980s, their population exploded. However, in recent years, we're seeing a dramatic decline in their population. Click. This happens to coincide with a microsporidian outbreak our research team discovered in 2019. We found 48% of individuals we collected in Trout Lake were infected with this microsporidian parasite. A microsporidian is a spore-forming parasite that causes has been known to cause high host mortality and is often species specific. This photo shows the difference between an infected and a healthy individual where the microsporidian infected individual has white muscle tissue. Click. So we wanted to know how whether the microsporidian impacts rusty crayfish's physiological condition and behavior. Click. To do this, we hand collected rusty crayfish from 3 sites within Trout Lake and collected 2 behavioral assays in the laboratory, which I'll explain in more detail momentarily. The crayfish were then dissected to determine infection status. Click. So we found that there was a decline in physiological condition of crayfish infected with this microsporidian. We tested this using a writing response assay. So each crayfish was placed on its dorsal surface and we timed how long it took for the crayfish to return to an upright position. A healthy crayfish can quickly return to this upright position. However, we found, shown by the red bar here, it took crayfish infected with the microsporidian significantly longer to right themselves than the uninfected individuals. Click. We also found that there was a decrease in activity in crayfish infected with this microsporidian. To test this, we placed each crayfish in a wading pool with 9 equal segments and counted how many lines were crossed in a 10-minute period. We can see by this bar graph here that the individuals infected with the microsporidium crossed much fewer lines in this 10-minute period than the uninfected crayfish. Click. So overall, according to the LTER data, we are seeing rusty crayfish densities are declining, and we actually found, or our research team collected viral crayfish in Trout Lake for the first time in 20 years. So this is very promising for the recovery of this system. We are seeing that microsporidium infection does lead to decreased condition and alters key behavioral traits of rusty crayfish. Our next steps are to conduct a whole lake survey over the next 3 years to see if the native communities are able to recover with the decline of the rusty crayfish, and to conduct a mesocosm experiment to test the relative importance of trait and density mediated effects of microsporidiums on crayfish ecological impacts. Next. And with that, I'd like to thank the following individuals for their help with this research. And if you have any questions, feel free to email me. Thank you.
Great, thank you, Cheyenne. Up next, in our last talk for this session, is Abigail Scrow, and she's giving a 5-minute talk, and she is advised by Dr. Behringer.
Thanks, Trent. So I'm going to talk today about expanding the invertebrate biosphere, and this is going to be a report on the first viruses that were isolated from bay scallops here in Florida. Click. There are only 3 previously identified viruses from other pectinid species, and those are listed here. I have scallops in quotes because these are not actually scallop infecting viruses. These are just viruses that have been isolated from scallops, likely bioaccumulated from the environment, since scallops are filter feeders. But I will note that the oyster herpes virus is a bivalve infecting virus, and that actually causes mass mortalities for the Pacific oysters out on the West Coast. But like I said, there's no known viruses of Argopectin irradians. There was a study recently by Shi et al. In 2016 that uncovered more than 1,400 new RNA viruses from invertebrate hosts. So a lot of the previous viral work was focused on vertebrates, but this new study kind of opened the doors and shows us that there's still a lot to uncover in the biosphere. Click. So what I did was I collected 20 scallops from 2 different sites in St. Joseph's Bay at the northwest end of our recreational scallop fishery in Florida. And I focused on 3 different tissue types, the gill, mantle, and digestive gland. And I took tissues and I flash froze them in liquid nitrogen for RNA extraction using a trizol method. I also fixed these tissues in Davidson's for histology. So I took all of my RNA extracts from the 3 tissue types, from the 20 scallops, and I pooled them together to send out 1 sample for next generation sequencing. And I did this in order to increase the likelihood of finding something new. And what next generation sequencing does is it's a parallel sequencing method, and it really allows you to isolate multiple sequences at the same time, entire genomes. And I got back an RNA-seq library. I had like 6 gigabytes of data, and then had to clean it up and run some phylogenetic analysis. Click. I uncovered 4 novel positive single-strand RNA viruses from 2 different families, the Picornaviridae and the Hepaviridae. As you can see in the graphics on the right, both viruses are very similar. They're icosahedron in shape, and they have that single-stranded RNA genome, and they end with a poly-A tail. Picornaviridae have 1 open reading frame, Hepaviridae have 3, and they overlap. And an open reading frame is basically a set of nucleotide that can be translated into amino acids without a stop codon, so it's in its entirety. Click. 3 of my isolates are likely from the Picornaviridae. I tentatively named them scallop picorno-like virus 1, 2, and 3. 1 and 2 are thought to be complete genomes, and they are depicted by the graphic on the top right. And those blue peaks represent strong coverage in my sequence library. I found 2 open reading frames, a polyprotein and a capsid protein. And the 3rd virus down at the bottom is actually incomplete. It's missing the poly-A tail. But the open reading frame that codes for the polyprotein actually codes for multiple different domains. And I use the RNA-dependent RNA polymerase domain and the capsid domain for my phylogenetic analysis. Click. So here's a phylogram. It's really hard to see, obviously, but this shows the relationship of my 3 Bucorno-like viruses. Click. We're just going to zoom in, and almost all of these other viruses come from that really large study. So they're not accepted viruses yet, but they're really well characterized. And what we found is that our 3 viruses actually fall out in different locations, and scallop Bucorno-like virus 1 and 2 group with viruses that were also isolated from bivalve hosts. But scallop Bucorno-like virus 3 is isolated actually from, or groups with a virus isolated from a gastropod host. Click. So I mentioned I had 4 viruses. My last virus is actually from the family Hepaviridae, and it's only a partial genome. Again, it's missing the poly A tail, but there are 4 open reading frames: the polyprotein, which has 3 domains, methyltransferase, helicase, and RDRP, the capsid protein, and the other 2 open reading frames were actually undetermined. Click. So using those capsid proteins and the RDRP protein sequences, I was able to construct 2 phylograms. And if you click again, it's actually going to show you that both have strong bootstrap support. Click for my scallop Hepalike virus grouping with the Barnes Nest sponge-like virus. Click. So there's 4 new viruses, but unfortunately I didn't see any viral inclusions in my histology. Now I was still learning to read my histological slides during this time, but Dr. Bajko will actually be going through those again with hopefully a more keen eye. And if we find any viral inclusions, we'll likely use transmission electron microscopy to view them a lot closer, and you'll see something that looks like the picture on the right. The biggest question is, though, did I find a scallop infecting virus, or did I just find viruses that infect other hosts and they were just bioaccumulated in the scallops themselves? For my 2 viruses that are incomplete, there's a number of different molecular techniques that I can use to finish out those genomes and hopefully complete them. Click. With that, I'd like to thank my committee members and my lab members, as well as FWC that really helped me with all my sampling and my funding sources. Thank you.
All right, thank you, Christina. Hello, good afternoon, everyone. I'm Shelby Thomas, and today I'll be presenting a brief portion of my dissertation, Bay Scallop Restoration in Tampa Bay, evaluating larval release as an effective restoration strategy. Click. Next slide, please. I don't know if it's lagging. There we go. So bay scallops are important ecologically and once supported commercial fisheries in Florida. As an annual species, scallop populations are sensitive to environmental impacts. Scallops in Tampa Bay declined significantly in conjunction with seagrass habitat loss in the 1970s due to poor water quality. Although seagrass habitat and water quality have rebounded, scallop populations have not. Click, please. The spike seen here in the graph, notated by the red arrow, may have been due to previous larval restoration efforts conducted in Pine Island Sound and Boca Ciega Bay from 2003 to 2010 by Jay Leveroni. Research suggests potential recruitment limitations may exist within the bay, calling for more targeted restoration approaches. Previous restoration efforts included using caged adults and releasing settlement-competent larvae, and larval releases may also offer an opportunity to examine the degree to which recruitment limitation isn't a factor in low scallop abundance. Click. For this study, scallop larvae were released on 5 occasions in Tampa Bay between 2019 and 2020. Larval releases ranged from 100,000 to 2 million larvae. Each release site had different contributing broodstocks for the spawn, and the site requirements included ideal salinity and the presence of continuous seagrass habitat. Click. Site selection was advised by FWC with Steve Geiger and Jen Graneman, along with the aid of previous research and monitoring efforts. Site parameters included ideal temperature, salinity, and the presence of continuous seagrass habitat. And Syringodium filiforme manatee grass was found to be the predominant seagrass between all of the sites. Click. Click, please. Sorry. This is a close-up of one of the release locations. At each release site, each spawn was divided to create a low density release, 25% of the spawn, and a high density, 75% of the total spawn. A third control site was monitored with no release as another additional treatment. Each treatment at every site was randomized and separated by 1,000 meters. Click. At each of the treatments, spat collectors were placed after the larval release in cardinal directions, north, south, east, and west, to quantify base scallop recruitment. These collectors were checked after 6 weeks post-larval release to collect the spat for further analyses. Click. A 6-month post-restoration survey was conducted at each site with a 100-meter snorkel survey. Adult scallops that were found were sampled for DNA and released. Click. Scallops collected from the spat collectors revealed no apparent difference between the density treatments, but a clear gradient of scallop density existed from the mouth of Tampa Bay to the inner estuary. Spat collector data revealed no apparent difference between the density treatments, but in fact, some of these sites were indeed the opposite of what you would expect. As seen in this release 1, the control site had more spat recruitment versus the high density site. Click. This is a close-up from release 1. Again, each treatment was separated by 1,000 meters, and the higher recruitment at the control site may have been attributed to the ability of the larvae to transport further than expected post-release. The high-density treatment was south of the control site with a northward current, which may have caused the higher recruitment numbers at the control site. There also may have been a presence of natural recruitment. Further distribution of the collector sites may be beneficial for future studies. Click. Release 2 resulted in less scallops collected at the collectors outside of natural spawning season, with no distinction between density treatments. Click. Release 3 resulted in no scallop recruitment, which may have been due to the location, which was the northernmost site in the bay. Click. And for release 4, a similar low recruitment occurring outside of natural spawning season occurred. Click. At the final release, the results resulted in similar conclusions. The 6-month monitoring resulted in the highest scallops detected at release site 5, which was the southernmost site in the bay, which also had the best conditions. Click. Parentage analyses have been used in a variety of studies to further understand parental contribution. A parentage analysis can be helpful in determining the variance in reproductive success, estimating numbers of contributing breeders, and helping determine breeding patterns. We used 3 previously developed microsatellites from Rebecca Lucas's master's thesis and added 6 new microsatellites to conduct a parentage analysis. I'm currently analyzing genetic data to collect further data and quantify successful recruitment and retention at these sites. The use of the genetics may also lead to further understanding recruitment limitations along with multi-generational translocation and reproduction. I am interested in seeing how parental contribution and the potential to determine effective population size for restoration efforts. This research may also lead to further assessing cost-effective methods for scallop restoration. Click. Thank you to all who have supported and contributed to this project, and thank you guys for watching.
All right, thanks, Shelby. Next up, we're going to have Joseph Henry, advised by Dr. Josh Patterson, giving us a 15-minute talk.
All right. Is it not up yet? Oh, sorry, I'm still seeing the other screen. Is it viewing now?
Christina has the wrong PowerPoint. We're not seeing Joseph's PowerPoint pulled up.
You're not seeing the PDF?
Unfortunately not.
Maybe try like unshare your screen and try just resharing the whole thing.
Okay. How about now?
There you go.
Okay. That's the one. This is
Sorry
something I'm
about
more familiar with. All right, awesome. Thank you very much. Good afternoon, everyone. My name is Joseph Henry, and I'm a PhD candidate in Dr. Josh Patterson's lab. I'd like to begin by thanking the organizers for putting everything together and giving me the opportunity to give a full-length talk. I'm excited to share with you all a project of mine that's taken several years to bring to completion. This project explores the integration
sexually propagated corals raised on land for use in current restoration efforts. Specifically, this study focuses on Acropora cervicornis, also known as staghorn coral. So I want to go ahead and start off with a little background and why we chose this species specifically. Click. There it is. Yeah. So why staghorn coral? In the early 1980s, populations of staghorn coral declined rapidly along the Florida Reef Tract. And that's primarily due to disease. As a result, the species was listed under the Endangered Species Act in 2006 and subsequently became one of the species primarily targeted for restoration. As restorations began, it became clear that the lack of genetic diversity in outplanted populations really needed to be addressed. For example, the Coral Restoration Foundation has the largest nursery in South Florida. And before we introduced the corals from our work with the Florida Aquarium, they only had about 150 genotypes in the nursery. Fortunately, in 2019, we were able to achieve high settlement and survival of staghorn coral larvae, which then allowed us to begin studies outplanting sexually propagated corals. It was also around this time that a study was published showing the low long-term survival rates of asexually fragmented produced outplants, which further emphasized the importance of this work and the need to determine really the feasibility and the value of using sexually propagated corals raised on land for use in restoration. Click. To perform this study, whoops, looks like one's got skipped over ahead. Can you go back one slide, Gene? There it is, yeah. So to perform this study, we collected gametes from 2 spawning seasons, resulting in the 2 separate year classes we evaluated in the study. Corals spawn only once a year, and achieving high settlement and survival is still challenging, making this a very difficult and time-consuming process. So going back in August of 2017 and 2018, we collected colonies from the Coral Restoration Foundation's Tavernier Nursery and brought them to land-based flow-through systems at Keys Marine Lab in Long Key, Florida. So corals spawn in these systems, and that's the photo in the top right under the red light. After a few days, we transported the larvae to the Center for Conservation in Apollo Beach. It's a photo in the center. And here the larvae were placed in settlement bins lined with conditioned ceramic tiles for settlement. Once settled, the coral recruits were grown on their tiles and separated by cutting when they just grew too close to one another. Ultimately, the corals from the 2017 spawn spent 20 months in the land-based system, and corals from the 2018 spawn spent 8 months in the system before being transported for outplanting. Click. So here's our 3 sites. The nursery site at the top, Chica Rocks in the middle, and Tennessee Reef is our most southern. Click. A little bit of why we chose these sites. Chica Rocks is an inshore patch reef right off the coast of Isla Mirada in the Keys. The location did not historically have staghorn coral. It was not initially planned to be part of the study, but during the permitting process, the sanctuary requested us to add it. So that's why it's here. Tennessee Reef, there in the middle, is a back reef located near Long Key. This was our primary location of interest, mainly because it's an ideal environment for staghorn coral, and it once had a historic abundance of the species. Lastly, the Coral Restoration Foundation's Tavernier Nursery was chosen as we were interested in comparing survival between the nursery and outplants, and really kind of further looking to see if that nursery could act as a transitional phase and help to acclimate corals heading to the reef from land-based systems. Click. There you go. Here are some representative photos of how we outplanted both year classes. So due to the differing sizes and morphologies, we elected to use 2 different methods. The 2018 year class seen on the left were approximately 22 millimeters in diameter and planted on raised pyramids and affixed using epoxy. The 2017 year class on the right were approximately 73 millimeters long branches and were planted using the traditional nail epoxy and zip tie method. Next. Quick. Here's how we did the 2-year classes that were hung in the nursery. The 2018s were hung from the tree on their tile using monofilament line and crimp, and the 2017s were attached directly to the tree using monofilament line and crimps as well. Again, the 2018s were approximately 22 millimeters in diameter, and the 2017s were about 73 millimeter long branch. Click. For monitoring, we monitored all 3 sites 5 times over a 480-day monitoring period. We monitored survival and growth during each of these trips and tracked growth in ellipsoid volume. In the nursery only, we cleaned the trees and dispersed the corals when they started to grow too close to one another. And this is really just to provide the same standard of care that is typically given to corals in a nursery setting. Click. So key findings. Exciting, sexually propagated stackhorn coral raised ex situ can survive and grow when outplanted. Not gonna lie, we had our doubts, but we did have some really neat results. Let's head over to the next slide, click. And in regard to survival, we found high survival across all outplants and in the nursery. So the Kaplan-Meier survival analysis on the right represents all corals at each site. So both year classes are represented here. Of the outplants, the lower survival at Chica Rocks was mainly driven by the 2017 year class, which was 28% at the end of the study. We speculate some possible reasons for the low survival found at Chica Rocks include really the method at which how we attach them, environmental conditions, including like Chica Rocks is much more shallow and much more exposed to heavy wave activity and sedimentation. Those are all possibilities. Click. As far as growth, we found no effect on that final ellipsoid volume of corals based on the site. It should be noted that the previous figure was a site comparison, and now this one is comparing year classes. We did find significantly different final ellipsoid volumes based on outplant year class and associated outplanting technique. So interesting, at the end, the smaller 2018 corals were larger than the 2017s at both outplant locations. In the nursery, we also found significantly different ellipsoid volumes between the 2018 and 2017 year classes. And this is where we observed what was probably expected, with the 2017 year class being larger than the 2018s at the conclusion of the study. So ultimately, we were able to see that the nursery was beneficial to growth, in which the corals grew approximately 40 times larger than those that were outplanted. Click. And here's a figure of 4 representative colonies at the initiation, so that's tiles A through D, and the conclusion E through H of the experiment. So these are the same corals at the beginning and the end of the study. And it just kind of goes to show you some of the high growth that was found in the nursery, that being those 4 tiles on the right, or the before and afters. Yeah, pretty big. Click. So management options and considerations. So the corals did well. Now we have to consider what comes next. If land-based efforts annually produce large quantities of sexually propagated corals, ocean-based nurseries could face logistical challenges managing the large influx, which is exactly what happened the year we did the outplant. The Florida Aquarium was successful at raising over 3,000 corals, and the corals had to be distributed amongst several institutions to make this work. Click. So there are some areas of concern with this. You would think that having an abundance of novel genets would be a good thing for restoration efforts. Ultimately, it can be a good thing, but several factors must be considered. These areas include genetic swamping. This is a scenario in which a numerical advantage could be given to introduced alleles, which could result in swamping of the local population without the ability to hybridize with them. Furthermore, outbreeding depression is possible. This is where we could potentially reduce the fitness of the local population by introducing alleles less adapt to the area and ultimately depressing the fitness of the local population. And then lastly, a lack of space and resources to manage the new corals has been a concern in the past and could be even more so in the future. Now that we put those things in mind, we want to like to propose some management options. Keep in mind, none of these options are mutually exclusive, and an approach that combines all 3 of these is certainly possible. Click. So option number 1, it would just be to do some direct outplanting. So direct outplanting avoids overwhelming nurseries and provides an opportunity for naturally successful genets to be identified and potentially return to the nurseries for propagation. Outbreeding depression or genetic swamping of populations must be considered here, especially when you're directly outplanting a large number of novel genets to a single site. However, given the highly fragmented and rapidly declining state of many reefs, the risk of genetic swamping and outbreeding depression may not be of great concern right now, or for some locations. Click. Option 2 looks at just distributing the corals to research. So basically, coral recruit numbers can be made more manageable by directing a portion of these novel genets to research facilities. Topics of interest that could stand to benefit from this include stress hardening, symbiont shuffling, and assisted gene flow programs, those AGF. And the remaining corals could then be sent to ocean nurseries for growth and later outplanting. Click. Third and finally, a clear solution would be to scale up ocean-based nurseries to support the influx of these corals. However, increasing nursery infrastructure and associated labor would result in increased material and personnel costs. Despite this increase in costs, a nursery phase may be relevant for properly managing all the novel genets, as nurseries can actually help to control the release of those genets into their targeted restoration sites. Click. some future research. So in conclusion, these are some things worth considering. So there's a lot of room to fine tune this process and determine best outplanting times, locations, and methods of attachment that can improve the long-term performance of outplants. For example, it could be possible that we can still see high survival of corals sent back to the ocean after an even shorter duration on land. If this is true, then we could help to reduce the cost of land-based propagation. We believe it'd also be interesting to do a follow-on study that outplants the corals that were given in nursery phase. It was clear that the nursery route allowed for additional growth, but would like to further study if it was beneficial in other ways that could allow for higher performance once outplanted to the reef. Or also, like, was it even harmful? Like, maybe that wasn't a good thing to do. So yeah, well, that's all I have for today. Click. Want to thank a big thank you to all our partners here that made this possible. It was a huge collaboration, of course. And then click. And then thank you for tuning in, and I welcome any questions you have. Thank you for the time, Mark.
Thanks, Joseph. We probably have time for maybe one quick question.
Sure.
All right, if there aren't any questions, we'll go ahead and move on. Next up, we have Gabrielle Love, who is advised by Dr. Baker and Dr. Camp, giving a 5-minute talk.
Okay, hi everyone. Today I'll be talking about the collapse of a historic oyster population and the impacts of that on the estuarine community in Apalachicola Bay, Florida. And I want to just start really quick with a thanks to everybody who helped this research happen. Click. Click. Okay. So Apalachicola Bay up there in red on the map was a historically productive oyster fishery, formerly producing 85% of the oysters from Florida. And it collapsed very suddenly in 2012 with an 80% loss of oysters within a year, no recovery and no expected recovery for a while. So there's been a lot of interest in researching the causes of this collapse, but little into the consequences. And that's what I'm interested in. Click. So what does this mean for the rest of the bay? Well, oysters create sprawling reef habitat that are used by a lot of estuarine species. And there's some debate as to whether oysters constitute essential fish habitat for these species. And this collapse could be an opportunity to better determine if oysters are essential fish habitat for certain species, and along with that carry a lot of implications for ecology and management of oysters and certain sport fishing species. Click. So my questions, which are my thesis chapters, but I'm only going to touch question 1 today, is did the collapse of the Apalachicola Bay oyster population impact the population abundances of other estuarine species? Click. So my methods, I used the Fisheries independent monitoring data from FWC collected in Apalachicola Bay from 1998 to 2018 via stratified random sampling. I did a literature review and selected 12 species, including 11 fish and 1 invertebrate, that are commonly found on or rely on residents of oyster reefs. I did a catch per unit effort standardization by gear, year, month, and sampling zone with a delta log normal generalized linear model. This serves as a proxy for species relative abundance. I created 2 time series for each species, 1 for the overall population and 1 for recruits, so 24 total time series. Then I did a change point analysis, which finds mean and variance changes within the abundance time series and separates it into segments based on those different parameters. And I can use the collapse in 2012 as a benchmark for drawing connections between what we see in those abundance changes and the oyster population. So click. So these are the 24 time series that I made, the vertical line indicating the oyster collapse in 2012. And then each species has the solid line for the overall population and the dashed line for recruits. The dashed line, the recruits populations, are especially useful for predicting changes that might happen in the overall population, especially for some of these long-lived species. There's no consistent trend that you can see visually among these, but I would like to point out in the top left corner, the sheep's head is one example where the overall population doesn't really seem impacted, but you can visually see a disruption in the trend of the recruit abundances. And some of the species, you can see apparent stability changes, like the center row, second to the right is the gray snapper. You can see a huge spike and then a large dip in the recruits right after 2012. Click. So these are my change point results. I found 11 time series out of the 24 had change points in or after 2012. And what's important to look at here is the blue box and the direction of the mean and variance changes associated with those. And what's really important is that there's no consistency among that direction, and although not presented here, the magnitude of those changes. And there are changes here among resident species and transients alike. And those two graphs that I have there include the vertical lines with the change points that were found in those time series. The top one is the sheep's head and the bottom is the gray snapper. So even though there is an apparent stability change in the gray snapper, it doesn't necessarily mean there was a change point and a significant change in the mean and variance of those abundances. Click. So from this, we can say that the Apalachicola Bay oyster population collapse did impact the community, but these responses were non-uniform and non-linear. There was apparent relative stability because none of these fish populations completely collapsed following the oysters, but we still do have those cascading effects evident. It's hard to solidly claim if it's essential fish habitat because these methods are descriptive and not diagnostic, but it does indicate that we ought to have explicit consideration for these species interactions in management strategies for oysters and those recreationally and economically important species that rely on them. Click. Future work will be my second thesis chapter, looking at a community composition approach to the same kind of questions, seeing if it altered species abundances and interactions at the community level. Click. And feel free to email me with any questions. Thank you.
Great, thanks, Gabrielle. Next up, we have Chelsea Conley, who's advised by Dr. Michael Allen.
Hello, my name is Chelsea Conley. I am a master's student at the Nature Coast Biological Station. My project is a collaborative study between UF and FWC focused on the impacts of oyster restoration on nekton assemblages. This project was focused on Lone Cabbage Reef, which is located just south of the main Suwannee River Delta. This reef had suffered a 77% decline over the past several decades. And in 2018, this reef was restored to its original 5 kilometers in length using limestone boulders and oyster and clam shell. The goal for our project was to evaluate the short-term effects of oyster restoration on nekton assemblages. We predicted an increase in relative abundance and species richness along the restored reef habitat, as well as a shift in species composition from an anchovy-dominated assemblage to one that included residential demersal species and juvenile fish and invertebrates using the restored reef as refuge and feeding grounds. We sampled Lone Cabbage Reef using directed sampling 1 year before and 1 year after the restoration project. We also isolated fisheries independent monitoring samples to compare our directed sampling to. We used the Great Suwannee Reef, which is located just offshore from Lone Cabbage Reef and had suffered 100% reef loss over several decades. This reef was not restored during the restoration process of Lone Cabbage Reef. We also used inshore marsh habitat to compare our samples to. We used a 21.3 meter center bag seine and followed the same fisheries independent monitoring procedures. So we were able to compare sites across locations since all sites use the same monitoring sampling procedures. We didn't see any huge changes in our species richness and relative abundance before and after restoration, but this top graph shows our average CPUE across sampling locations. Our lone cabbage reef post-restoration CPUE did have an increase compared to pre-restoration, but our inshore and offshore control sites did not differ before and after restoration. The species richness and Shannon's diversity index also did not differ before and after restoration across all locations. We did on average have higher richness, diversity, and abundance on our inshore control sites compared to lone cabbage and offshore control sites. Our nekton assemblages also did not differ before and after restoration for our control sites, so those were pooled for this MDF plot. Our offshore control is shown in green, the inshore control is in pink, lone cabbage pre-restoration in blue, and lone cabbage post-restoration in red. The lone cabbage pre-restoration assemblage structure was more similar to our offshore control, whereas lone cabbage post-restoration was different from all other locations. Now this table shows the species that contributed to those differences. Across the board, the bait anchovy was the highest contributing species for all locations except the offshore control, which had the hardhead catfish as their highest contributor. If you look at the lone cabbage post-restoration sampling, the silverside and juvenile spot also contributed very highly, whereas in other locations, the blue crab was the next highest contributor, followed by other species. So there was a difference in that species composition. As well as some of these species that you see above, the sport fish were caught during our post-restoration sampling, whereas we did not catch those same species during pre-restoration sampling. So that was another interesting find with this study. Now, hopefully that gives you a brief overview of my project. And I would like to thank all of the FWC and NCBS volunteers, as well as my committee, Mike, Charlie, and Caleb, for helping me out with this and making this study possible. Thank you so much.
All right, great. So next up, we're gonna have Stephen Stang, who's advised by Dr. Michael Allen.
Hey everybody, I'm Stephen. I'm a second semester master's student in the School of Fisheries and Aquatic Sciences and current freshwater fisheries biologist with Florida FWC. And today I'm going to talk to you about part of my project looking at the potential effects that climate change, fishing mortality, or a combination of both factors are having on a unique Gulf strain of striped bass in Florida's Panhandle. Click. So striped bass have a pretty big range in the United States. They've been introduced in a lot of reservoirs throughout the country, and they're a very popular sport fish among the angling public in both their native and introduced ranges. Click. Click. And my project is looking at the Gulf strain of striped bass, which historically ranged from the Suwannee River west to Texas. This strain of stripers are a little bit genetically distinct from the Atlantic strain striped bass. They're more of a riverine species. They very seldom enter salt water, and they rely very heavily on springs and creeks as a thermal refuge source in the summer months when water temperatures get higher than their optimal conditions. They were heavily removed from much of their native range with the construction of dams, with the last stronghold occurring in the Apalachicola River system. And currently there is a broodstock source in the Aquatic River where this project is being looked at. Next, please, click. So the Aquatic River serves as a major brood source every year, every spring. Fish are collected below the dam and sent to the Blackwater hatchery to be used for the production of Gulf strain striped bass and hybrid striped bass. But recently a question is coming up as to where most of the fish over 20 pounds are. They're not seeing that number of fish in that size range like they used to. And so we're trying to look at, is this due to heavy fishing pressure, warming water temperatures due to climate change, or a combination of both factors? Click. So we know that these fish, once they get over the age of 5, really prefer water temperatures around 25 degrees Celsius in the summertime. So we dropped hobo temperature loggers in a few thermal refuges in Lake Talcott this summer and paired that hourly temperature data with weekly dissolved oxygen recordings to get a better idea of what these refuges were looking like and to see if they're actually capable of supporting striped bass of that size class during the summer months. Click. And our results show that there were suitable temperature and dissolved oxygen parameters found at Hammock Creek, Ocklawaha Creek, and the Little River. And although the deepest sections of the lake did have suitable water temperatures, dissolved oxygen was lacking during most of the summer. Click. And this winter, we conducted our high dollar reward tagging study. We tagged 200 individuals, half in the reservoir and half below the dam, to get a better understanding of overall fishing mortality. Click. And this is shows you the lake frequency of these fish that we've tagged. And since we started tagging in December, we've already had 27 individuals recaptured. And we expect that number to greatly increase this winter and spring. And next year, we're hoping to obtain an age sample to get a better idea of the overall mortality in the system, so we can parse out fishing and natural mortality. Thank you, guys.
Great, thanks, Stephen. Moving on, next up we have Amy Oxton, advised by Dr. Don Behringer.
Hello all, my name is Amy Oxton, and today I'm going to be presenting a portion of my master's thesis, which is on the sublethal and lethal effects of Microcystis species blooms on juvenile Calycynectes sapidus. Click. It is of no surprise to any Florida native or resident that algal blooms have been at the forefront of environmental news reports. They're inundated with articles and stories like this one from my local newspaper, the Fort Myers News Press. What most stakeholders don't realize is that blooms are naturally occurring and are present year-round in background levels. When conditions arise, namely temperature and excess nutrients, background levels can grow into persistent blooms that are referred to as harmful algal blooms, or HABs. Large-scale blooms have increased in prevalence and occurrence in recent years. Anthropogenic impacts such as nutrient loading have been shown to increase natural blooms, resulting in dense cyanobacterial mats, also called cyanohabs, which can be found in both marine and freshwater systems. There are many reasons that people are concerned with harmful algal blooms, including human health impacts. Click. This is because some harmful algal blooms can produce toxins. Some cyanobacterial species are non-toxic, while others produce secondary metabolites. The most common hepatotoxic cyanotoxin is microcystin. Of the toxic strains, microcystin LR, or MCLR, is the most studied. Microcystin is a hepatotoxin which causes hepatosis, or a functional disorder of the liver, in afflicted species. Widespread and intense blooms have negative impacts on the ecosystem and humans around them. Microcystin pose threats to human health through direct ingestion, like drinking water or contaminated foods. In aquatic systems, some toxic strains have proved to have lethal and sublethal effects on a variety of aquatic organisms, including crustaceans. Click. Historically, Lake Okeechobee flowed south through the Everglades, eventually terminating in Florida Bay. Man-made alterations resulted in canals from Lake Okeechobee to the Caloosahatchee River and the St. Lucie River. I apologize that my slide does say the St. Johns River. Discharged water often contains cyanohabs and their associated toxins, which originate from Lake Okeechobee. This is reflected in toxin levels that are reported in weekly monitoring of the lake and associated rivers by the Florida Department of Environmental Protection. Click. The blue crab, Calycynectes sapidus, is a commercially and recreationally important species to the state of Florida. Blue crabs undergo an extensive and complex life cycle through saltwater and freshwater environments. The blue crab fisheries in the Caloosahatchee and St. Lucie can overlap with microcystis blooms released from Lake Okeechobee. Blooms release toxins that have the potential to impede the development and movement of blue crabs. Sublethal physiological impacts have been shown for other decapod crustaceans. Juvenile blue crabs move up the estuary to lower salinity areas where they eventually sexually mature and mate. Therefore, juveniles represent the most at-risk life cycle stage to microcystin LR exposure. With no available literature on this direct interaction between blue crabs and microcystin, the effects are not truly known for this species. Click. To explore this interaction, I devised 4 objectives for my thesis. For the purpose of this presentation, I'm going to focus on the first 3, which is to determine the effects of microcystin exposure on the physiology of juvenile blue crabs, determine the effects of microcystin exposure on juvenile blue crab behavior, and to examine potential pathological effects in blue crabs after prolonged microcystin exposure. Click. To accomplish the first 3 objectives of my thesis, I used an exposure trial. All work with microcystin LR was approved through the University of Florida Environmental Health and Safety Acute Atoxin Approval AT 4805. Because of limitations and regulations associated with this toxin, we chose to use a static non-renewal test. In this system, test organisms are exposed to the same solution for the duration of the trial, in this case, 28 days. Juvenile blue crabs were collected with dip nets in the marsh off Cedar Key with the help of Scott Alford. Animals were acclimated to laboratory conditions that mimicked the habitat from which they were collected. Concentrations were established by reviewing the weekly monitoring I had previously mentioned from the years 2018 and 2019. We chose 4 representative treatment levels, including a control. They were a low treatment of 5 micrograms per liter, a medium treatment of 15 micrograms per liter, and a high treatment of 50 micrograms per liter. Microcystin LR was chosen because it is naturally occurring and represents one of the more toxic and well-studied strains. Click. Here's an example of our static non-renewal drip system. In the bottom left-hand corner of the left picture, you will see our holding tank. Water was pumped from this tank up into the drip lines and flowed into individual beakers where the crabs were housed. Water flowed out of the beakers and back into the tank where it was recirculated into the system. Click. There was a suite of variables that we monitored through this study. Mortality and molting were checked 3 times daily, and food was done daily beginning in the first check. Crabs were fed approximately 5 to 8 pieces of food and given 4 hours to eat. Crabs' remaining food was then scored on a 1 to 5 scale. Food and molts were removed from the system and disposed of according to EH&S protocol. Click. Riding time was conducted weekly as a standard method to assess overall muscular functioning of crabs and crayfish, you might remember from Cheyenne's presentation, where an increase in riding time indicates decreased functioning. And riding time is defined as the time required for a crab to right itself after being turned on its dorsal side 180 degrees. Histology was used to examine any pathological effects of exposure. As a hepatotoxin targeting the liver of vertebrate animals, the hepatopancreas fills a similar functioning of the intestine, liver, and exocrine pancreas of vertebrate species. Therefore, any histological alterations of exposure should be visible in the hepatopancreas. Click. Now I'm going to walk you through the results of the first 3 objectives of my thesis. Click. A survival analysis was applied to the mortality data using the RStudio survival package in the Kaplan-Meier method. Mortality, or lack thereof, was compared between treatments over time. To orient you to the survival curve, the control is in blue, the low treatment is in green, the medium treatment is in an orangey yellow, and the high treatment is in red. These colors will remain constant through the duration of my presentation. Solid lines represent the population of surviving crabs in each treatment over the exposure period. Transparent polygons are the 95% confidence interval of the populations. The x-axis is the time of the trial, so 28 days. I would like to direct your attention to the y-axis. I've adjusted the population to go from 100% survival to 70% survival for better visualization since the mortality was so low. There was no significant difference in survival throughout this trial period. Click. To analyze food scores, I used a cumulative logistic regression, where the amount of food consumed by crabs in the high toxin treatment was significantly lower than the other treatments. I used a predictor function in R to take the results from the regression and create this visual you see. The X is the rating scale of scoring 1, representing no food consumed, and a 5 representing all food consumed. The y-axis represents the predicted probability of a crab in a treatment, let's say the control, to receive a feed score of a 1, a 2, a 3, and so on. Click. To analyze molting, I also used a cumulative logistic regression, where a number of molts were totaled throughout the trial time. Animals ranged from 0 to 2 molts in the 28-day period. There was a significant decrease in the total number of molts in the high and medium treatments. Predicted variables were used to create a visual again. And I would like everybody to focus on the red bars of the high treatment. The high treatment had the highest probability of not molting at all and the lowest probability of molting 2 times. Conversely, the control had the lowest probability of not molting at all and the highest probability of molting 2 times. Click. A linear mixed effects model was applied to the writing time data, and a general correlation function was added to account for repeated measurement of the same specimens. At all treatment levels, there was no significant difference in change over the trial period. And it demonstrates that an exposure to microcystin LR does not significantly affect the muscular functioning responsible for a writing response. However, these positive slopes that you see here for all treatment levels reveal that crabs did take longer to right themselves as the trial progressed. Though not significant, this slope across groups could be a result of weakly handling or holding containers. Click. The hepatopancreas from the control group were circular and tubular in structure with normal lumen deposits as seen in figure A, where the lumen deposits resembled a fitted star shape. However, crabs in the low treatment group exhibited minimal histopathological changes that I don't have on the slide here. Tubules in the high treatment had a loss of circular and tubular shape, and the lumen structure was observed to be abnormal, which you can see in figure B, denoted by AL. Delamination of the epithelium was also present in the medium and high treatments that you can see in figure 3, where the epithelium has come away from the hepatopancreas tubules. Click. The low mortality of blue crabs following a 28-day exposure to MCLR demonstrates high survival in the presence of microcystin splumes. High levels of MCLR in the water column significantly suppress the food consumption of Kaitlyn sapidus. An exposure had varying effects on the number of molts completed per treatment. This suggests that crabs in the high treatment may have had to allocate more energy to a different function rather than producing a molt. Reduced ability to molt and thus reach sexual maturity could have lasting effects on the blue crab fishery in these regions. Thus, populations undergoing MCLR exposure could have slow growth rates, ultimately impacting population dynamics and regulatory models. There was no significant difference in writing time over the exposure trials. Of all treatment groups, the high treatment had the highest time to write in 5 weeks. This suggests, though not significant, that the muscular functioning decreased as time elapsed. This could be a physiological consequence of decreased feeding and thus decreased metabolic energy required to complete the time to write trial. Severe histological responses that we saw in histology is supported by other work and suggests that MCLR has damaging effects on the hepatopancreas structure and potential functioning. Our study demonstrates that MCLR has sublethal physiological impacts on juvenile Kaitlyn sapidus. Click. If you would like to hear a more detailed version of this talk and hear what the 4th objective was about, feel free to tune into my thesis seminar, which is being held on March 3rd at 2 p.m. Click. And with that, I would also like to thank my funding sources and everyone a part of the Behringer Lab that is also listed here. Without their help, this project would not have been possible, and I will take any questions in my time left.
Great. We have about 2 to 3 minutes for questions. So does anybody have any questions? Feel free to also write them in the chat if you'd rather do that.
You can also save your questions for my defense if you want. You have until March 3rd to think of new questions. Fresh, so I am responding to Mark Q. Martindale's question, which is, what is the half-life of the toxin? Many small molecules are oxidized in 24 hours in seawater. Was fresh toxin added every day? No, fresh toxin was not added every day. It was remained the same in the 28-day period. Microcystin LR was purchased in a solid form from Cayman Chemical and is extremely expensive in nature. So that is a disadvantage of the static non-renewal test, is that you are using the same solution throughout the duration of the trial.
All right, well, if there's no more questions, we'll go ahead and move on. Thanks, Amy.
Thank you.
Next up, we have Ryan Henry from the Dr. Lorenzen's lab. He's going to be giving a 5-minute talk.
All right, thank you. My name is Ryan Henry, and I'm a master's student, and I'm also a fisheries biologist at the Florida Fish and Wildlife Conservation Commission. I work out of the Joe Budd Field Office in Quincy, Florida, and today I'm going to be talking about Chipola River shoal bass and the effects of Hurricane Michael. Click. So for those that don't know what a shoal bass is, it is a rare species of black bass that is similar to smallmouth bass. It was formally described in 1999, so it's a relatively new species. They inhabit areas with rocky substrate and moderate to fast currents, and they are native to the ACF, which is the Apalachicola, Chattahoochee, Flint River drainage, which is located in Florida, Alabama, and Georgia. Their diet consists primarily of crayfish, insects, and fish. They grow pretty fast, and they can reach maximum sizes around 7 pounds. And in the spring, they spawn in areas with boulder and fine rocky habitat. They can live up to 11 years, and they are currently labeled as a species of high conservation concern by FWC. Next. So the Chipola River is a river in the Florida Panhandle. The headwaters are formed by Marshall and Cowarts Creek in Jackson County, Florida. It is a tributary of the Apalachicola River. It spans 149 kilometers long, with 63 active springs throughout the river. The habitat of the river primarily consists of boulders, rock shelves, and shoals, which is perfect habitat for shoal bass spawning. And shoal bass have been historically found between the cities of Mariana and Alta, Florida. And the Chipola contains the only known sustained population of shoal bass in the state of Florida. And angling pressure throughout the Chipola River is quite low, mostly due to difficult navigation because of the shallow shoal areas, making it difficult to cross. All right, next slide. So Hurricane Michael made landfall in Florida on October 10th, 2018, had sustained winds of 155 miles per hour, and it caused devastating damage throughout the Florida Panhandle, mostly affecting towns such as Panama City, Mexico Beach, and Mariana. And it was recorded as the 4th most intense hurricane based on wind speed in United States history, causing an estimated $24 billion in damages. Along the Chipola River, the hurricane downed trees in flooded areas, and days following the hurricane, there were severe fish kills reported throughout the Chipola River. Click. And the 2 pictures above are just showing a before and after picture of Hurricane Michael on the Chipola River. The left being before, you can see there's quite a bit of canopy cover, and just on the right, the devastating damage that it caused to the trees along the riverbank. And the picture below is just showing the amount of fish kills that were reported to the FWC fish kill hotline on the Chipola River after Hurricane Michael. Click. And our office with FWC did an electrofishing survey following the hurricane in May of 2019. And our survey revealed a 91% decrease in relative abundance of shoal bass. And shortly after that, FWC decided that we would start gathering adults to serve as broodstock for supplemental stocking in the future. And there was also an executive order put into place restricting harvest of shoal bass throughout the river. And currently there are 24 fish being kept at the Blackwater Fisheries Research and Development Center in Holt. And there are also 8 fish being held at the Florida Bass Conservation Center in Webster. And the plan currently is to stock advanced fingerlings in the spring of this year. Click. So I'd like to talk about the genetic concerns that we have for the Chipola River shoal bass really quick. The first being that non-native black bass have negatively affected shoal bass through introgressive hybridization throughout their native range. The 2 species being spotted bass, being known to be able to spawn with and hybridize with shoal bass, and the other being a provisional species of black bass known as the Choctaw bass. And the Chipola River actually currently has one of the most genetically pure populations of shoal bass, and we like to keep it that way. And we have estimated that the genetic purity of the population is 93%. Another concern that we have is that population depletion may amplify any hybridization that occurs after the hurricane, and also that with low population abundance, that the genetic diversity of future generations of shoal bass might have issues. Click. So my research is in the early stages right now. We're in the planning stage, but as of right now, the plan is to do a stocking evaluation using the contribution of hatchery fish to the adult population, using genetic markers to identify the hatchery fish, and also just monitoring the abundance of the shoal bass population after stocking. And also monitoring of genetics in the shoal bass population, such as the hybridization and the effective population size. And the final being mapping of suitable spawning habitat for shoal bass and doing a comparison before and after the hurricane. And that is all I have. Thank you for your time.
Great, thanks, Brian. Up next, we're gonna have Catherine Rose from Dr. Dawn Behringer's lab. She's gonna be giving a 5-minute talk.
Hi, everyone. As Christina mentioned, my name is Catherine Rose, and today I'm going to be presenting on some preliminary results for one of the objectives in my master's thesis, which is meant to examine the impact of trap confinement on the condition of the Florida stone crab. Click. So the Florida stone crab is a cultural and an economic pillar to the Florida community. Recently, it's contributed about $30 million annually to the Florida economy in catch landings. It is a claw-only fishery, which means that fishermen are allowed to take one or both claws before returning the live animal back to the water. And because stone crabs can regenerate their claws, it's very tempting to think of the fishery as sustainable, but there's a lot of evidence that it is in fact overfished. The 2011 stock assessment suggested that despite the fact that effort has been increasing in the 21st century, that catch landings are actually down. There's also evidence that a large portion of crabs that have one or both claws removed don't actually survive to regenerate their claws, which is why a large proportion of the claws in the fishery are the original claws rather than the regenerative claws. Click. Unfortunately, it looks like it's only going to get worse for the poor stone crabs. Having fished out inshore habitat, stone crab fishermen have been setting their traps further offshore and opting to check them less frequently to compensate for the increased fuel cost and the increased effort associated with checking traps that are further away. So I and some members of the FWC stone crab team, particularly in the Florida Keys, were interested to see how this change in behavior and potentially increasing soak times was going to affect the stone crab population and potentially the fishery. Click. So I plan to accomplish this with the 3 objectives of my thesis, the first of which I'm going to be focused on today, which is to measure the effect that trap confinement has on the condition of the Florida stone crab. The second of which is meant to measure the effect of a simulated expected starvation that crabs experience while they're in traps. And the third of which is meant to improve the accuracy of sort of a quick and dirty method that is often used to assess the nutritional condition of crabs in the field. Click. So with members of FWC, particularly in the Keys stone crab team, we developed a trap study that we conducted in the fall and are currently conducting another round of as we speak in Cedar Key and in Marathon, in which we deploy 35 traps at each location, allow them to fish openly for 5 to 7 days. And then on that 5 to 7, on that day 5 or day 7, we pull the traps and we collect the crabs from about 1/3 of them, and we block the remaining so that while those traps continue to soak for an additional treatment of 14 days and an additional treatment of 30 days, that no other animals are climbing into or out of the traps. And when we collect those crabs, we're measuring a large myriad of physical and nutritional indicators. For the sake of this presentation, I'm going to focus just on the first two that are outlined in that box there. The first of which is the dry weight index, which is one of the nutritional indices that comes most highly recommended for crustaceans given its accuracy. The second of which is the RAMP, or the reflex action mortality predictor, which is a neurological assessment that it can be directly correlated with a crab's immediate survival. And it was honed specifically for stone crabs by Stephanie Kronstadt in 2017. Click. So I've summarized the results for our dry weight index into this box plot here. You'll notice that the dry weight index is on the y-axis and the location is on the x-axis, with Cedar Key on the right and Marathon on, Cedar Key on the left and Marathon on the right, with colors differentiating the treatments. And as you can see in both locations, there seems to be a consistent decrease in the dry weight index as soak time increases, which is what we expected to see. The longer that crabs are soaking in these traps, the more nutritionally deficient that they become. And an ANOVA as well as a linear model showed that these differences are statistically significant. And there's also a significant difference between the locations. Click. The good news is that these soak times don't seem to be immediately affecting our crabs' health. I've summarized the RAMP scores on histograms here with the number of crabs that have with one RAMP score on the y-axis and the RAMP score on the x-axis. It's an integer from 1 to 8 with higher RAMP scores indicating a more imminent mortality. And you might see some bars there that don't fall exactly on a whole number, and that's because to use this figure, I used trap averages. But either way, you can see that a large proportion of our crabs are sitting right at 0, which means that there is almost no implication to their neurological state. Some of the crabs that maybe soaked for slightly longer are still only sitting at a 1 or a 2, which is not at all threatening to their immediate survival. So this is good news for stone crab fishermen who like to take claws from live crabs in order to improve their catch quality, and also potentially a good thing for the population, given that their immediate physicality isn't affected, and they might be able to perhaps make up for that nutritional deficiency that crabs are suffering at longer soak times. Click. So I look forward to being able to further tease apart the intricacies of these relationships with my next 2 objectives and keep this community updated. But with that, click. I have to acknowledge everybody that was involved in the planning and execution of these field elements and dissections. So thanks so much for listening.
Thanks, Kate. The last talk of the session is going to be by Erin Walters, who advised by Dr. Behringer as well. She's going to be doing a 15-minute talk. Let's go ahead.
Hi there. Can everybody hear me? I think you can, so I'm just going to keep rolling. If not, let me know.
We can hear you.
So you can hear me? Okay. So my project is focused on assessing and estimating the discard mortality in the Florida blue crab fishery using the reflex action mortality predictor, which it sounds like we've already heard a little bit about. Prior to this study, the discard mortality in the blue crab fishery was entirely unknown. Click. So diving into a little history of the Florida fishery, we have been collecting landings data since the 1960s. And the most recent stock assessment had determined that neither the east or the west coast is overfished, nor is it undergoing overfishing. But looking at our landings graph here, if we focus on that blue line, we can see that the pounds harvested through time is in a general decline. And while we do see that there are short periods of recovery, they never seem to exceed the levels of previous years. There could be numerous reasons for this, right? You know, our assessment is saying that the blue crab fishery is not overfished, but we're not seeing any growth. And a lot of it could be contributed to the type of data we are inputting into our stock assessments. There's a lot of data gaps. For example, we have no idea what the discard mortality rate, which could contribute to some of this uncertainty. So any of this information would be very helpful to better assess the stock and inform our fishery managers. Click. So RAMP actually got its start up in the Pacific Northwest in the salmon fin fishery. The protocol was developed up there to relate reflex actions to the well-being or mortality of salmon finfish. And since it has been readily adapted to numerous other crustacean species like that of red king crab, snow crab, tanner crab, Dungeness crab, even the Florida stone crab, and it has been done so with a high success rate. So given this, we decided to adapt RAMP to the Florida blue crab in hopes that we would be able to use this method to predict mortality of these discarded animals in the fishery. So what does RAMP do exactly? Well, it defines the relationship between reflex impairment and the likelihood of mortality. And this is based off the notion that those reflex actions accurately represent the vitality of the animal and can then be used to predict the mortality. So how it works is a RAMP assessment is applied to each individual crab, and this quantifies what we call a reflex impairment score. A reflex impairment score is quantified by the sum of lost reflexes. So looking at the graph here on the left demonstrates what a general RAMP index or RAMP curve looks like. Let's say a crab incurs a reflex impairment score of 2. This would mean that the animal did not respond to the 2 of those 9 reflexes. Therefore, its probability of mortality is probably pretty low. But let's say a blue crab would incur a reflex impairment score of 8. This means that it did not respond to 8 of the 9 reflex actions, and therefore its probability of mortality is probably substantially higher. So the general rule of thumb with RAMP is the higher that reflex impairment score, the higher the odds are to mortality. Click. So again, because RAMP protocols have been pretty well adapted to other crustacean species, we had a fairly good idea of some good reflex actions to test for the Florida blue crab. And we initially tested 12, but after a series of tests, we determined that only 9 of them were reliable for this species and provided somewhat easy manipulation during assessment. If anybody's worked with blue crab, I'm sure you can only imagine. So the 9 reflexes tested from here on out in this study are eye retraction, leg retraction, mouth closure, intennual reaction, antennae reaction, joint reaction, chelae closure, appendage turgor, and chelae wave. Click. And I'm sure by now we are probably wondering what a RAMP exam looks like on a blue crab. I think you can play the video on here. Can you? Yeah, perfect. So here I'm testing eye retraction. There's antennae reaction, intennual reaction, mouth closure, leg retraction, joint reaction, and chelae closure. And on average, it takes me about 30 seconds to RAMP each individual crab, depending on how much the animal fights me through the exam. But this is great because this method allows us to gather a lot of data on any given sampling period. Click. So my very first objective is, of course, to establish this RAMP curve, right? We need to relate the relationship between reflex impairment score and the probability of mortality. And to carry out this objective, we decided to bring our experiments out to the live field operations on the commercial vessels. So we would ride aboard these commercial vessels, allow the stress of typical and routine fishing activity to induce the various ranges of the reflex impairment scores, and then we would monitor those discarded animals with the use of modified holding pens for a series of 2 days. So how it worked was we would ride aboard these commercial vessels and we would collect data on these crabs. We would RAMP them, so we would incur that reflex impairment score. We would take information on their size, the sex, the time the animal was out of water, so that desiccation period may provide some influence on the mortality. We would also record the number of injuries each crab had. During fishing, they can lose limbs, lose chelae, they can get cracks or holes in their carapace. And we also recorded abiotic variables that also may provide some influence to the mortality. So we took data on water temperature and salinity during the timeframes those animals were held. So again, I mentioned that we monitored that mortality for 2 days, and we would check these holding pens every 24 hours after fishing. And so we would simply go out back to those locations. We would pull these pens one at a time and simply just do a quick look over if there was any animal that was found dead. We would remove it and record that tag number and then redeploy the pen back over. Each pen held 15 animals, 10 of those being discards and 5 are controls. So we had controls in each pen to just be sure that our holding methods were not causing any additional mortality, which we found that was the case. And I was able to complete this experiment 9 separate times in the Tampa Bay Estuary and in the St. Johns River during the summer and winter months. And we had those locations chosen simply because those were locations that we had commercial fishermen agree to work with us for a couple of years for this extended project. And we did in the summer and winter months because we had hypothesized that we may see some seasonal differences in mortality. Click. So generally, logistic regression is what is used to model ramp data. And so we did just that. You can see here that our generated relationship between reflex impairment score and the probability of mortality is shown in that right top graph. And we had ran a total of 12 or I think it was 13 model combinations of various variables in those different models. And our top model had come out and shown that the reflex impairment score, injury score, and salinity were the best predictors for determining the mortality of those animals in the 2-day holding period. And that top model came in with a 91% accuracy prediction rating, which we were pretty happy about. That second ranked model included the same 3 variables, but it also included water temperature. And if we look at that bottom right graph, we can see that this just shows the influence of salinity on mortality for these animals. And we had found that actually the blue crabs seem to fare better in the fresher water versus the salt water. And so how it influenced them is the fresher the water that was, the ramp curve would be not so steep and it would be pushed further to the right. But with increasing salinity, that ramp curve would become a little steeper and be pushed to the left. And for injury score, it kind of worked in the same way. So if the animal had no injuries, that ramp curve was pushed to the left and not quite so steep, but with increasing injury score, the ramp curve was pushed to the left and became further steep. Click. Okay, so now that I have generated that relationship between the reflex impairment score and the probability of mortality, I can now simply use that index from objective A and just take ramp exams on discarded blue crabs and just be able to estimate the mortality based off our index. So if we see here on the map on the right, these were all the locations that we fished in aboard commercial vessels to collect our data. We did so again in the summer and winter months to see if there was any seasonal influence. And we were able to collect discard data on over 1,000 crabs. Click. Okay, so our mortality estimate based on that top model. So in the Northeast region, which I've labeled JV on the left, the predicted mortality from those animals in there was about 5.2%, so pretty low and kind of what you would hope. But in our Central West region, or the Tampa Bay region on the right, we had a bit higher mortality. We had it predicted at 8.9%. And keep in mind that the salinities in those different areas probably had a decent effect on those estimates as suggested by our top model. Click. And with that, I'll take any questions. I actually think I had one down here.
Yeah, you had one question from Declan Burton. They asked, did the order of the reflex test vary If so, did you notice any significant impacts from this? If not, why choose the order that you did?
The order of the reflex test did not vary. We kept that very consistent. I didn't notice, I know that in previous ramp studies that everybody keeps the consistency and the order the same. And so we just followed suit with that.
Yep.
Does anybody else have any questions?
Hi, Erin. I had a question.
Absolutely.
I was curious. This study seems to look at the mortality due to injury, but do you think there could be an impact from increased predation? Like if the blue crabs aren't moving as quickly, they may be more vulnerable to being eaten, and that could really compound the potential discard mortality.
Absolutely. So that's the one downfall with assessing discard mortality with the ramp method. You basically have two ways of doing it. You can do this in a lab, or you can bring this out into the live field and either hold them with holding pens, or you can use like expensive methods like biotelemetry, which just wasn't an option for a budget.
Right.
So the holding methods, we do understand that there could very well be the predation aspect after this, but maybe in a future study, we can look at that with more expensive means in doing so.
Cool, thank you.
Yes, sir.
Will Patterson asks, how did Erin do so many 1,000+ ramp analyses offshore if they took 30 seconds each to complete? Did time out of water affect the ramp score?
Yes, I would say that time out of water probably affects ramp score. So that desiccation period, just from my observation, I would say does. Although our models did determine that time out of water or desiccation wasn't super influential. And I think that is simply because some of the crabbers that I had fished with make a very good effort to keep those animals time out of water very shortened because they are trying to respect the resource. Now, not everybody fishes that way. I think I answered your question.
Great. Well, I think that's all the time we have for questions. That concludes session 2 of today.
All right, well, I'd like to welcome everybody to day 2 of the Fisheries and Aquatic Sciences Virtual Graduate Student Symposium. Once again, I'd like to thank my fellow committee members for all that they've done to make this possible, as well as our faculty coordinator, Dr. Shirley Baker. I will hand the reins over to Kate Rose, who is our first session moderator of today.
My pleasure to introduce Sarah Hutchins, who is a student of Dr. DiMaggio.
Okay, well, thanks for the introduction. Hi, everyone. I'm Sarah Hutchins, and I'm a biologist and master's student at the UF Tropical Aquaculture Lab. And today I'm presenting my research on manipulating copepod escape responses. Click. Many species of marine ornamental and food fish can be difficult to culture because they rely on small, nutritious live prey items for their first feed. Newly hatched copepod nauplii are an ideal prey item and have a quick escape response thought to elicit predatory feeding behaviors in larval fish. However, these escape responses may be so fast that larvae cannot catch them. For example, when Pacific blue tang larvae were stocked in tanks with high prey densities and allowed to feed for 5 hours, only 50% had food in their guts, which leads us to believe that they had difficulty catching these prey items. These experiments will use 2 species of copepod, Parvocalnus phrasirostris, which is a calanoid copepod and a staple in the aquaculture industry, as well as the species Oethona calcarva, which is a cyclopoid copepod and is an ideal candidate for aquaculture. Click. Since nauplii can evade predatory fish, how can we slow them down enough for larvae to catch them without reaching the naupli's lethal limits. So which treatment level will properly slow down the copepods, and how quickly will they become responsive when returned to normal seawater conditions? Once treatment levels have been identified, escape responses will be quantified using high-speed video analysis. Click. So treatment options currently being considered include altering temperature and salinity, as well as adding lipid emulsions such as Algamac and Selco, which are highly viscous enrichment products commonly used for rotifers and Artemia. The goal of these experiments is to find treatments that are simple and easy and cheap to for farmers to use in order to slow down their copepods. For the first temperature study, nauplii were placed in refrigerated water baths for a few seconds and then returned to room temperature water baths and observed under the microscope. After 10 minutes of recovery, P. crassirostris nauplii were only about 26% responsive, whereas after only 6 minutes of recovery, Ochocarva nauplii were about 62% responsive. This study, as well as preliminary salinity studies, suggest that Ochocarva are more tolerant to acute environmental changes and will require more severe treatments, such as a freezer dip instead of a fridge dip, in order to sufficiently slow them down. So these pilot studies will help determine treatment levels for future formal experiments. Click. This setup shows the USF Gemma lab setup with the high-speed video camera on the left, a cuvette in the middle holding the copepod samples with a piezoelectric stimulus probe, which is triggered by a pulse generator to simulate an approaching predator. We'll be measuring copepod response latency, speed, and distance, and measuring these using ImageJ. Click. If you watch the copepod that's in the top right corner of this video, you can see it swimming in response to the stimulus probe. And we'll see if the video loads.
Oops, sorry about that, Sarah.
Oh, it's okay. So in case it doesn't, this video is recorded at 1,000 frames per second and shows the copepod escape in response to the stimulus probe moving. And the response only takes 0.057 seconds. Oh, yep, maybe it's still not loading. Well, either way, the graph on the right represents the copepod's escape movement. So each data point shows one frame of video to show the copepod's overall trajectory. And we can use this data to determine max and average velocities for each escape response. Click. So once experiments have been completed using these simulated predators, these same methods will be applied using natural predators, starting with Pacific blue tang. And these experiments aim to improve larviculture practices and reduce reliance on copepods. Click.
Sarah, I'm sorry, I'm gonna have to cut you off, but you're
it's okay. That was it.
Are you good? Perfect.
Yeah.
Cool. All right. So if that is the case, thank you so much for that, Sarah.
Yep. Sorry that video wasn't working.
No, it's okay. Technical difficulties of Zoom, right?
Yep, it happens.
So next, we're going to be moving on to Kaitlyn McCord's talk, which is a 5-minute presentation, and she is a student of Dr. Courtney O's.
Thank you. My name is Katie McCord, and like Kate said, my advisor is Dr. O's, and I'm a graduate research assistant at the Indian River Research and Education Center. I'd like to thank UF IFIS CALS and rising tide conservation, Dr. Oz and Dr. DiMaggio and Dr. Yanong, my committee members, for their support. So my project is the use of fluorescent microspheres to define prey preferences of fish larvae throughout larval development, with the objective of determining the prey preferences and the ultimate goal of optimizing survival and larval feed use efficiency. Click. Click, please. Thank you. So the fluorescent microspheres are small pieces of plastic. Mine are 1 micron in size, and they visibly light up under a fluorescent microscope so you can see them. So the idea is that the plankton, the zooplankton that the larvae eat, will consume the microspheres, and then depending on the color of the microsphere, I can identify which organism was eaten by the larva. The microspheres have traditionally been used in research to investigate microplastics in marine trophic webs. And it was Isaac Lee, another student of Dr. O's, that came up with the idea of using the fluorescent microspheres to identify prey preferences. Click. So here's a visual graphic. It's just, like I said, live feeds eat the microspheres, the larvae eat the live feeds. The larvae are then euthanized with MS222 and fixed in formalin, and they're photographed, and the photographs are used to determine what was consumed. Click. So here I have some images of some of the prey items. On the left is the rotifer, Brachionis plicatillus. It ate green microspheres. And on the right is a copepod species, Apocyclops panamensis, that ate some reds. Click. So the experiments that I have planned are a Parvo Calanus crassirostris versus Apocyclops panamensis. Those are 2 different species of copepods, and Parvo are more commonly used in aquaculture, but I'm presenting the idea that maybe Apocyclops might be a little bit better of a prey item. And then the next experiment will be introducing the rotifers to the mix by comparing rotifers versus the parvos, and then putting all 3 together to see what the larvae prefer. Then repeating at progressing days post-hatch at about 1 to 15 days post-hatch, depending on when the larvae become too colorful themselves to be able to be photographed accurately. So the images you see on the left at the top, that's a bicolor blenny that shows that this method can be used for other species other than the ones that I'm planning to research, which are Florida hogfish, bicolor doggybacks, and hopefully some blue tangs, and whatever else tends to spawn around here. And then the other 2 images are Florida hogfish. They're actually the same fish with different lighting. It's in the same position. So you see on the top image how the gut is highlighted by the fluorescent microscope, and you can see all the little green rotifers in there. And then on the bottom image, I turned up the stage light so you can see the fish better. Click. So the next steps are completing the evaluations, just getting more pictures, more larvae, and making kind of a timeline that shows the changes in prey preferences during larval growth, because it's believed that sometimes, well, it's clear that larvae transition to different foods as they grow larger and larger. And then verifying the results using growth trials, using the recommended food organisms to make sure that it does accomplish the ultimate goal of optimizing survival. And then on the right, you can see 2 more images. The top image is a Florida hogfish. The green is Apocyclops, and the orange is Parvocalanus. And then the lower image, green is rotifers, red is Parvocalanus. Click. I'd like to thank again UF IFIS CALS and Rising Tide Conservation for their support, and my references from Isaac Lee's work and the microplastics. Thank you.
Great, thanks, Katie. So next we are going to hear from Marilyn Susanna Kalau, who is a student of Dr. Yang's, who will be giving a 5-minute talk.
Hello.
We can hear you.
Okay. Can you guys see my video?
Yes, we can.
Okay. Hello, I'm Marilyn Kalau. I'm a PhD student under Dr. Hiping Yang. So today I'm going to present my research about total lipid determination of microalgae used in molluscan aquaculture by use of flow cytometry. Click. Yeah, molluscan aquaculture is a culture of mostly marine bivalve species such as manila clams, oysters, hard clams, mussels, cockles, and other type of mollusks. Click. In the past 10 years, world molluscan aquaculture production had grown from 15 to 35 million tons, making it as one alternative to for seafood consumption in the world's market. Click. Yeah, in aquaculture setting, microalgae are a primary food source, especially in hatchery, where mostly broodstocks and larvae are fed with flagellates, diatoms, and green algae. Click. The algae are the source of lipid that important for early larvae development and gonad maturation. So by providing feeding plant with a mix of microalgae species is expected to provide complete nutrients with suitable cell size and density. There is a limited resource regarding to lipid utilization in hard clams, which are our main focus for next phase in the research. So therefore, investigation of lipid accumulation and characteristic of microalgae as feed is needed. Click. One more click. Yeah, the first step to do is to develop a protocol for total lipid determination. So we choose to using a high throughput method that allows for a rapid determination, which is the main goal for this experiment. Click. Yeah, one more click. One more click. Yeah, the method is a flow cytometry, which is a technique used to detect and measure physical and chemical characteristic of cells or particles. So basically it measures cells in liquid suspension, and the way it works is cells are fluorescently labeled and then excited by the laser to emit light at varying wavelengths. Click. For this experiment, the dye, which is a body pie, as it labels lipid droplets. There is also like another dye that can be used to label lipid droplets, but we choose a body pie because it has an advantage to be able to penetrate algae with a thick cell wall, such as green algae. And also like the total result of the total lipid measurement is close to gravimetry analysis, which is often used as a standard for the total lipid measurement. Click. Yeah, for this experiment, macroalgae are grown for a period of 10 days and then harvest. Later, they are measured with flow cytometer. So in the right, there are two types of flow cytometer in our lab. The upper one is the standard flow cytometer. So I use in this experiment, while the bottom one is the newcomer. So for the next experiment, I'm going to use the Attune next instead of the old one. Click. Yeah, and this is the list of the macroalgae that I use. So in total, I have like 5 species ranging from flagellates to diatoms and also green algae. Click. Yeah, just to summarize, so the general protocol that I developed consists of labeling first with a fluorescence dye, and then I'm going to staining that with a certain procedure and following by fluorescence reading. Next, click. Click. Yeah, this is the sample for the flow cytometer output for the second staining concentration. So basically, this is the output of the flow cytometer itself. Click. Yeah, And this is the complete result for the T-ISO. So this is just the first for the first species, T-ISO. So a different color in lines represent a different staining concentrations, and concentration from 0.5 microgram per milliliters and above give a significant difference. And for the staining time, there is no difference after 5 minutes for all concentration. So further work are needed, such as develop a standard curve to validate the total lipid measurement and also adjust the protocol with the new machines. Click. Yeah, so I hope I meet the time. Many thanks for listening to my presentation.
Great, thanks, Marilyn. That was great. So next, we are going to be moving on to Casey Murray's presentation, who is another student of Dr. DiMaggio, and she will be giving a 15-minute presentation.
Hi everyone, my name is Casey Murray. I am a second-year PhD student at the Tropical Aquaculture Lab in Ruskin. My advisor is Matt DiMaggio, and today I'll be talking about developing larval culture protocols for the black-banded sunfish. This research is just one part of my dissertation, where I'm focusing on studying fish digestive physiology throughout the larval period of both marine and freshwater ornamental fishes, to design efficient nutrition and production protocols. Click. Ornamental aquaculture is the industry that supplies sustainable alternatives of freshwater and marine ornamental fish to the aquarium trade. Not only is ornamental aquaculture the largest sector of aquaculture in Florida, Florida is the #1 producer of ornamentals in the United States. Often, Florida producers have to compete with exporting countries such as those in Asia that can produce ornamentals such as the freshwater angelfish on the right and the neon tetras at cheaper costs due to cheaper labor and production costs. So Florida producers can capitalize on native ornamentals that are considered exotics in other countries and can command a higher price. Click. The black-banded sunfish is a small freshwater centrarchid that is native to stagnant and slow-moving water bodies from New Jersey to Central Florida. Its population status has been declining over the past few decades due to habitat degradation and introduction of native species, or non-native species, sorry. The populations have also been declining due to overcollection for the aquarium trade in some places. And you can see on the map that the population status across its range is variable, but it varies from vulnerable to critically imperiled. The FWC recently conducted population surveys of black-banded sunfish in Florida, and they actually had a lot of difficulties finding populations of black-banded sunfish in places that they were historically known to inhabit. Click. Black-banded sunfish are good candidates for aquaculture, not only to increase production for the ornamental trade in Florida, but also to potentially help out with future stock enhancement efforts. Until now, no aquaculture protocols have been developed for black-banded sunfish. And it is important to note that other cultured centrarchids, such as the largemouth bass, have been difficult to wean from live diets, such as Artemia, to inert microdiets, such as pellets. And this is important because weaning off fish larvae from live diets increases the production efficiency and increases nutritional intake of the larvae. Click. To design some production protocols for the black-banded sunfish, I conducted 3 different trials. I started with describing the digestive enzyme ontogeny and GI tract morphology of larval black-banded sunfish. I then used this data to design a weaning trial where I tried to identify the best timing for microdiet transition sometime in the larval period. And then lastly, I used that data to design a dietetics trial where I tested 3 different types of commercially available microdiets on the length and survival of black-banded sunfish larvae. Click. For the first trial, I stocked black banded sunfish larvae into 10-gallon recirculating tanks connected to a recirculating aquarium system. I sampled larvae from 2 days post-hatch all the way till 50 days post-hatch. And at each sampling time point, I measured and counted the larvae, and I also preserved them for histology and digestive enzyme assays. At each sampling time point, I conducted digestive enzyme assays for lipase, trypsin, and pepsin to look at how these activities change throughout the larval period. Click. The graph on the left shows the growth of black banded sunfish larvae from 2 to 50 days post-hatch. Their growth was steady, however, it was a bit slow compared to other freshwater ornamentals in production. The picture on the right is a progression of their development, and we found eye pigmentation at 2 days post-hatch, swim bladder inflation at 4 days post-hatch, notochord flexion at 21 days post-hatch, and the beginning of body pigmentation at 48 days. Click. These photomicrographs show the difference between the digestive system before a stomach is functional and the digestive system after the stomach is functional. So on the left at 32 days post-hatch, this photo shows the digestive tract epithelium, which has just general digestive tract epithelial cells and some goblet cells. It kind of looks like an intestinal photomicrograph. But on the right, we have a functional stomach which is indicated by the presence of the gastric glands, which are shown with the arrows. Click. The patterns of lipase and trypsin followed similar. They had similar patterns throughout the larval period, so the activities were pretty steady until about 40 days post-hatch, and after that, they increased dramatically. Click. This graph shows the activity of pepsin, which is an acid protease enzyme and indicates the presence of a functional stomach. So we didn't see any pepsin activity until around 32 days post-hatch, but it peaked at 40 days post-hatch. Click. So we used the developmental milestones we found in the previous trial to design this next trial. So since we found a functional stomach at 40 days post-hatch, we decided to introduce microdiets in a window around that time. So we introduced microdiets at 36, 42, and 48 days post-hatch against an all Artemia control, so an all live feed diet. Each one of these treatments had 4 replicates, and you can see the system on the right-hand side, which is where the fish were stocked into. It was a recirculating system. And after 25 days of feeding, larvae were measured and counted, and survival data were analyzed using a generalized linear mixed model with a Bonferroni correction, and length data were analyzed using a Kruskal-Wallis test. Click. We did not find any significant differences in length across treatments, which is interesting to note because that kind of shows that the larvae that are being fed the microdiets earlier on are still growing at the same rate as the control and all the other treatments. We did find differences in survival between larvae that were fed the microdiet earlier on in the larval period versus later on. So the survival of larvae fed at 36 and 42 days post-hatch were significantly lower compared to the survival of larvae fed at 48 days post-hatch, and then larvae that were just fed the live diet the whole time. Click. Lastly, we designed a dietetics trial using the time point of 48 days post-hatch determined in the previous trial to test different commercially available microdiets. So these are all used by Florida aquaculture farms, and we just wanted to test different brands. They're all around the same size, but they do have differing proximate analyses. We had 3 different diets, so Golden Pearl, Otahime, and Ziegler, against an all Artemia control. Again, each one of these treatments had 4 replicates. And 48 days post-hatch larvae were fed the microdiet or the control twice daily for 20 days. And after 20 days, we collected the larvae, counted them and measured them, and analyzed the data in the same manner as the previous trial. Click. Click. Thanks. So we also found no differences in length for this trial, and that's also important to note because the larvae were all kind of growing at the same rate no matter what they were eating. However, we did find that the larvae fed the golden pearl diet had lower survival compared to the larvae fed the all Artemia control diet. We did find that the Otohime and Ziegler diets performed equally to the Artemia control. Click. To sum up everything we found in each trial, in the descriptive trial, we found that the pancreatic enzyme activities followed similar patterns and increased dramatically after 40 days post-hatch. We also found evidence of a functional stomach at 40 days post-hatch with the presence of gastric glands and a peak in pepsin activity. In the weaning trial, we found that survival was highest when microdiets were introduced later on in the larval period at 48 days post-hatch. And in the dietetics trial, we found that Otohime and Ziegler diets performed equally to the Artemia control. Click. Click. Oh, I'm sorry. Yeah, okay, right there. Thanks. Overall, black-banded sunfish are slow-growing larvae with a prolonged agastric stage. We saw that they did not transition to an adult mode of digestion, meaning from pancreatic enzymes to acid protease enzymes until at least 40 days post-hatch. We also found that microdiets should not be introduced before 48 days post-hatch, or you could sacrifice some of the survival of the larvae. Therefore, extended use of Artemia can be beneficial with black-banded sunfish. We also found that certain types of microdiets were working better than others, but it is important to note that black-banded sunfish can be transitioned to a microdiet successfully without significantly affecting the survival of the larvae. Click. Click. Okay, I'd like to thank the Southern Regional Aquaculture Center for supporting this research, and my committee members and co-workers at the Tropical Aquaculture Lab for helping me design my trials and do my research. Click. I'll take any questions.
We do have about 3 minutes for questions I'm going to go into the chat here. You have one question from Maya Glaubach that said, do you have any inferences as to why there was less survivability when fish larvae were fed earlier compared to those who were fed 48 hours after DPH?
So, yeah, so that's a good question. Because we did find the evidence of the stomach at 40 days post-hatch. So we did introduce the microdiet at 36 days post-hatch, which was pretty much before they physiologically had a stomach. So that kind of makes sense why they wouldn't accept the microdiet that early. They don't have, if they don't have a functional stomach, they don't have that pepsin activity that is used to break down the proteins and everything in an acidic manner, which is a more efficient way to break down a complex diet like a microdiet. So that's probably why the earlier ones didn't make it as well. Since we did find it at 40 days post-hatch that there was a stomach, it might take a little while for the stomach to really become truly developed into an adult form of a stomach. We were marking the development of a functional stomach with the gastric glands, which is what we found. But it can take a little bit longer to grow more gastric glands and to produce more and more of that pepsin enzyme to better break down the more complex diets. I hope that answered your question.
So you have another question from Kaitlyn O'Neil in the chat that said, did you have a period of co-feeding microdiet with Artemia, or was it a hard transition?
So we did do a 5-day co-feeding period with 50% Artemia and 50% microdiet. And then after that 5 days, we went straight to microdiet. So we just didn't want to, do a hard transition because it's not realistic of what the industry is doing. So like producers in Florida won't just take them off of Artemia one day and throw microdiet in the next day. So they will do that transition. So we wanted to make it as realistic and relatable to the industry as possible.
Great. Thanks, Casey. That was awesome. I think we are going to move on to the next presentation, which is a recorded presentation from Kenneth Black, who is one of Dr. Baker's students. And let me just try to find, interesting. Just trying to find it to share here. For some reason, Zoom is not recognizing the fact that I have this video window open.
Try just unsharing your screen and just sharing the video file, Kate.
Okay. Oh, yep, here we go. Let me just get it set up here. Sorry, guys. Let me pull this up properly again before I get to clicking. Okay. Can't seem to, sorry, Cheyenne, I can't find it. Like I can see it on my computer, but Zoom doesn't seem, will only pull up the app. It won't pull up the actual.
Right. I'll try, I have it pulled up. If you want to stop sharing your screen, or Scott, do you have it?
I'm having the same problem. Give it a try.
Okay. Can you guys see that?
Yep,
Yeah,
it's up there.
Hello, my name is Ken Black, and I'm a student of Shirley Baker's.
Can we hear this?
I apologize for not giving it, but here is a recorded speed talk on modeling.
Hey, Cheyenne, I think you got to unmute for us to hear the video.
Environmental parameters to simulate filtration rates, ingestion rates, assimilation, and production, and reproduction. And then through those, they simulated changes in weight and change in length. We built off of this by taking the filtration and assimilation functions and using those to develop functions that simulate nitrogen transformations, especially ammonia excretion and the biodeposition of particulate organic nitrogen. This was the equation list that we used. It was a system of 4 differentials and 24 state equations. And the initial implementation of the model looked like this. The endpoint was about what we expected based on observed data, but we were consistently underestimating early growth and overestimating later growth. In order to bring this curve back in line with reality, we first made sure that we had a really robust environmental data set to work with, and then we tweaked a couple of the equations and recalibrated. The 4 parameters that we needed were temperature, salinity, total suspended solids and chlorophyll A concentration. For temperature, we had actual data from Dog Island, which was our study site. We had an 11-year data set to work with, courtesy of Leslie Sturmer. So we used that to develop this time series as the input for our simulations. We had a mean for each day and a standard deviation for each day in the time series. Similarly, we had this time series for salinity. Again, the 11-year data set was courtesy of Leslie Sturmer. For chlorophyll, we didn't have that robust data set to work with, but we did have 1 year's worth of data thanks to Aaron Quinlan and Ed Flips. And then we used that data to create a sinusoidal function that simulated the seasonal change of chlorophyll concentration across the year. And we had turbidity data, but we didn't have total suspended solids data. So we used turbidity to simulate total suspended solids. And then we validated it using 1 year's worth of data, again, thanks to Aaron Quinlan and Dr. Flips. So with that environmental data set, we were able to run a series of Monte Carlo simulations to give the likelihood of potential outcomes. And this is what we ended up with. Again, the endpoint was okay, but we were missing the mark in this middle portion of the curve. The biggest modification that we made to the model was in the differential equation for length. The original equation consisted of a maximum intrinsic growth rate that was fixed, a set of terms that determined what proportion of that maximum was possible based on the condition of the clam, and then a logistic style limit that imposed an asymptote for the growth curve. We substituted the fixed maximum growth rate with a dynamic one that was calculated using the von Bertalanffy differential equation. And this, we got the parameters from the literature. We kept the condition terms because this made sure that the system was still sensitive to the environmental data. With this, this is the outcome. And as you can see, we're much closer to what we expected to see. When we had a production model that was functioning, we used that to build out our nitrogen transformations, and we were able to simulate biodeposition of particulate organic nitrogen across the growth curve and ammonia excretions across the growth curve. And then we used these data to inform our evaluation of the impacts of hard clam aquaculture on nitrogen cycling. And these we validated with a series of feeding experiments. So thank you very much, and special thanks to Shirley, to Leslie Sturmer, and Aaron Quinlan for their data sets, and to my dissertation committee. And this was funded by Florida Sea Grant. Thank you.
Thanks, Cheyenne, for taking over on that. Next, we're going to be heading into Aaron Pilnick's talk, who is advised by Dr. Josh Patterson, and he will be giving a 5-minute presentation.
All right, thanks everyone. My name is Aaron Pilnick. I'm a PhD student in the School of Natural Resources and Environment. My advisor is Dr. Josh Patterson. Today I'm going to talk about larval disease and antibiotic response during long spine sea urchin or Diadema antennarium aquaculture. Click. Just some really brief ecological context. Diadema is historically a really important coral reef grazer and bioeroder. They've been described as both foundation species and keystone species due to the really large impact on the benthic community. Sadly, in the early 1980s, there was a mass mortality event that resulted in up to 99% population reductions throughout the Caribbean and Western Atlantic coral reefs where they're primarily found. And this largely contributed to this ecological phase shift away from a hard coral dominated ecosystems towards macroalgae dominated ecosystems. Next. So therefore, these coral reef restoration managers are really interested in methods to augment Diadema populations in the wild. And one question is, can we actually aquaculture the species on a scale to try to put them back out and restore some of the herbivory that was lost. So it's really hard to do this. They have a really complex life history, like shown on the left here. They exist as these planktotrophic larvae for up to 70 days or sometimes longer before they settle out as urchins. There's no commercial process. The larvae are really delicate due to a lot of different things, such as metal toxicity. They have really fragile morphology, tend to be poor swimmers. And as I'm going to talk about now, they're also susceptible to larval disease. Click. So while we actually have been able to culture the species in a hatchery setting, click, more often than not, what we run into is larval disease around 30 to 35 days post-fertilization. So the image on the left here is a healthy or normal larvae. And what we see typically is there's a sort of light gut coloration. There's these pigments that are found within the tissue that are sort of red and purple in color, and they tend to concentrate around these calcite appendages. In comparison, here's a disease larvae on the right where we see a gradual darkening of the gut, and these pigments tend to move away from the appendages and onto the surface of the tissue. Click. Zooming out a little bit, again, on the left here, we see a healthy or normal larvae with uniform tissue thickness and symmetrical appendages compared to a larvae on the right, where we see some tissue erosion and thinning of the appendages. Next. So typically when you see some disease signs such as this, you want to run through a host of diagnostic techniques to try to really understand the genesis of the disease or the disease dynamics. But seeing as we're really primarily interested in the question of whether or not we're able to culture the species in a hatchery setting, we sort of skipped some of those initial diagnostics and went straight to antibiotics. So we set up an experiment where we actually homogenized some of these 38-day-old larvae and equally distributed them into 8 of these 40-liter culture tanks shown on the right. 4 tanks were designated as no antibiotic control replicates, and 4 were designated as medicated replicates that received a broad-spectrum antibiotic or oxytetracycline. And both the control and medicated treatments were subjected to the same shaded and 16-hour no flow-through periods. And then we took volumetric subsamples during a time series to try to understand or quantify larval survival over time. Click. And what we found was that the medicated larvae survived around 91% compared to the unmedicated larvae survived at 69% after 7 days. So pretty encouraging results there. Click. And furthermore, some of these larvae that were treated actually recovered and then developed through the normal progression through metamorphosis and then settlement into juvenile sea urchin shown on the right. Click. In conclusion, what did we learn? We know that the oxytetracycline broad-spectrum antibiotic works fairly well. Why? This is a good question. Our working hypothesis is that maybe a broad-spectrum antibiotic actually is able to reduce the abundance of potentially pathogenic bacteria that could be found within either the water column or on the culture tank surfaces, or maybe even on the larval tissue itself. And then our next steps in the research are really to return to some of those baseline histology or baseline diagnostic tools that we kind of skipped, including histology and some of the bacteriological identification, so we can really understand the disease dynamics and improve our yield and hopefully get the species back out into an actual restoration stocking program. Click. And that is the conclusion of my presentation. I'd just like to acknowledge the following partners, collaborators. Thank you very much.
Great, thanks, Aaron. Next, we are going to be moving on to a 15-minute presentation by Noah Bolt, who is advised by Dr. DiMaggio.
Hi, everyone. Thanks, Kate, for the introduction. So my name is Noah Bolt, and I'm a second-year master's student at the University of Florida Tropical Aquaculture Lab. And like Kate mentioned, I am co-advised by Dr. Frank Ash and Dr. Matthew DiMaggio. So today I'll be talking to you guys about an overview of the ornamental aquaculture industry with a preliminary regulatory impact assessment. Click. So the ornamental industry is very diverse here in Florida, with hundreds of species currently being cultured and new species being added annually to the industry. So like other aquaculture sectors, ornamental fish are very common on international markets. So according to the FAO, the United States imports approximately $65.3 million worth of fish annually, which is more than the next 5 countries combined. However, the United States only exports $11.4 million worth of fish annually. So this large trade deficit reflects a common problem that we see with our farmers here in Florida, and that they have to compete with an often cheaper foreign product. So in the United States, Florida leads the US in production with 42% of all farms located in the state. And while these farms are typically scattered around the state, production is mainly centered in Tampa because of the high water table, location below the freeze line, proximity to international airports, and relatively cheap land. Click. So regulations, we notice, have a substantial impact on aquaculture, and redundancies in these regulations have been identified. So a reduction of these redundancies has shown to improve profitability on bait fish, sport fish, and salmonid farms. However, researchers on these projects and researchers on this one understand that regulations are in place to protect the public health and the environment from potential negative externalities from industry. So a build off these preliminary studies, the USDA has begun an initiative to assess 5 other aquaculture sectors, including tilapia, shellfish, hybrid striped bass, catfish, and ornamental fish here in Florida. Click. So to collect this data, we censused the entire industry in Florida. And to get the list that we used to create that census, we contacted the Florida Department of Ag and Consumer Services. So they maintain a list of all active aquaculture farms in Florida. And the reason they do that is because to have an aquaculture facility in the state, you have to be issued an AQ number. So these numbers are unique to individuals, and that is how we developed our list of about 100 farms after we eliminated farms that we did not deem meet our criteria. So our survey was developed from those previous bait fish and sport fish studies, however, it was obviously modified to fit the uniqueness of our industry. So our interviews were initially face-to-face. However, we had to transition to phone and Zoom conversations after the COVID pandemic. So as opposed to grouping farms by acreage of production or volume of fish sold like previous studies, we decided to group our farms by total sales for our industry. So analysis included costs and losses due to federal, state, and local regulations, as well as the economic effect of those regulations. Click. So we broke our survey down into 7 regulatory categories. So they're listed here. I'll go through each one a little bit, tell you guys a little bit about each category. So the first was predation, which was losses due to fish-eating predators, such as birds, turtles, walking catfish, snake, even sometimes alligators. The second category was the FDAC's Best Management Practices Program, which is a required program that producers must comply with in order to be issued that AQ certificate. So some components of this program include effluent treatment, non-native species control, things like that. The next section was water, which is regulations that regulate the ability to construct wells and access the Florida aquifer. The next category was restricted species, which are regulations that prohibit the culture and sale of certain species within the state and around the country. Next, we had drugs and chemicals, which covered regulations that ban the use of certain chemicals which would be beneficial to culture. Next, we covered interstate shipping and fish health regulations, which dealt with the movement of fish within the state, outside of the state, and even internationally. And finally, we just had a miscellaneous regulation category, which included regulations such as taxes, business licenses, insurance regulations, and so on. So as well as on-farm losses and cost due to regulations, permits such as well dig permits, bird depredation permits, and USDA APHIS export certificates were counted in their respective categories. And after an analysis of all of these regulations, the regulatory categories, excuse me, we noticed that the 3 with the highest impact were the drugs and chemicals category, predation category, and restricted species. Click. So we identified 3 drugs and chemicals that have substantial impact on ornamental producers in Florida. So one of these drugs was methyl-testosterone, which can be added directly to feeds, which creates phenotypic males, which typically demand a higher price on the market because of their bright colors and larger size. However, this drug in the United States is currently restricted under the Drug Enforcement Agency at the federal level. However, it has a quite substantial impact on percent of total sales. So the lost production from the restriction of this drug is approximately 55% of total sales across the industry. Dialox and Baytex are two other pesticides that were very commonly used in prep for ponds for larval fish by eliminating harmful insects and crustaceans. So both of these chemicals are restricted under the Environmental Protection Agency, at the federal level. So lost production for Dialox was over 100% of total sales, and lost production for Baytex was about 43% of total sales. Click. So the next regulatory category we identified was predation. So outdoor culture is fairly common in the ornamental industry because of its low labor and maintenance cost, as well as the favorable climate found in Florida year-round. So live bears are commonly used raising raised using this method, and farmers typically stock ponds with breeders and leave them there just harvesting the offspring, so super low maintenance. So common types of non-lethal deterrents includes things like bird netting, which cover the entire pond, bangers, firecrackers, and propane cannons, which are used to create loud sounds to scare away birds and other types of predators. So we noticed across the industry, lost production from predation on average was about 1/4 of sales annually. So lethal options do exist to deal specifically with birds. However, it's heavily regulated at the federal level, and the process to apply for the permit can be quite lengthy, and the number of birds allotted often does not solve the issue. So for each type of predator, the level of damage was determined by 3 qualitative categories. So we have takes quite a few fish, takes some fish, and does not take many fish as the least impactful. So as you can see, birds definitely had the largest impact on the industry, with 70% of farms experiencing issues with them, and 61% of those farms stating that birds took the most amount of fish. Turtles were the next impactful, with half of farms reporting issues with them. However, their impact was not as great as birds, with only 26% of farms reporting that they take the maximum amount of fish. The last major predator that we noticed was otters, with 46% of farms experiencing issues with them, and 57% of them stating that it took the maximum amount of fish. Click. So the final regulatory category that we assessed that determined the highest impact was restricted species. So there are international, federal, and state laws prohibiting the collection, culture, and movement or sale of some species. Some of these species are unique to Florida. 3 out of the 4 on this list are regulated at the state level. However, all of these species listed here have never been available for culture in Florida. So the way we asked this question was to ask the farmers anticipated sales based on market knowledge and customer requests, people calling in and asking if they could purchase these species from them. So while not many farmers indicated that they were interested in culturing restricted species, those that did report it indicated that there was a very large market potential. So some of these species we have listed here are leucistic alligator gar, Asian arowana, the ornamental varietal of Cherax crayfish, and peacock bass. Click. So while the impact of some regulations were substantial in a few regulatory categories, we observed that the actual cost of the permits, so those well permits, the bird depredation permits, export certificates, and the like, were relatively low, making up only a fraction of the total cost. So farmers across the industry were impacted at various levels, reflecting the uniqueness of the industry and culture circumstances, making it quite difficult for our regulators to regulate across the board. So because there's so many different scenarios, so many different types of culture methods, regulations are often difficult. So most regulations that we discussed were complete bans on the activity, such as bans on certain species or drugs. However, targeted bans, such as conditional uses of drugs, may benefit the industry while protecting those people in the public and the environment. So we also noticed that competitors have less regulatory restrictions than our domestic producers. So that could also be a reason for that large trade deficit that we noticed at the beginning. So labor laws are more laxed overseas, as well as drug and chemical restrictions and restricted species. So our farmers often are in a tough space when competing with them. So farmers have also begun to adapt and innovate to tackle some of these regulatory challenges. Examples of innovation are alternatives to masculization and the transitioning of culture to indoors to avoid predation. Next. So a preliminary analysis was done to assess the impacts of regulations in their categories. However, further analysis is being completed to understand the cost across these categories. So within the state of Florida, farmers work primarily with the Division of Aquaculture under FDACS, which is unique in that the process is centralized. So this model may prove an example for other states where farmers are struggling with a heavy regulatory burden and often have to deal with many different agencies. When discussing with farmers, regulations were commonly ranked behind other issues, such as access to cheap or good labor and managing costly disease outbreaks. So we noticed that regulations might not be the biggest of concerns in Florida. So growing research and new production techniques allows farmers to navigate the regulatory process and continue to grow their business. Next. So I'd like to thank the following people for their help in the project. The third member of my committee, Dr. Andrew Rapicki, as well as our extension agent here at the lab, Mr. Eric Cassiano, has been essential in making those connections with farmers here in the industry. Dr. Jonathan Van Sant and Dr. Carol Engel up in Virginia Tech have been instrumental in the development of my data and my project, as well as my funding sources, the USDA, the University of Florida IFAS, the University of Florida Institute of Sustainable Food Systems, and last but definitely not least, the students, faculty, and staff here at the Tropical Aquaculture Lab. Next. Thank you, and at this time, I'll open it up for questions.
We do have about 2 minutes for questions, so anybody is willing to speak or put it in the chat if they're so willing.
So I got a question here from Liz. So she said, interesting talk. What are some alternatives to the drugs used in masculinization? So we had a PhD student here, Shane Ramey. He graduated and now works for the U.S. Fish and Wildlife Service up in Montana. I'm not 100% familiar with his work. I know he tried some alternative uses in feeds. However, I believe most of his work is published in the Journal of Aquaculture. So you can definitely check there and take a look at some of his results.
All right, thanks, Noah. Unless anybody else is going to jump in the remaining 50 seconds, I think it would be good to try to make up some time and move on to Robert Boda's presentation. And he will be giving a 15-minute talk and is advised by Dr. Ash and Dr. Camp.
Awesome. Thanks, Kate. I appreciate the introduction. And thanks, everybody, for tuning in to my talk this afternoon. So my name is Robert Boda. I'm currently a PhD candidate in the School of Natural Resources and Environment. And I'll be talking about some work that I just recently started with Ed Camp and Lee Grove. I'm discussing the economic impacts of bass tournaments on Lake Okeechobee. Click. So I'm sure we're all aware, Florida is considered the fishing capital of the world, thanks to its saltwater and freshwater recreational fishing opportunities and the commercial fishing industry that we have here. In 2016 alone, Florida recreational fishing industries were generated about $10.9 million in sales revenue, and that in turn supported 96,000 jobs. So it's pretty evident that the recreational fishing industry is a pretty substantial part of our economy, and there's a lot of social and cultural ties to recreational fishing, a lot around the state. In particular, Lake Okeechobee is a pretty big hotspot for both casual recreational fishing and competitive tournaments. These tournaments are conducted year-round and usually organized by FWC and other organizations that put these tournaments on. And it's important to understand these economic effects of these tournaments when managing our resources and trying to understand how different types of participants utilize these resources and participate in these activities, whether or not it's just a competitive tournament or just a casual tournament. Within literature, a lot of the studies have kind of looked at the economic valuation side of recreational fishing and tournaments, and oftentimes this is looking at just the value that the participants place upon the resource and place upon being able to participate in actual fishery. And these utilize social welfare values, but we don't often look at the exchange-based values and looking at how industries actually generate money thanks to these tournaments. And that's where the economic impact analysis can provide useful. Click. So what exactly is economic impact analysis? It's an analysis conducted through input-output analysis, and really what it's trying to get at is quantifying the economic activity that is generated by different spending patterns. So if we're trying to look at how a change in an event, in a policy, or even an activity or industry, we want to see how that change affects the economy, we can conduct an economic impact analysis. And we can look at how the expenditures from these changes in activities flow through our regional economy and can support other industries and can lead to additional employment opportunities and such. And so we use these direct expenditures from a given activity that occur within a specific region, and we can calculate final metrics such as direct effects, indirect effects, and induced effects. Click. So these three effects come together and create total effects, and through the direct effects, these are the total expenditures that are being spent directly on an activity. So in our case, we're looking at the impacts of recreational fishing tournaments. So we're looking at the direct expenditures that are caused because of these fishing tournament activities. And so we also have the indirect effects, which are the inter-industry transactions that occur through different rounds of spending. And we also have the induced effects, which involve the spending of employee wages from both directly and indirectly supported industries. Click. So the direct expenditures that occur because of an activity create what we call the ripple effect. And so we have, sorry, it says indirect contributions here. It's supposed to say indirect effects, but this creates the indirect effects. So here in the top part of the figure, you can see the sales revenue that's generated because of expenditures that are caused by the freshwater fishing tournament. And so each of these expenditures are spent in different types of industries, such as the boat and vehicle repair, lodging, fishing equipment, et cetera, et cetera. And each of those industries has to spend additional money so that way they can produce their product and their goods and services. So you can see here, the first round of spending, boat and vehicle repair, gets some of that money from the freshwater fishing tournament expenditures, and then they use some of that money to pay for things like utilities, maintenance, administrative costs. And this kind of continues through the economy up until that money leaks out through imports or taxes and whatnot. Click. And so we also have the induced effects, and all these indirectly affected industries have to spend a portion of their expenditures on labor income, of course. And labor income can be spent through different household spending patterns by their employees, and this kind of results in additional economic activity. And this can include expenditures on housing, food and restaurants, entertainment, utilities, medical care. So all of these activities and expenditures kind of come together because of the direct expenditures initially caused by the freshwater fishing tournament. And that's kind of how we're getting at the economic impacts of these freshwater fishing tournaments. Click. So what we do is we conduct an input-output analysis through InPlan. And InPlan is a modeling program that is comprised of basically a regional economic structure. And what this means is that within this model, it has how each industry within that region interacts with each other. So how much input one industry needs from another to create a specific amount of output. And because of these linkages within the regional industries, we're able to put in our direct expenditures within specific industries, so that way we can see how that money is flowing through the economy and how it can generate additional economic activity. And to do this, you have to define a specific region. So here we define this 5-county economic region surrounding Lake Okeechobee, as you can see. That includes Glades, Hendry, Martin, Okeechobee, and Palm Beach County. And we also used online and mail-in survey responses from the participants of fishing tournaments during 2019 and 2020. And we were able to use these surveys to calculate the average expenditure per person per tournament type. And we were able to compare this to the 2018-2019 bass fishing tournament season and scale up our average expenditure per person per tournament type to get a total expenditure per tournament type. And one thing that is important to note here is that within the analysis, we only use the responses, the expenditures from non-local participants. And in this case, non-local means anybody that does not live within this 5-county region. And the reason being is that we're looking at the economic impact. So this means we're looking at the change in economic activity caused by recreational fishing. So if you're going to look at local expenditures, it doesn't necessarily, that would not necessarily indicate that this money is being spent in the region because of the recreational fishing tournament, because you can assume that that money would be spent elsewhere for some other reason within that region. But the non-local expenditures are coming into this county, 5-county region, because the recreational fishing tournament exists. So when conducting these types of analysis, we want to make sure that we're only looking at non-local expenditures and including that within the analysis. Click. So we have 10 different expenditure categories within the surveys. You can see them here. Includes fuel, repairs, maintenance, tournament entry, fishing equipment, lodging, meals. Our other category, it was a little difficult to assign, but most of these expenditures were like tolls and various travel costs. So we were able to put that in a specific sector. So when actually conducting this analysis with an InPlan, you want to take all of your expenditures and assign it to a particular InPlan sector, which is representative of that industry's spending patterns. And that's kind of how we're able to see how this money will flow through the economy. Next, or click. And here you can see the total expenditures per tournament. We have 5 different types of tournament: club and open, professional, semi-professional, high school, trail, and wildcat. And so the total expenditures for all 5 tournament types was $261,000. And as we'd expect, the professional and semi-professional had the highest amount of expenditures. And so we were able to use this to come up with our final results. And so here we have the direct, indirect, and induced effects for 4 different categories, including employment, labor income, value added to the region's GDP, and the industry output. And so I think it's notable here to look at the industry output, where we have the total effect of $392,000. And it's interesting because we have an imputed multiplier from this of 1.58, which is basically just the ratio of the total industry output to the direct industry output. And what this means is that for every $100 spent on or because of recreational fishing tournaments, you're generating an additional $158 in that region's economy. So while this industry output might not seem huge at the moment, it clearly does have a pretty substantial effect on this 5-county region, and it does generate a significant amount of economic activity. Click. And here we can look at the results for tournament type, and I just aggregated the total effects for each tournament type instead of looking at the induced and indirect. And notably, you can see professional and semi-professional has a total output of $219,000. And we'd expect the highest amount of output for all 4 categories in this type of tournament, just because they did have the most amount of spending. Click. And with economic impact analysis, you can also see all of the different industries that were affected by the direct expenditures. So here we have the aggregated industries that were affected by the recreational fishing expenditures. So you can see other services, retail, trade, information, transportation, warehousing, and then government industry groups all had substantial direct expenditures because most of our expenditures fell into these categories, which led to a high total output. But you can see here, professional, scientific, and technical service industry groups and all other industries had no direct expenditures, but they had a large amount of indirect and induced expenditures. So that means that there's quite a few amount of industries that are being indirectly supported by recreational fishing tournaments, which makes these expenditures and these tournaments something that is important for the local economy. It's not just affecting only companies and businesses that are related to recreational fishing, but it also is generating activity within unrelated industries. Click. So to really just wrap this all up, like I was saying, the initial expenditures caused by fishing tournaments can have a pretty substantial effect on the local economy. And it's not like we should not also disregard those local expenditures. Just because we don't use local participants within our analysis doesn't mean that the money that they spend on this, on the recreational fishing tournament should be discounted. In our case, local participants spent $39,000 on recreational fishing expenditures, which is still a pretty important and high amount of money within this 5-county region. And these outputs are important, and they can be used as a component to understand the full economic picture of fishing tournaments on Lake Okeechobee. For example, they can be used by FWC when they're trying to make future decisions, including switching priorities towards certain types of tournaments, increasing or decreasing their investment into tournaments in general, and even just bolstering relationships with their participants to improve survey and expenditure data quality, because the better data quality and expenditure data that we have, the better the results are going to be and the more accurate the economic impacts of vast tournaments are. So with that, I'll click one more time and I'll open up for questions if we have some time. If not, feel free to reach out to me. I'd be more than happy to talk about any of this with you. So thank you.
We probably have time for one question if anybody
Awesome.
wants to.
Appreciate it.
Hey, Robert, I'll ask one.
Awesome.
Nice job on that. I wondered whether your analysis factored in pre-tournament days. You know, a lot of particularly the professional tournaments, they come down and fish for multiple days before the actual event, and sometimes weeks before. And I wondered if that was factored in because it could add substantial.
Yeah, that is a pretty important part of the expenditures. And I believe that the survey did, the way that it was worded within the survey was, please list your expenditures on these different categories in this region while you were visiting for this tournament. So we might want to separate out these questions like during the tournament expenditures and then like how much did you spend just leading up to this. But the way that I read the question, it seemed like it kind of was an all-encompassing factor or figure. So.
Okay. All right. Excellent.
Awesome. Thanks, everybody.
Thanks, Robert. And we are going to move on to our final presentation for the session, which is a 5-minute talk by Lisa Chong, who is advised by Dr. Camp.
Hi. Hello. Thanks, Kate. My name is Lisa, and I'm working with Ed Camp. So today I'm going to give a methods talk about one chapter of my dissertation. So just a heads up, I did leave a lot of theory out, but hopefully I still get the general idea of the research across. Click. So I'm going to start out with giving an example here. So there's a common perception that fishers like artificial reefs. So then they'll go out there and fish at these artificial reefs, which will likely increase their catchability in terms of effective search rate. And then this will possibly lead to declines in productivity, which then managers would be obligated to make stricter regulations. And then this would also make fishers more unhappy. So this is what I mean by unintended effects here. And then, so there's a lot of research done on the ecology and the biology of fish, but not so much on the angler behavior. Click. So here our objective, therefore, is to do empirical assessments that evaluate how artificial reefs will affect fisher utility, behavior, and decisions that they make on the fishing trips. Click. So we could quantify this by using a random utility model framework. So discrete choices are modeled under the assumption that the decision maker, which in this case is the fisher, wants to maximize their utility, which is a measure of satisfaction. So fishers get some utility from each of the alternatives. Alternatives are possible choices available for fishers, meaning that different attributes, such as changing the bag limit size limit, fishing at an artificial or a natural reef, and then you can include costs and target species, and you can add so much more. So the goal is to find combinations of these attributes, and then you could also explore trade-offs between alternatives. Click. So this is the random utility model formulation, but I'm not going to go into the math today. So the important parts is that you could estimate each of these parameters and then also the total utility. But one thing to note here is that you can't estimate total utility from asking the fishers directly. It's not measurable. So then we need some kind of data to fit this RUM model. So we use a discrete choice experiment, which is a quantitative method to elicit preferences from fishers without asking them directly. And then from this, we could infer total utility and then the influence of each of the fishing attributes I mentioned in the previous slide. Click. So this is what an example of a discrete choice experiment is. So you're going to list all the attributes. I only listed 3 here. And then, yes, between trip A and trip B, they're going to change somehow. And then here, for example, the fisher said, oh, I like trip B better. And then from that, you could also rank them from each of the trip options from 1 to 10. So this is just one example of the many of these that they're going to fill out. They're probably going to do maybe like 20 to 30. So the ones that are picked for the survey are chosen based on a factorial design. And then also the overall survey can also include general demographic questions. Click. So in summary, I did kind of reverse how I introduced these topics, but first you create the survey for the DCE, and then you fit the data with the RUB model. So you expect the expected results is to gain understanding of how fishers make decisions about fishing trips with artificial reefs. So for example, you'll have empirical estimation of fisher satisfaction, which will also include estimation of how much each fishing attribute influences fisher decisions and how they contribute to the overall satisfaction. And then you could also have economic metrics such as willingness to pay. So there are other future steps that we would like to take beyond this chapter. So that would be, for example, parameterizing a simulation model. And then this would control the angler behavior and effort allocation in the simulation model. And then you could also forecast the influence of management decisions on fishers. So this is, for example, like how will changes in bag limit affect the fishers' decisions? Click. And then, yeah, if you have any questions and you wanted to learn more about the math, probably not, but you could contact me with this email. Thank you.
The revenue, and it's been about $30 million a year in the past couple of years. And it's expected to increase. Click. So this study is part of a larger collaborative project with the University of Florida, Florida Sea Grant, and Florida Fish and Wildlife. The main objective is to improve the management and harvest practices of the Florida stone crab fishery, with 3 other objectives: to integrate new data sets into population models, develop new bioeconomic models, and to increase outreach to stakeholders. This study is working towards that second objective. Click. So how do we investigate what's causing the variation in stone crab prices? Like I mentioned earlier, we're using something called a hedonic model, and that explains price as a function of attributes. They're used to estimate the influence certain independent variables have on the price of a good. These models are typically used in housing and real estate markets. And as you know, there's certain attributes of a house that can make it worth more or less. This here is the base equation for a hedonic model. As you can see, the natural log of prices in the y. There's an A term, which is the base or reference term, and then there's a combination of attribute terms and an error term. Click. So what does this model output and what does it mean? The outputs are coefficients or intercepts of independent variables that can be interpreted as weights that a buyer places on various qualities of the good. These are also referred to as premiums. Here's an example of a hedonic model for real estate. As you can see, prices on the y. The A term in this model represents the average price or base price of a house with 4,000 square feet in downtown Miami that costs about $200,000. You set the reference point. You can set any reference point, but your premium or coefficient outputs are always going to be with respect to that reference point that you set. So if you want to know the average price of a different house, say a larger house that's about 5,000 square feet in a more desirable location, such as Miami Beach, you would add those premium values to that base term A, which is that $200,000 house. And then if you had a premium of about $100,000 for a size increase, and then about $200,000 for the location increase in desirability, then you get the new average value for that house, the new house, the larger and better location one, and that could be about $500,000. Click. So how does this real estate model apply to fisheries? Well, it has been found that seafood prices are a function of attribute size, grade, or fishing technique from McConnell and Strand. When buying tuna, attributes such as the size of the fish, the fat content, grade, fishing technique, and market conditions all play a role in the price. Buyers will pay more for qualities or attributes that they want or what they infer their customers to want, therefore placing a higher premium on those attributes. Click. So what data do we use to build these models for stone crabs? We use commercial landing data or trip ticket data spanning from 2009 to 2020. Trip tickets contain a lot of information on the claws specifically sold, as well as the fishing methods and landing locations. Click. These figures here are summaries of landings by county. The map on the left is the % landings by county, and you can see that the fisheries predominantly focus in the southwest region of the state. The dark red county, including the Keys, is Monroe County, and that holds about 50% of the total landings. In the analysis, we only used counties with landings over 0.5% of the total landings, which starts in about Wakulla in the Big Bend and extends all the way down to the Miami area, with Brevard County being the only county included on the East Coast. The next figure is a heat map that shows the percentage of each grade by county, moving from north to south. As you can see, most of the claws landed are graded as either medium, large, or jumbo, and it's not evenly distributed amongst counties. Some land more of certain grades versus others. Click. Now let's look at the estimated hedonic model for stone crabs. We included 5 different attribute variables, such as fishing year, month, grade, county landed, and fishing area, or the waters that the crab was harvested in. Our A term, which is that base or reference price, was the average price of a light stone crab claw harvested in October of 2009-2010 fishing year from offshore waters and was landed in Monroe County. So all other prices are inferred based with respect to that specific A term. And we can infer any average price of any of those combinations of included variables based off that. Click. This complicated and hard to read table is the output of the model. As you can see, there's about 40 included variables. And since this is a log linear model, they all have respective coefficients, which equate roughly to the % premium each of those variables has with respect to that reference term. The main takeaway from this table is that high R-squared value of 0.821. Now let's take a closer look at the premium values by attribute type. Click. This is the all the grade attributes, as you can see, that they're all within respect to light. Is that the reference value, the largest influence on price, or this attribute has the largest influence on price. There's not much value in landing smaller light claws. As you can see that there's about a 200% premium on colossal and only about a 50% premium on small in reference to light. Click. This figure here is the fishing years with respect to 2009, 2010. As you can see, there's a decent amount of variation between years with up to a 60% variation in the 18-19 year. Click. This shows that there's generally a peak in the middle of the year with reference to October, and it slowly declines towards the beginning and end of each fishing year. Click. This figure here is the fishing area premiums. There's not much of a premium based on where the crabs were landed, with only about a 6% preference on federal waters. Click. This figure here is the county premiums. There's a decent amount of variation in where the crabs were landed. As you can see, there's generally a preference between crabs landed in the southwest region of the state, which is the right side of the graph, with only Sarasota and Miami-Dade fetching a higher premium than Monroe County. Click. So the assumptions for this model, it assumes the relationship between groups of attributes is constant. When investigated, that holds true for most groups except for months. There's been high level of variation for months between fishing year to fishing year. Possible resolutions for this is the inclusion of interaction term or splitting the model to run fishing year by fishing year. Click. This figure here shows each model is run year by year by year, with which each of the models are run with respect to October. And the general trend follows where the winter months of January and February fetch the highest premiums in the beginning and the end fetch the lowest premiums. This black line is the total landings. And we theorize that the years that fetched lower landings, lower premiums had higher landings, which shows generally true. Click. And in conclusion, this was the first attempt to analyze the influence of price variation in Florida stone crabs. Grades have the highest influence on price. Light and small claws, counties furthest away from Monroe, and the start and the end of the fishing year fetch lower premiums. There's significant interannual variation for the monthly attribute, and this model can be used to assess economic influences of different management strategies. The next steps we're going to take are to explore demand relationships and population dynamics of this fishery. Thanks. That's it.
With that, we have a few minutes for questions while I pull up our next presenter's presentation. So Liam, great job. If anyone has a question, go ahead.
Hey, Liam, I have a question. Do you have any theories as to why the premium is so high in the middle of the season as opposed to the beginning and the end?
Talking with some of the members of FWC, they had a general idea that the price in October, which is the reference point for what those premiums are based off of, kind of sets the precedent for the season. And depending on the landings, as you can kind of see in that one figure, if there's a really low premium on October, then there will be a lower premium on the rest of the year. And there's generally less crabs landed in those colder months. So since there's less crabs, there's going to be people are going to be willing to pay more for the fewer crabs that are available.
Awesome. Cool. Thanks.
Liam? The Federal Reserve calculates a parameter called disposable income for individuals. And that disposable income influences a lot of our marketing. So for example, in the housing market, Floridians may be able to pay X, but all those folks moving down from New York with higher disposable income are paying outrageous prices. Same with our seafood. Some people, certain markets may be able to buy X amount or such a price, but a rich community, higher disposable income could go higher. Are you able to incorporate that concept in your modeling?
So It's not directly incorporated, but we are theorizing that the counties like Miami-Dade and Sarasota, which generally have wealthier populations than counties on the Big Bend and around the West Coast, the reasons they fetch the higher premiums are because they can be sold for more in those regions. And that's kind of how that is interpreted.
So I hate to cut a good conversation short, but for the sake of time, any additional questions that we want to ask Liam, please direct to him via his email or any of the other things he's provided for contact. Thank you again, Liam, for your presentation. Our next speaker is Christina Malika, and her advisor is Dr. LaCours. Christina?
Hi, everyone. My presentation is going to be on my master's thesis research proposal for using long-term photomosaics to investigate ecological and physical patterns of colony success among coral species in the Florida Keys. Click. So this study will take place along the Florida Keys reef tract. The keys themselves are split into 4 distinct regions: upper, middle, lower keys, and the dry tortugas, which includes 3 marine protected areas. So the coral reefs here provide numerous ecosystem services, such as their biodiversity hotspot. They offer coastal protection, provide nursery grounds for commercially important fish species, and support local economies. Click. However, the reefs are in decline globally and in the Keys as well. This decline is a result of anthropogenic threats at both the regional and global scale. So here's a timeline of regional disturbances since 1996. Several temperature anomalies were observed in addition to storms and the stony coral tissue loss disease outbreak. So these disturbances are occurring frequently with little to no recovery periods in between. Click. So you can see this here. This data is from FWC's Coral Reef Evaluation and Monitoring Program, also known as CREMP, which has monitored these reefs annually since 1996. So on the left-hand side, you see percent cover of stony corals changing through time. So this percent cover has decreased from 12 to 6% keys wide since 1996. While on the right-hand side, you see the percent cover of 3 spatial opportunists, macroalgae, octocorals, and sponges. These 3 groups have either grown in abundance or their populations have remained pretty stable throughout. Click. So the evidence of coral populations in the Keys degrading over time is ample. However, the remaining colonies that have persisted offer an important story to tell. Click. One more ahead. So because of this rapid decline in corals, condition indicators previously used, such as percent cover and individual coral health, are no longer sufficient in reef monitoring. So this is where mosaics come in. A photomosaic is a large landscape image comprised of usually thousands of individual and overlapping images digitally stitched together. Typically, this is done with the program Agisoft Metashape. Click. So in the end, this is what that looks like. You get one large-scale image of the seafloor without compromising resolution. This allows for a more ecosystem-based approach to reef monitoring without sacrificing accuracy, time, or money, along with the added ability to look at 3D attributes of the seafloor. Click. So my research question is, what ecological and physical characteristics are associated with the remaining colonies in the Florida Keys? To do this, I will use photomosaics created from historical and contemporary imagery to locate and fate track large colonies. Then I'll characterize the natural process of recovery for surviving colonies or recruitment post-mortality. Finally, I'll identify physical characteristics that could be attributed to colony success. Click. So first off, FWC's photomosaic archive encompasses about 50 sites throughout the region. The mosaics were done in 2001, 2004, 2009, and then the past 2 years. So the earliest mosaics in 2001 will be used to locate the large colonies that appear healthy to be fate tracked throughout the analysis. Click. Once the colonies are identified, parameters will be collected, including max diameter and height, percent cover of disease, recent and old mortality, and any identifiable stressors. Then 1 meter by 1 meter quadrats will be placed on the colony to encompass the live tissue and the surrounding benthos. Percent cover for present species will be calculated for each quadrat, which will be placed in the same area throughout all the mosaics. Click. Finally, for each mosaic, the resulting digital terrain model will be used to calculate 3D terrain attributes of each colony, including rugosity, slope, curvature, and relative vertical position using ArcGIS. So statistics will be used to compare the 3D metrics of these attributes for the tracked colonies to tease apart any pattern by which placement on the reef could influence colony success. Click. At its fullest extent, this project will provide a workflow demonstrating how photomosaics allow for the transformation of spatial geomatic data into information. More specifically, this type of information is beneficial to current restoration efforts in this region. Understanding the complexities of what drive colony success will enable restoration projects to mimic the ecological characteristics and seafloor attributes that most benefit the coral alg plants. Click. So that's all I have. I'd like to thank my committee, my lab mates, and then my coworkers on FWC's coral team. And there's my email if anybody would like to ask me any questions. Thank you.
Thank you, Christina. Excellent work. Our next presenter is Daniel Villas-Gonzalez, and with his advisor, Dr. Chigueras. Sorry if I messed that up. After you, Daniel.
Hi, everyone. My name is Daniel Villas, and I'm a PhD student under Dr. David Chigueras. And today I will talk to you about the effects of red tides on the West Florida shelf, and I will focus on the gag grouper. This study is part of a big ecosystem modeling project funded by the NOAA Restore Science program. Click. The West Florida Shelf is a region located in the northeastern Gulf of Mexico. It fosters a highly diverse fish communities and it supports a valuable commercial and recreational fisheries. But it faces many pressures at different temporal and spatial scales, such as overfishing and red tides. Therefore, it is important to evaluate those impacts in order to anticipate, mitigate, and better understand perturbation effects under an ecosystem-based fisheries management framework. Click. Currently, one of the most concerned perturbations in the West Florida shelf are harmful algal blooms. Harmful algal blooms or red tides consist of the rapid proliferation of unicellular algae species, here most commonly caused by Karenia brevis. Those events are originated by a combination of multiple factors like dust deposition or upwelling events. It caused negative effects on multiple sectors, tourism, human health, fisheries, and ecosystems. For example, ecosystem modeling studies suggested changes on community structure and cascading effects on the food web when considering harmful algal bloom effects. Click. Ecosystem management is a holistic approach that considers multiple interaction, trophic, environmental, and anthropogenic. Among all ecosystem management levels, ecosystem-based fisheries management is focused on fisheries systems. In 2016, the NOAA released a roadmap for the ecosystem-based fisheries management implementation. And ecosystem models can serve as potential tools to achieve ecosystem-based fisheries management. Among all ecosystem models, EcoPath with ecosystems supports ecosystem-based fisheries management and is the most applied tool for modeling marine and aquatic ecosystem. Special ecosystem models are increasingly applied for management advice because some fisheries management plans require a special trophic approach. Ecosystem models can be combined with single stock assessment models to assist in decision management processes. For example, the last Cedar GAG stock assessment incorporate ecosystem considerations such as red tide effects. Click. So the main goals of this study were to improve the spatial temporal ecosystem modeling tools for management advice by incorporating disturbances, to investigate the red tide mortality rates in gag grouper in the West Florida shelf, and to evaluate whether spatial temporal ecosystem models can capture the signal of red tide impacts at population and individual levels. Click. To model the West Florida shelf ecosystem, we use the EcoPath with Ecosyn software that is composed by three main components. The EcoPath, that is the static version, provides a snapshot of the ecosystem and simulates the food web by representing functional groups that exchange matter and energy, while assuming a mass balance between production and consumption. Ecosyn, that is the time dynamic model, represents the functional groups over time using differential equations that represent biomass fluxes. And ecospace, that it represents, predict the biomass of those functional groups over a two-dimensional spatial grid. The ecospace incorporates an habitat capacity model that calculates the suitability index for each cell in the basement and for each functional group. This suitability index is calculated as a product of multiple response functions to multiple environmental factors. The habitat capacity model allows to drive the functional groups on the base map through the foraging capacity. The ecospace also incorporates the spatial temporal framework that allows to incorporate geospatial information and makes the model fully spatial and temporal. Click. The West Florida Shelf Ecospace model is an updated version of Chagari's 2013, and it simulates over a 10 minutes spatial resolution over 34 years a modeling step. It includes 83 functional groups and 17 fleets. It incorporates multiple environmental variables, and it also includes environmental response functions that were computed by fitting binomial generalized additive models. The model stability was checked by a stepwise procedure in which we ensure the persistent and the non-exponential increase of functional groups over time, while increasing the complexity in terms of the incorporation of environmental variables in the model. The model validation was achieved by a spatial comparison between the long-term ecospace predictions with the spatial predictions from binomial generalized additive models. Click. The red tide effects were simulated by first computing the red tide severity maps. Those maps were calculated by kriging and clipping fluorescence maps and cell concentrations monitoring data from FWC. And then because the lack of empirical information on the red tide effects, we use and test different mortality and foraging response functions, which are shown on those graphs here. And by the combination of those multiple mortality and foraging response functions, and by applying mortality and foraging response function and applying those functions to consumer groups, to all consumers groups, or just GAG groups, we end with 160 runs. And those 160 runs, we use them to calculate the red tide mortality rate for GAG grouper, and also we calculated the GAG vulnerable biomass indices. We also evaluate the red tide effects by a spatial biomass evaluation. Click. So these are the results for the red tide mortality rate. And the figures show that the year 2005 was the most severe year. And also it pointed out the GAG5 plus as the least impacted GAG group among all GAG groups, which may indicate that the higher the spatial overlap with red tide events, the higher is the mortality. For example, those maps indicate spatial predictions for the other GAG groups before the red tide event and after the red tide event, and in the inside the red ellipses that shows where the red tide event happened, we see a decrease on biomass. Click. The GAG vulnerable indices are shown in this figure. In this figure, the red line represents the ecospace prediction when including red tide effects. The blue line represents the GAG stock assessment prediction. The dots represent the observed values, and the gray line represents the ecospace predictions without including the red tide effects. And we see that the red line, the ecospace prediction when including red tide effects fits pretty reasonably well the data. And also it got similar results to the GAG stock assessment. If we take a look on the H0 index, it shows that it's pointed out with the red arrow. We see an increase on recruitment after year 2005 because of the big red tide event. And this peak was more noticeable when we include red tide effects to all consumer groups. And this is telling us that while stock recruitment relies on fishing and recruitment deviation to explain this variability, the West Florida shelf ecospace model relies on fishing and red tide effects. Click. The evaluation on the effects of red tides especially were done by extracting the biomass through along a transect that captured the red tide event on 2005. This is shown on the map as a black line. And if we take a look on the figure shows a green rectangle that represents the impacted area. And those lines represent the biomass prediction from ecospace through this transect. So if there are two kinds of lines, the solid line was when we apply only mortality effects, and the dashed line represents when we applied mortality and foraging effects to red tides. So the darker, if we take a look on the darker, if we focus on the darker lines, we see that there are no biomass on the green rectangle. So it's showing us the mortality effects on the red tide impacted area. And if we see the adjacent areas, we see that the dashed line predict higher biomass values than the solid line, which indicate us that the model was able to capture fish migration and avoidance to red tide impacted areas. This movement was able to be captured because we include the mortality and foraging responses. And this was captured just not just for GAGs, also for other groups like sardine and reef associated species. Click. So to conclude, our model was able to show that the implementation of disturbances on spatial ecosystem models can produce models more reliable and robust to achieve ecosystem-based fisheries management. This study set a step forward to make operational spatial ecosystem models. The West Florida shelf ecospace model showed that red tide effects were shown at stock and individual level. And while recruitment compensation effects is likely driven by food web processes when including red tide effects, we were able also to capture the avoidance that allowed to mitigate biomass losses due to mortality. And finally, it shows that there is a lack of spatial temporal information on red tide at fine resolution to capture empirical negative effects for validation and parameterization. Click. And I would like to finish by acknowledging my advisor, Dr. David Chagarias, for his support, and Joe Bukowski for the implementations of new features on the software, and also Zach Siders, who helped us with the environmental information. Sorry, Zach, because I forgot to mention you here in this slide. And also thanks NOAA Restore Science Program for the funding. Click.
Thank you, Daniel. We have time for one quick question.
Okay.
I got one real quick.
Yeah.
Zach. Hey, really cool work, man. It's awesome to see this. I was curious, could you use this as a kind of forecasting tool to say, well, what would the red tide have to be to really generate huge effects, right, down the line? Because that might be useful on the back end for them to kind of see how things are going to change down the line.
Yeah, I mean, Ecosim and Ecospace are able to forecast like several years, and we can incorporate different set of maps like predictions on those special predictions to allow to investigate those effects on a near future. And this is done also with climate changes scenarios. So I think that it's feasible to do it with red tide predictions.
Awesome, thanks, really cool work.
Thank you.
Thank you again, Daniel. Our next presenter is Dylan Sinnickson, again with his advisor, Dr. Chigueras. Daniel? Oh, I'm sorry, Dylan.
Hi, everyone. My name is Dylan Sinnickson. I'll be presenting upon the ability to use empirical dynamic modeling to assess ecosystem dynamics of estuaries. Click. So our selected study site was the Suwannee River Estuary, and we attempted to assess the impacts of freshwater discharge on estuarine fish productivity. We also wanted to analyze time lags in productivity between a series of different low trophic level taxa. And we wanted to answer the question, how long would it take for changes in discharge to be observed in populations of anchovies, which are an ecologically important forage fish within the estuary? Click. So in order to model trophic dynamics and ecological productivity, we utilized EcoPath with EcoSim, which is a frequently used ecological modeling software for aquatic ecosystems. And this served as our operating model, so we simulated data for use in our EDM assessment. The functional groups that were used in the model range from top predators to planktonic species. These groups are linked together from known diet compositions, and biomass is estimated from the FIM sampling program of FWC. These groups are linked together by a series of differential equations, and they're based on known consumption rates and prey availability. It's also important to note that the only trophic levels that we analyzed were the links between nutrients to phytoplankton, to zooplankton, and subsequently to anchovies. We also used fisheries dependent data collected from the EMIRT program of NOAA, and our model was fit with total nutrient concentrations from the Project Coast sampling program. Click. So moving on to the empirical dynamic modeling assessment, EDM is a nonlinear statistical technique which analyzes chaotic time series data, and time lags of a response variable are used to model future changes. It's been used extensively in fisheries, medicine, finance, and other fields, and it's based on a state space modeling approach. So the dynamics of a response variable are dependent on the state of the system. And with respect to EDM's use in fisheries, it's been used particularly to assess recruitment dynamics, and recruitment is predicted by spawning stock biomass and environmental factors. In our analysis, EDM served as the estimation model. So we estimated time lags from data that was simulated in EcoSim. Click. So we had 3 different scenarios to test the efficacy of EDM. We used, in all these assessments, we identified the lags between nutrients to phytoplankton, to zooplankton, and subsequently to anchovies. In our first scenario, this was a no error scenario. So this was just purely using EcoSim output. In our second scenario, we had estimates of observation error, and we multiplied that to our EcoSim output. And we used coefficients of variations of density estimates to give us estimates of observation error, and these are based on empirical measurements or literature reviews. In our third scenario, we used real data collected from the Project Coast sampling program and FIM. Click. So these are the results from our EDM assessment, and in general, we found that EDM is very effective in analyzing time lags for deterministic systems, but when you add stochasticity, EDM's efficacy is significantly reduced. So the black line represents our first scenario, pure EcoSim output. The blue line represents the EcoSim output with observation error, and the red line represents the real data scenario. And as you can see, generally, rho values were highest with the pure EcoSim output. So In a deterministic system, you can have high statistical significance when you're only using pure ecosim data, but when you add some degree of observation error to that blue line, you generally are going to reduce your statistical significance. So in general, we saw a pattern of increasing time lags moving up the food web. Lags generally range from 1 to 6 months, and these relationships generally break down when you add observation error. Click. And here are my references, and click also to my acknowledgements. Thank you.
Thank you, Dylan. That was wonderful. Our next presenter is Nicholas Fish with his advisor, Dr. Ed Camp. Just one moment. I don't seem to have that presentation.
I have it pulled. Okay, Scott, do you have it? Okay, you're in like the middle of the presentation, Scott.
There we go.
Just change to the different display setting.
Is that good now? All right, I mean, I can go from here.
It's like really, there you go.
Hi, everyone. Thanks for tuning in today. Click. So my research focuses on how we fit age composition data in fisheries assessments. And so when I say age composition data, what I'm referring to is over the total harvest for a given year, what were the proportions caught at age? Click. And these data are critically important to modern fisheries stock assessments like catch at age assessments because they provide all sorts of information on relative year class strength, so on recruitment, on mortality, so both natural and fishing, and on vulnerability to fishery. Click. Now, the way these data have been historically fit in stock assessments has been using the multinomial likelihood. Now, the multinomial likelihood assumes that your samples are independent and identically distributed from a population of interest. Now, in this case, our population of interest is the harvest for a given year in a fishery. Now, the issue lies in that in reality, rarely is our sampling as elegant as this IID assumption might imply in fisheries. Rarely do we have a harvested or final pool of harvested fish at the end of the fishing season to take a nice IID sample from. Instead, what occurs, click, is that we're taking samples throughout the year from many different ports, many different vessels, maybe even different hulls within vessels. What occurs when we're sampling these nested groups is we're not necessarily getting a nice IID sample that fishery harvests for the year. Instead, what we're getting are samples that are correlated both in space and in time. Click. And this occurs because fish don't distribute randomly over space, and neither do fishers. Fish tend to associate with other fish of similar sizes and ages. And so I think, at least for me, a convenient concept to help wrap my head around this is called intra-haul correlation. It's based on the idea that fish caught in any given haul are more similar to each other in size and age than fish caught from a variety of different hauls. You can imagine if our bottom trawler here was to go to port, made a port sample there, ready to sample 20 of his fish. If we were to fit those 20 fish in a stock assessment using the multinomial likelihood, we'd inherently be assuming those were 20 fish IID sampled from the harvest for that year. When in reality, those were 20 fish correlated spatially and temporally. Click. And so the effect this has is we get these correlations in our age composition data sets, positive correlations between bins that are close together and negative between bins that are far apart. So for example, if you were to sample a lot of age 5s, you were more than likely to sample a lot of age 6s and a lot of age 4s, and less than likely to sample a lot of age 12, say. And this causes overdispersion, which is based on the idea that the variance in your data is much greater than what you'd expect it to be given the number of fish you sampled. So effectively, your age composition data have less information than what you'd expect them to, given that sample size. And this is important because it can affect our point estimates and the precision of those estimates we get from our assessments. And ultimately, if we're making management decisions based on these models, which we often are, it can affect the sustainability of the resource being managed, or we could be underutilizing the resource. Click. Click. So based on this, we're sort of curious, is there an alternative likelihood we could swap in for the multinomial that might better be able to account for the fact that these data contain correlations and are overdispersed relative to those multinomial assumptions? And there's been a variety of them suggested in the literature. However, very few of them have received simulation testing. And so we're particularly interested in, with this study, is trying to simulate the issue or some of the mechanisms we think give rise to a majority of the issue, and that's the spatial distribution of fish and that of fishers. And then if we were able to generate sort of overdispersed composition data, we could fit that in more standard stock assessments under a variety of likelihoods to see which does best at recreating the true dynamics of the system. Click. So that's what we did. We developed a spatially explicit operating model based on the life history of red snapper on the Gulf Coast of Florida. And so the way this model works is that you have red snapper that move as a function of age based on preferences for substrate type, depth, density of fish in a cell, and then fishers chase around those fish based on a gravity model, which says that you're more likely to fish cells with high abundances, and you're less likely to fish cells that are far away from your port. And there's a port placed along each coastal county there. And so the key to this is that because we went with a spatially explicit framework, what we can do is we can zoom in on any one of these spatial cells and sample the catch at age from that specific cell, which would be analogous to sampling, say, those 20 fish from our bottom trawler to be able to create those overdispersed samples we see in the real data. Click. So once we had that operation model parameterized, what we did was we ran 1,000 replicates of it, which gave us 1,000 different data sets. Then we fit those using what I'm calling more standard stock assessment models. So these are one stock spatially aggregated assessment models. And some of the details are listed there. I won't go into them. But what I will mention is that they are guaranteed to be misspecified in their fishery selectivity. So if you remember from and that's because we're fitting a one stock model to system dynamics that came from a spatially explicit model. And if you remember from Intro to Fish Population Dynamics, your fishery selectivity is going to be a function of both the contact selectivity and the spatial availability of fish. And in a spatially explicit framework, the spatial availability component becomes a function of how fish move and how fisher effort distributes. And that's going to dynamically change each year. So what we found is that with our operating model, the true selectivity pattern was time varying. And I've got a depiction of it there on the bottom right in light blue. Are the true selectivities from a run of the operating model, which get darker as the time series goes on. So you can see that descending asymptote on the right-hand limb decreases as the time series goes on. Whereas in the assessment model, we're just going to be approximating that with a double logistic form there in red that sort of approximates that shape, but isn't necessarily able to capture those year-to-year deviations in selectivity. And so we're going to be comparing these models against one another using depletion and exploitation rate that are estimated in the terminal year of the assessment. Click. So the main treatment for this study is we're looking at different likelihoods for fitting those overdispersed composition data within the assessment, specifically with respect to two desirable qualities. One, does it have an estimable weighting parameter? So that would be a weighting parameter that essentially is able to recognize that the data contain less information content than the sample size implies and is able to accordingly downweight that data within the assessment. And two, is it able to account for correlations? What I mean by this is if we were to sample from that distribution, would we get those positive correlations between bins that are close together and negative between bins that are far apart that we see in our actual data? And so there were 4 likelihoods that I'm going to talk about that I tested within this study or within this talk. One is the multinomial, which, as I mentioned, has been historically used in assessment. The second there is the iteratively weighted multinomial, which has been sort of the state of the art in the last 10 or so years, where you fit an assessment a number of different times, each time changing your sample size of your composition data using what's known as an effective sample size to essentially reflect the fact that you don't think those composition data contain as much information as the raw sample size implies. Then you have a Dirichlet multinomial, which has an actual overdispersion parameter which can be estimated within the assessment that essentially accounts for the fact that the data are overdispersed. Then you have a logistic normal, which also has an estimable weighting parameter, and through parameterizing a variance-covariance matrix is theoretically able to account for the correlations we see in the real data. Click. And so the two additional treatments I'm going to talk about today, we varied the number of fish age that we gave to the assessment. And so as you increase the number of fish age in the assessment, what it's going to do is it's going to try extra hard to fit those composition data because it thinks they're a higher quality data source at the expense of fitting other data sources like harvest or indices, catch indices. Click. The last treatment I'm going to talk about is we sort of played with the degree of model misspecification between the assessment model and the true simulation model. And so we have a case called baseline, which I previously described, where we have those true time-varying selectivity pattern in blue in the background, and we're approximating the shape of that with a double logistic functional form that won't be able to capture the year-to-year deviations, but gets the general shape right. Then we have a case called maximum process error, where we're more so deliberately misspecifying the fishery selectivity, this time using a simple logistic form, which you see doesn't do nearly as well of a job at recreating that shape. And finally, we have a scenario called minimum process error, where I went back into the operating model and said, instead of fishing according to the gravity model, you can fish randomly over space, at which point the spatial or the fishery selectivity collapses down to whatever you set as your contact selectivity, which in our case was a simple logistic. And we're also fitting that with a simple logistic in the assessment model. So this model is effectively correctly specified. Click. Okay, so some results. This is for the baseline scenario. What you're looking at are each panel's or plots a different level of sample size. In black are results for exploitation rate, in blue depletion. And these are relative errors, so the closer you are to zero or that dotted line, the better. And these are interquartile ranges. Click. So what I'd like you to, you go back. What I'd like you to key on here is that at small sample sizes, the iteratively weighted multinomial perform best, followed by the Dirichlet multinomial. However, as we increase the number of fish age, the logistic normal actually perform better than the other likelihoods. Click. Click. And so we move to our maximum process error case, where the fishery selectivity is more so sort of misspecified. What we found is that for all sample sizes and most of the performance metrics, the logistic normal perform better than the other likelihoods, followed by the Dirichlet multinomial. Click. And then we have the case where our models is effectively correctly specified. Click. And what I'd like you to key in on here is that at small sample sizes, the logistic normal performed actually quite poorly, where the other models were effectively unbiased. However, when we did increase sample size, the logistic normal seemed to become effectively unbiased like the other models. Click. So some of the patterns we're seeing here, it seems that when there is process error or significant process error, as there was in the baseline and the maximum process error case where selectivity is misspecified, the logistic normal for the most part seems to perform best across all the different likelihoods. And we think this is occurring because through its ability to incorporate a variance-covariance matrix, it has a specific variance and covariance associated with each individual age bin within a composition data set, so it can target their influence more directly through recruitment deviations or fishing mortality, whereas all the other likelihoods have to weight the entire composition data set at once effectively. However, they do seem to perform more poorly at smaller sample sizes. It may be that you need a sufficient number of fish age to even characterize that matrix, and yeah, there may be too much noise when the sample size is small. The other thing I'd like you to key in on is that although the Dirichlet multinomials, they were bested by the iteratively weighted multinomial in the baseline case, they did outperform it in the maximum process error case. And I say this is encouraging because Previously, the state of the art for the last decade or so has been the iteratively weighted multinomial. However, more and more assessments are switching the Dirichlet multinomial because the iteratively rating process is so inefficient. And so it's encouraging that they produce better results in some cases and similar results in others. And we're effectively unbiased when the process was correctly specified. Click. So with that, I'd like to acknowledge my funding sources and my partner institutions.
Thank you, Nicholas. That was a great presentation. If anybody has any questions, we have time for one brief question.
That's really cool work, Nick. What would be the challenges for operationalizing the logistic normal model for stock assessments? Would you foresee that happening, being used, especially in these high data fisheries in the Gulf of Mexico?
Yeah, I think it might need a little more testing just because of how it's a little worrisome that it doesn't perform well at small sample sizes, and specifically when it was correctly specified, it performed quite poorly. But yeah. I mean, they do have it in their canned package in North Carolina, the Beaufort assessment model package. They do have it coded in there. It's not in stock synthesis yet, but as far as is it ready to go for assessments, it is. But I would argue it needs a little more testing maybe, and yeah. But you're right, in those fisheries that sample a ton of fish, may be prudent to switch over.
Yeah, that's really cool.
Thank you, Nicholas. Our next presenter is Trenton Aguilar with his advisor, Dr. Allen. After you, Trent.
Great, thank you. Hello, everyone. My name is Trenton Aguilar, and I'm a third-year PhD candidate working under Dr. Mike Allen. I'm currently working on creating an encounter rate model for green turtles and recreational boaters in Florida. And today I will be giving a quick overview of this project, some preliminary outcomes, and my next steps. Click. I'll begin with some background on vessel strikes and why I chose this topic for my dissertation research and this project specifically. Click. Vessel strikes have been an issue for marine animals for quite some time. Studies have observed many different species incurring injuries or death due to vessel strikes around the globe. Here in Florida, vessel strikes have been an issue for various species, and more recently have increased in my species of interest, green turtles, or Coloniomytis. Florida's manatees suffered very high mortality from vessel strikes. However, thanks to increasing statewide management, vessel strike-related mortality has been reduced. Using this as a motivation and a springboard for green turtle management seemed like a logical next step for me. Click. Which led me to an encounter rate model that was created to simulate encounter rates between manatees and recreational boaters and help optimize these management studies. Click. First, I'd like to thank Dr. Julian Martin, who originally created this model and has helped me to modify it to incorporate green turtles. This model allows for the calculation of encounter rates, the number of expected encounters between one animal and one boat, and the expected number of encounters between one animal and a number of boats over a number of days. It takes into account the size of the animal and the boat, the study site's dimensions, the speed of the animal, whether that is fixed or incorporating uncertainty, the boat speed, and the time or distance traveled by the boat. Click. So here are some preliminary outputs from the model. In this case, I've chosen to keep most of the parameters from the original manatee scenario, only changing the animal-specific values with data for green turtles found from literature reviews that I have done. As you can see, the outcomes are very similar. Both animals experience a heightened encounter rate with higher speeds, which leads to higher expected number of encounters. This is using a fixed time scenario for the boats in the study area, as that is how most recreational boaters use their boats and their time, rather than traveling a fixed distance like, say, a commercial vessel would. Click. So now that we have been able to show that the model can output effective results similar to its previous use for manatees, it can be used to further simulate green turtles specifically. I will include site-specific parameters for areas of interest for green turtles in Florida. My goal is to create a management map to show areas of high encounter rates with varying degrees of densities for both turtles and boaters. Ultimately, I plan for this work to help management policy and to increase protections for green turtles in the state. Click. Thank you.
Thank you, Trenton. Our final speaker today is Kent Semin, and he is advisor Dr. Canfield, and he will round out our session 4.
Hey, everyone. My proposed research emphasis will be on the promotion of healthier aquatic environments with nanobubbles. Click. So generally, water bubbles can be categorized into 3 major types, ordinary or macro bubbles, then there's micro bubbles and nanobubbles. The diameter of a macro bubble ranges from 100 microns to 2 millimeters, and these bubbles quickly rise to the surface of a liquid and collapse, while micro bubbles are smaller than macro bubbles. They have a diameter range of 1 micron to 100 microns, and these bubbles shrink in the water and then dissolve into it. In contrast, nanobubbles are extremely small gas bubbles that have several unique physical properties that make them very different from normal bubbles. Nanobubbles that are less than 100 nanometers in diameter randomly drift in what's termed Brownian motion, and with a lower buoyancy can remain suspended in liquids for an extended period of time and have the ability to change the typical characteristics of water. Click. Okay, you can go ahead and click. I apologize for all I probably should have not animated this one, but you can go ahead and click 10 times. I'll go ahead and show these bullet points. I just wanted to highlight the history of the research with nanobubbles. It's quite interesting, actually. And I'd like to point out, as recently as 1997, the existence of nanobubbles was dismissed based on expected lifetime. And despite subsequent studies, there are still controversies over nanobubble size and their existence in solutions under atmospheric conditions. According to classical thermodynamics theory, nanobubbles cannot exist or be stable due to the limitation of radius curvature. So the small radius of curvature gives nanobubbles a higher internal pressure relative to the external pressure, which should lead to rapid dissolution. As an example, a nanobubble with a radius of 100 nanometers should have an internal pressure of about 30 atmospheres. Click. Go ahead and click to the next slide. Yeah, you can go ahead and click through to the next slide. There we go. Okay, so this is the current configuration. We are beta testing on 110,000 gallon batches of our marine aquarium water at Epcot here at Disney. It's comprised from left to right of an Onsite carbon ceramic membrane, a nanobubble diffuser. You can see in the upper left in the trailer there. Ozone generator is next to it in that white box. And then we've got an air compressor down on the floor in the back. And in the forefront on the right, you can see the oxygen delivery tank. Okay, you can click. This is an animation, if we can animate it. I don't know if the video came through. But anyway, the video shows 100 nanometer oxygen nanobubble formation of less than 100 micron microbubbles are sheared off of the membrane upon protrusion at the surface, and that's how they're formed. You can click. So higher DO levels can be achieved and sustained with nanobubbles than by conventional aeration methods, as you can see by this chart. And so it's less conformed to certain saturation levels that we have with normal bubbles and normal aeration methodologies. You can click. In this study, you can see after a dosing duration of 30 minutes, both the persistence of oxygen nanobubbles with sustained elevated DO levels were documented for up to 70 days. So both the existence of the nanobubbles themselves, as well as the sustained high DO level was sustained for up to even 70 days, which is pretty amazing. You can click. Ibina et al. 2013 demonstrated the growth promotion of sweetfish and rainbow trout with just air nanobubbles. So you can see here the N weights are considerably higher as compared to just normal water, comparing to air nanobubble water. You can click. Okay, so this is, yeah, looks like it will automatically go. It's a proposed nanobubble treatment configuration for our 5.7 million gallon marine aquarium here at Epcot. Basically, nanobubbles can be applied in many different ways. This is a proposal for possibly doing it both in our aeration tower as well as in foam fractionation, since it aids in that process. Click. This here is another animation that shows the process water flow rate, and flow direction as it goes through sand filtration from the aquarium. It goes through side stream chemical treatments and different types of water conditioning, disinfection. And then finally, the dirty water goes to a backwash reclaim system, which is completely separate, and that's what we're experimenting on currently, separate from the main system. Click. And these diagrams just show how these nanobubble ozone treatments can aid in both the nitrogen cycle, as you can see here, for enhanced biofiltration, and click. I'm sorry, you can click through that. Okay, and then for our phosphorus cycle, for enhanced biofiltration methodologies in phosphorus cycling as well, with the oxidation of both particulate and dissolved phosphorus. That's it.